For the next subject matter in this course, we have a two-part lecture series on the topic of acute muscular fatigue and the fundamental science of conditioning, often referred to as endurance training. So first, let's do a brief overview of basic terminology here. Acute muscular fatigue. What do you think this is referring to? We should all know by now the term acute simply refers to something that is immediate and temporary. So immediate and temporary muscular fatigue is the topic here. But what is a bit more complex is this term fatigue. Now, if I asked you what is muscular fatigue, most of you will simply say your muscles getting tired, like when you exercise hard. Not a bad answer, but not a technically sound answer. This term, acute muscular fatigue, in this lecture series and in exercise science, is used to describe a situation in which muscle contractions and therefore muscular force production are temporarily inhibited due to the presence of a stressor during heavy exercise or high to maximum intensity exercise. Now let me give you a practical example of acute muscular fatigue. When you perform a Wingate test, which many of you have performed in exercise physiology lab, you perform an all-out cycling sprint against a bit of resistance for 30 seconds straight. You initially accelerate and ramp up until you reach peak RPMs or peak power. Then what happens? No matter how fast or how hard you are trying to sprint, you are going to slow down. The RPMs and thus power output start to drop steadily or sometimes rapidly depending on the individual. And this usually starts to occur within about 5 seconds of this all-out effort test. So why does this occur? Why does the power output during this test eventually decline despite putting forth maximum effort? It's not because you want to decline the power output. You are trying to cycle as hard as you possibly can. It's because something is inhibiting your muscles from contracting and producing force at its maximum capability. Something is literally slowing you down. More specifically, quote unquote, slowing down the muscles, putting the brakes on the active muscles, in other words. This is an example of acute muscular fatigue. Here's another example. Imagine performing a set of loaded back squats. As you perform one rep after another, we all know what will eventually occur. We know that the movements will become slower and slower, and we can tell that our muscles are not putting out as much contractile force as it did on the first rep. This is again, despite the fact that you are trying as hard as possible to move that weight. Something is inhibiting the muscle from contracting as hard as you want it to. Eventually, you get to a rep where you could barely push the weight up. So the question is, what is inhibiting your muscles during these situations? And more importantly, why? So in other words, how does acute muscular fatigue work and why does it exist? These are the questions that we will answer in part 1 of this lecture series. Now, some of you may be wondering, isn't fatigue simple? Isn't it just muscles getting tired? No, it's not that simple, both in the mechanisms underlying fatigue and the biological reason as to why we experience fatigue when our muscles are working really, really hard. Some of you may believe that whenever your muscles fatigue, like during the sprint or during the set of back squats, it is simply because your muscles have ran out of fuel, and thus energy for muscle contractions are depleted. We will explain this further later, but no, that is not the reason why we fatigue, say during a sprint or a set of back squats. We have plenty of fuel in the tank after a single sprint, or even during the sprint. So running out of fuel is not what explains acute muscular fatigue and why we slow down during an all-out sprint or, say, a set of back squats. Some may even say it's because the muscle cannot provide enough ATP, which we all know is a cell's energy currency to power the muscle contractions. In other words, we run out of ATP. Nope, that is not the explanation for acute muscular fatigue. In fact, as I will discuss later, our muscles have the ability to meet ATP demands no matter how hard you are exercising. Even during that Wingate test, as your RPMs are declining due to fatigue, the muscle's ATP demands are being met. So what is the explanation then for acute muscular fatigue? Well, this is the fundamental question, as I said, that we must answer in this lecture series. And why is it important for us to understand acute muscular fatigue on a deeper scientific level? Why do we need to understand why at a certain level of muscular work or exercise intensity, we experience this muscle inhibition, this fatigue effect? The reason why it is important to understand fatigue is because fatigue is the most limiting factor to human performance. I'll say it again. The reason why it is important to understand fatigue is because fatigue, as described earlier, is the most limiting factor to human performance. Think about it from a sporting perspective. The major factor that influences how one performs during a match or game is fatigue. The athlete who may be a very fast sprinter or the quickest on a team, but fatigues easily, will have overall poor performance. In other words, it doesn't matter how fast you can run, it matters how long you can run fast. Now keep in mind, the importance of fatigue is not just relevant to sports settings. It's also for the everyday individual and even clinical or older populations. What is the major limiter to normal physical function other than, say, pain? It is fatigue. For example, if fatigue prevents one from being able to go up a full flight of stairs, well, that is going to be a major limiter to normal physical functioning. We see this often in the older adult populations. Now, many of you will be going into PT and OT, and I will say, after years of consulting and doing seminars for PTs and OTs, the common missing piece in rehab programs, as well as prehab programs, is dealing with fatigue. Many programs do not condition the patient so that they may perform more muscular work without fatigue. Many programs restore mobility and the ability for patients to perform certain movements, but in everyday life, certain movements are repeated over and over again, such as when you walk. So it's not just about getting a patient to be able to walk normally, but it's also about the patient being able to walk at a certain pace longer without fatigue, or being able to walk faster without fatigue for longer periods. Now, as you can see, fatigue is relative. Some may experience fatigue within 10 seconds of running 10 miles per hour, while another experiences the same level of fatigue walking 2 miles per hour. The fatigue is the same, but it can occur at different levels of muscular work depending on the individual. So the information we are presenting here has a lot of implications to human performance, not just in athletes and sport performance, but also in the general population and in the activities of daily living. The great thing about this topic is that no matter what field you are getting into, whether it is working with performance development in athletes or post-op rehabilitation of older adults, all of the upcoming information is applicable and relevant. Now, before getting into the topic of acute muscular fatigue, we need to go over some foundational information leading into this lecture series. So with that said, we will first do a good refresher on exercise bioenergetics, which was introduced in your exercise physiology course. Now, after this comprehensive refresher on exercise bioenergetics, I will discuss the role of aerobic and anaerobic metabolism during exercise and how both work in an integrative manner to meet the ATP demands of working muscles. I introduced this topic in exercise physiology, but here I will discuss this in much more advanced detail. And this is where we begin to understand the whys of fatigue. Now, going along with the topic of aerobic and anaerobic integration during exercise, we will discuss the topic of the anaerobic threshold and what it represents. We will discuss the environment of the muscle cells that are working below and above the anaerobic threshold. And we will focus largely on explaining why the post-anaerobic threshold, so exercise intensities above the anaerobic threshold, is where fatigue occurs. So in this section, we will explain why acute muscular fatigue occurs during these types of exercise. So what is that stressor that causes fatigue during exercise that is above the anaerobic threshold? Then, of course, a huge component of this lecture series will be on the mechanisms of acute muscular fatigue. This is the question of how acute muscular fatigue occurs. Then in part 2 of this lecture series, I will discuss the science of conditioning and the adaptations that result in one being able to produce more muscular work without fatigue. For example, before the conditioning program, an athlete can sustain a 12-mile-per-hour run for only 5 minutes due to fatigue. But after a month of conditioning, the athlete is now able to sustain 12 miles per hour for much longer because there is no fatigue. So in part 2, I will discuss what explains these improvements. So again, the science underlying conditioning. In addition, we will discuss how to achieve these improvements. So explain the question of how do we get to the point where we can do more muscular work with less fatigue. Again, this will be in part 2 of this lecture series. All right, so like I said, we need to do a comprehensive refresher on exercise bioenergetics. Now, we won't go over all the different bioenergetic pathways, but we need to understand the fundamentals of exercise bioenergetics. Now, this is the foundation of everything we will be discussing with regards to acute muscular fatigue. So without understanding bioenergetics, at least fundamentally, it's really hard for you to understand acute muscular fatigue. Now, I use this figure in my exercise physiology course, and it may be familiar to some of you. And I use this to help students visualize the role of bioenergetics during exercise. This may or may not be familiar to you, but we will go over it regardless. So here we have what we call a reverse causal model, so a reverse cause and effect model. So here we will present the overall process, but in reverse. So we have the end point. The end point is exercise. We want to perform exercise movements such as the back squat shown in this animation. So let's ask some basic questions. In order for us to exercise and move, what needs to happen? Well, we know generally that muscle contractions need to happen. In order for us to move our body, we need specific muscles to contract to produce enough forces to generate joint actions. Here we have an example of a joint action called elbow flexion. In order for us to produce this elbow flexion, we need the biceps to contract and apply force on the bones of our limbs. Okay, so let's dissect this even further. In order for our muscles to contract, what needs to occur in our muscles? Now we have to go way back to our exercise physiology course. As we learned, muscles contract because inside muscle fibers, we have the myofilaments undergoing the sliding filament process. Remember, muscles shorten and lengthen during contractions because the myofilaments, the thick and thin myofilaments within each of the muscle fibers, they slide past one another. So let's dissect this even more. In order for the thick and thin filament to slide past one another to produce a muscle contraction to perform exercise, what needs to occur? Let's zoom in on these myofilaments. We know from our exercise physiology course that the thick and thin myofilaments need to physically interact with one another through a process called cross-bridge cycling. The myosin head attaches to the actin of the thin filament and slides it across. Now, if you remember the first lecture in exercise physiology, we discussed the requirements for cross-bridge cycling. We discussed how in order for the myosin head to connect to the thin filament, more specifically the actin on the thin filament, to do work on the thin filament, like sliding it across, it requires energy. And we discussed briefly that the energy comes from this little molecule called ATP, adenosine triphosphate. And the process by which the energy is released from ATP, we said, was referred to as ATP hydrolysis, which is essentially breaking up the ATP molecule to release the energy inside it. In other words, ATP is hydrolyzed. Now, more on this later in much more detail. Just for now, understand that ATP is an energy molecule, and through the process of ATP hydrolysis, it releases the energy to the myosin head so the myosin head can perform work on the thin filament, sliding it across to perform the sliding filament process for muscles to contract and for you to exercise. So with that being said, in order for cross-bridge cycling to occur, there needs to be ATP available. So just to recap, to perform exercise, we need muscle contractions. To contract muscles, the myofilaments need to slide across one another per the sliding filament theory. For the myofilaments to slide across one another, you need cross-bridge cycling. For cross-bridge cycling to occur, you need energy. The energy is derived from ATP and the breakup of ATP, which is again referred to as ATP hydrolysis. So here now we see ATP, and we know that ATP is the immediate source of energy for cross-bridge cycling. I also alluded to the fact that ATP is an intermediate fuel molecule, and that it holds energy for a very brief period of time, meaning once it is available, it will be hydrolyzed or broken apart. It doesn't just hang around, nor is it stored in the cell in high amounts. When we exercise, our muscle cells require a constant availability of energy, which means it needs a constant availability of ATP. Since ATP is not stored in high amounts, ATP must be constantly generated to meet the energy demand of the muscle cells during exercise. So where does this ATP come from? It comes from the transfer of energy from fuel molecules, and that whole process is called bioenergetics. So we have bioenergetic systems that serve to break down fuel molecules to release the energy from the fuel molecules to hold it temporarily in ATP. Through this process, you form ATP. So when energy is transferred from a fuel molecule to ATP, you form ATP. So essentially, bioenergetics is the process of generating ATP, energy released from fuel and temporarily held in ATP, thereby forming ATP. Now, bioenergetic systems can be generally categorized into two systems. We have the anaerobic system and the aerobic system, also referred to as the anaerobic metabolism and aerobic metabolism. Now, most of you, if I ask you what differentiates these two systems, you would say oxygen, and that is correct. The aerobic system breaks down fuel molecules to release energy to form ATP, which again holds that energy temporarily, and it does this with the help of oxygen. And this is the whole reason why we need oxygen to live. Aerobic ATP production is not just relevant to muscle, it is important for most cells in the body. So anaerobic metabolism involves the breakdown of fuel molecules, the release of energy from said fuel molecules, and the formation of ATP, which temporarily holds that energy without the need of oxygen. So this is an oxygen requiring ATP producing system, and this is a system that does not require oxygen to produce ATP. So the next question is, what are the fuel sources? What are the fuel molecules? The body's fuel sources are referred to as macronutrients. These comprise of carbohydrates, fat, and protein molecules, with carbohydrates and fats being the more utilized fuel sources. These molecules in their chemical bonds hold energy that can eventually be released. And like I said, through aerobic or anaerobic systems, the chemical bonds of these fuel molecules can be broken up and energy is released. Some of this energy is gone in the form of heat, and some of the energy is transferred and held inside the bonds of ATP. And as I said, this process results in the formation of ATP. So now just to recap, the ATP is then broken up through hydrolysis on the myosin head, releasing the energy to the myosin to produce cross-burst cycling with a thin filament, which causes the sliding filament process, which causes muscle contractions, which allow for movement or exercise. Okay, so let's dissect this even further. Where do these fuel molecules come from? We have two sources for fuel. First, we have fuel stored inside various cells in the body within different organs. Fuel stored within the body is described as endogenous, E-N-D-O-G-E-N-O-U-S. Endo meaning inside. So there are fat, carbohydrate, and protein stores within various cells in the body. The major areas of fuel storage include the liver, adipose, and skeletal muscle. And we will discuss later in what forms these fuel molecules are stored as in these areas. But generally, the liver not only stores fuel, but also distributes fuel to other parts of the body. Adipose is mainly meant for fuel storage, largely in the form of fat molecules, which is why most people refer to adipose simply as fat. And muscle stores fuel in the form of all three, although protein is not favored as a fuel source. It is mostly carbohydrates and fats that muscle utilizes as fuel. Now the next question is, where do these endogenous fuel stores come from? Meaning, after they are used, how are they replenished? I think most of you know. It's by eating food that contains these fuel molecules. In food, you have fat, carbs, and protein molecules that you can consume. The digestive system will break down fat, carb, and protein molecules from food into smaller molecules. Large fat molecules are broken down into fatty acids, large carb molecules are broken down into glucose mainly, and proteins are broken down into its building blocks, amino acids. As it passes through the intestinal tract, these smaller molecules can be absorbed into blood circulation, and these fuel molecules can then be stored somewhere in the body, such as adipose, the liver, or muscle. So the fuel that is provided through food is described as exogenous, EXOGENOUS. Exo referring to outside the body. So we have exogenous and endogenous fuel sources. The exogenous fuel molecules replenish endogenous fuel stores, and these endogenous fuels are then utilized by cells like muscle cells for its energy and therefore ATP needs. And this is through, again, anaerobic and or aerobic metabolism, your bioenergetic systems. So before you move forward in this lecture series, you must be able to understand this overview, since this will allow you to visualize the bioenergetics during exercise. Now, everything on the left side of this red border are the direct processes underlying muscle contractions. It is the machinery involved in muscle contractions. On the right side of this red border, we have the processes that power the machinery for muscle contractions. In other words, the process by which energy is provided for the machinery of muscle contractions. So this is exercise bioenergetics in a nutshell. Now, to provide a technical definition of bioenergetics, bioenergetics, sometimes referred to as energy metabolism, are the metabolic processes which include biochemical reactions that release energy from fuel and temporarily storing them in ATP, aka energy currency. Remember, energy is never created or destroyed, only released and transferred from one thing to another. So for example, from a fuel molecule to ATP. Now, a specific area of study of bioenergetics is exercise bioenergetics, which is the response in these bioenergetic systems to a situation in which there is an increase in ATP/energy demand in active tissues or cells, especially skeletal muscle, since it is the major expender of energy during exercise. Remember, energy is necessary to power the process of muscle contraction. So when you exercise, there will be an increase in energy and therefore ATP demand. And as we said in the previous slide, when ATP demand goes up, the bioenergetic systems respond by providing that ATP. Now, more specifically, however, when muscle activity/contractions increase, like during exercise, the rate of ATP demand increases, meaning the muscle cells need energy and thus ATP at a faster rate. Remember, rate is a certain quantity of something in relation to a unit of another thing, such as time. So for example, ATP per second. So in response to this increased rate of ATP demand, there is an increased rate in ATP production. So the takeaway here is that the level of muscle activity determines the rate of ATP demand, which determines the rate of ATP production. This here determines how active those bioenergetic systems are in taking energy from fuel molecules and using it to form ATP, which contains that energy for a temporary period of time. Now here we have some more key definitions that I want to discuss now before we get into the intricate details in the meat of this lecture. As I introduced in the start of this lecture, what is acute muscular fatigue? Acute muscular fatigue is a state in which muscle contractions and therefore force production is inhibited due to the presence of high metabolic stress caused by high to max intensity exercise. We will discuss what this metabolic stress is, but in essence, fatigue is a negative feedback mechanism to relieve the muscle and even the body from this metabolic stress. It is an acute stress response. So like I said in the beginning, fatigue is what you experience when you are, for example, sprinting and you slow down despite putting forth as much effort as possible. Something is inhibiting your muscles from contracting as hard as you want them to. This fatigue occurs because there is something stressful to the muscle when you are sprinting, and we will discuss again what that stress is. But for now, just know that this stress is harmful to the muscle, and thus the muscle is inhibiting itself to relieve itself from the stress. It pumps its own brakes. So let's put this into the context of, say, a VO2 max test. If you remember your exercise physiology lab, The subject performed the VO2 max test, which involved increasing running intensity, whether through increased speed or incline, at progressive stages. So after every stage, the subject would have to run at higher intensities. Now, in the beginning of the test, the running intensity was not difficult. It is something that the subject can sustain for a good period of time. In other words, they don't experience fatigue. And as each stage passes, the intensity increases. Now, if you remember, there was a stage in which the intensity started to become very difficult for the subject to sustain. There was an intensity where the subject, no matter how hard he or she was trying, it was becoming more difficult to sustain that intensity. In other words, something was inhibiting their muscles from working as hard as he or she wanted them to. This is the point where the muscles are experiencing this metabolic stress, and the stress response, which is the inhibition of the muscle, is now initiated. Then another stage passes and the intensity increases. At this point, the subject is experiencing more fatigue because there is more metabolic stress to the muscle. This inhibition of the muscle, i.e. fatigue, is becoming more severe, and it becomes more apparent that the muscle is trying to slow itself down, and exercising at this intensity is becoming harder and harder to sustain. This is what we are referring to when we are talking about fatigue. It is acute, meaning it is an immediate and temporary process. It is an immediate and temporary inhibition of muscle in response to a stressor that occurs with very high amounts of muscular work or exercise intensity. So now that we are clear as to what fatigue is, then what is the difference between fatigue and this term exhaustion? These two terms are often used interchangeably, but they are not the same thing. Exhaustion is referring to a situation in which muscle contractions are not necessarily inhibited, but rather impaired due to a lack of energy support. This is due to the depletion of energy substrates, aka fuel. So exhaustion is referring to fuel exhaustion or fuel depletion. The fuel necessary to provide the ATP and thus energy for muscle contractions have been exhausted or depleted. Since the major fuel for skeletal muscle contractions is muscle glycogen, which is a stored carbohydrate molecule, exhaustion is synonymous with the term muscle glycogen depletion. So as you can see, exhaustion and fatigue both result in decreased performance and reduced muscular force production, but it is due to different factors. Fatigue is due to an immediate and temporary inhibition of muscle contractions in response to a metabolic stress, while exhaustion is an impairment of muscle contractions due to the depletion of fuel. So if I ask the question, which of the following is responsible for the eventual decline in speed during a single all-out sprint effort, the answer would be fatigue, not exhaustion. If I ask which of the following is responsible for the eventual decline in muscular force production after 5 hours of steady state running, it would be exhaustion, fuel depletion. As you can see, fatigue is a matter of exercise intensity, while exhaustion is a matter of exercise duration. So please know the differences between fatigue and exhaustion, and especially understand the definition of fatigue, since that is the topic of discussion in this lecture series. Now, we will return to the topic of fatigue later on, but first we still need to go over basic information regarding bioenergetics. Again, this is the process by which fuel is broken down and the energy is released from the fuel molecules and transferred and held temporarily in the cell's energy currency, ATP. The ATP then can be split apart, releasing that energy for the muscle cells to use to power things like muscle contractions. Now in this figure, you will see an overview of the body's fuel sources that are used for ATP production. So let's start up here with food intake. Now the primary function of food is to provide the body with fuel in the form of energy-containing molecules called macronutrients. These are categorized into 3 types of fuel molecules: fats, carbohydrates, and proteins. Now in food, there are multiple types of fats, carbohydrates, and proteins, but when it is introduced to the digestive system, they will all be broken down into its simplest form, or its building blocks, if you will. So fats will be broken down into its simplest form, which are called fatty acids. Carbohydrates will be broken down into its simplest form, which is primarily glucose, and proteins into its building blocks, which are amino acids. Now, these fatty acids, glucose, and amino acids are absorbed through your intestinal tract, and then they enter blood circulation. Then they get distributed throughout the body and can get taken up by various cells that can utilize it for fuel, such as muscle cells. Now let's just say this third box here represents the muscle cell. Now in your PowerPoints, you'll see arrows here, here, and here. Go ahead and ignore those. When fatty acids, glucose, or amino acids get taken up by cells, they enter the free pool of free fatty acids, glucose, and amino acids respectively inside the cell. Now from here, the free fatty acids within this pool have one of two fates. They can be stored up in the form of a large fat store molecule called a triglyceride or triacylglycerol. And that process is generally described as anabolism. It's an anabolic process. Anabolism is the building of a larger molecule from smaller constituents. So anabolism of fats involves three free fatty acids combined together by a glycerol to form a large fat molecule, a triglyceride. The anabolism of fats, which again is the formation of a triglyceride from fatty acids, is referred to as lipogenesis. So fat anabolism is lipogenesis, the formation of triglyceride from three fatty acids combined by glycerol. Now, the other fate of fatty acids within this pool is that it can undergo oxidation. Now, oxidation is a process involving multiple biochemical reactions to break apart a fuel molecule like a fatty acid for the purpose of releasing its energy, transferring that energy, and holding that energy temporarily in the cell's energy currency, ATP. So free fatty acids can undergo fatty acid oxidation, and through that process, energy is released from that fatty acid and is temporarily held in the bonds of ATP, thus forming ATP. Now, when triglycerides that are stored in the muscle cell, for example, are broken down or catabolized, the fatty acids are freed and they enter the free fatty acid pool within the cell. This process is generally described as catabolism. Catabolism is the breakdown of larger molecules into smaller constituents, and catabolism of triglycerides is called lipolysis. Remember, lysis refers to catabolic reactions and lipo refers to fat. So lipolysis is the breakdown of triglyceride. And as a result, the three fatty acids within that triglyceride are freed up and they enter this free fatty acid pool. So what about carbohydrates? So glucose, just like the free fatty acids, they will be absorbed and circulated and can get taken up by cells like the muscle cell. The glucose, just like free fatty acids, has two fates after it enters the glucose pool within the muscle cell. A bunch of glucose can be bonded together to form glycogen, which is a larger carbohydrate molecule meant for storage. This is referred to as glycogenesis, formation of glycogen. And this, just like lipogenesis, is an anabolic process. So the anabolism of carbohydrates from glucose to glycogen is called glycogenesis. Now, the glucose can also undergo oxidation, just like the free fatty acids. This again is the process of breaking up the glucose molecule to release its energy to form ATP, which temporarily holds that energy in its bonds. Now, when the glycogen is broken up or catabolized to provide glucose to the muscle cell, this is a process called glycogenolysis, lysis of glycogen. Now, finally, we have the amino acids. Now, once the amino acids are absorbed and circulated, cells like the muscle cell can take them up. The amino acids can then enter the free amino acid pool inside the cell. Now from here, the amino acids can be used to build proteins in the cells, which is the primary function of amino acids. Proteins are essential for cellular function since there are so many different proteins that play a certain role inside the cell. For example, in muscle cells, there are proteins that are responsible for producing contractions like actin, myosin, tropomyosin, and so forth. Now, each individual protein is characterized by a unique amino acid sequence. So every protein has a different combination of amino acids. This unique combination determines what type of protein it is. So like actin has a different amino acid sequence than myosin and so forth. The process of building proteins from amino acids is of course an anabolic process and is simply referred to as protein synthesis. Now, the other fate of these free amino acids within this pool is oxidation for the purpose of producing ATP. However, as you can see here, it is not a preferred fuel molecule. And this is mainly because amino acids are needed to form proteins with, which have very important functions in cells. Now, glycogen and triglycerides only serve one purpose. It is to just exist as a fuel source. So proteins and its amino acids are used highly as a fuel during situations when there is a depletion of carbohydrates and fat stores, basically when the cell has no other option. Now, one unique aspect about amino acids is that amino acids called glucogenic amino acids can be used to form new glucose with. And this occurs primarily in the liver, not in the muscle cell. And this process is called gluconeogenesis, formation of new glucose. Now, when proteins are broken down, this is a catabolic process and is commonly referred to as protein degradation or protein breakdown. A more technical term is proteolysis, lysis of proteins. Okay, so this is a complete overview of fuel metabolism in the body, showing what specific fuel molecules are oxidized to release its energy to form ATP, which will hold that energy temporarily. So let's talk more about ATP in some good detail. Now, most of you are somewhat familiar with ATP and generally understand it as the cell's energy currency, all cells in the body, not just muscle cells. So when the cell needs energy to do something, it gets it from ATP. So for example, when the muscle cell wants to contract, it needs energy, and ATP is what provides it. Now in the human body, energy doesn't go directly from the fuel to the cellular component that needs it. The energy from the fuel is released and then temporarily contained within an intermediary fuel molecule called ATP, and it's contained only for a very brief period of time. So what is ATP and where is this energy contained? ATP, as most of you know, is adenosine triphosphate. Here we have an adenosine backbone made up of two molecules, adenine and a ribose sugar. Next, we know that triphosphate stands for three phosphate groups, or simply three phosphates. And that is what we have here. A phosphate or phosphate group is made up of a phosphorus, which is in the middle here, and four oxygens, which are bonded to the phosphorus, and you see that surrounding each of these phosphorus. This is together called a phosphate or a phosphate group. Now, these three phosphates are termed alpha, beta, and gamma phosphates. Remember that. Alpha, beta, and gamma phosphates. And they are all bonded together via phosphoanhydride bonds, or in simpler terms, phosphate bonds. Now, as you can see, we have one phosphoanhydride bond holding the gamma and the beta phosphates together, and we have another phosphoanhydride bond holding the beta and alpha phosphates together. Now, as you can see, these phosphoanhydride bonds, again simply called phosphate bonds, are described as high energy. Now, here's an important detail. These bonds contain high amounts of energy, but this does not mean that they are strong bonds. These are very weak and unstable bonds, meaning they do not last very long and can be broken up in a single quick reaction, which we'll discuss shortly. Now, the reason why these bonds are considered high energy is because it takes a lot of energy to maintain these bonds. I want you to think of these bonds as large compressed springs. We know that energy is required to keep the spring compressed and that the compressed spring has a lot of potential energy. And to harness that energy, you need to release the spring. So think of these bonds as those compressed springs. And these bonds are holding a lot of potential energy. Now the question before moving on is, where does this energy come from that are contained within these phosphate bonds? Remember the laws of thermodynamics. Energy cannot be created or destroyed. It can only be transferred from one thing to another. So as I introduced earlier in the recap of bioenergetics, the energy to form these phosphate bonds comes from fuel molecules, more specifically the oxidation of fuel molecules, the fats, carbs, and proteins, more specifically amino acids. This is how ATP is formed. So generally speaking, ATP is formed when you have an ADP, adenosine diphosphate, with only two phosphates, and energy from a fuel molecule is used to form another phosphate bond, thus adding a third phosphate, creating ATP, triphosphate, which will now contain that energy in the phosphate bond for later use. So the next question is, how is the energy released from these phosphate bonds to power cellular processes such as muscle contractions? How do we release this quote-unquote compressed spring? So let's take a closer look at this process of splitting the phosphate bond to release the energy contained within it. Again, this process is referred to as ATP hydrolysis. Like I mentioned earlier, whenever you see the term end in lysis, it is a reaction that breaks apart a molecule. So ATP hydrolysis is the breaking apart of ATP. Hydro is referring to water, so we know water is somehow involved in the lysis of ATP. Now up here we have a more visual representation of ATP hydrolysis, and below we have a more formulated depiction. So first we have ATP. And as I said earlier, these phosphate bonds contain potential energy, like a compressed spring. This energy, again, came from a fuel source that was oxidized. And that energy that was released from those fuels is being used to hold these phosphate bonds together. Now let's put this into the context of muscle contraction. ATP attaches to the myosin head, and in order for the myosin to do work on that thin filament, it requires energy, and ATP is going to provide it. So as we said, the energy in ATP is released by splitting up these bonds. In this case, this last bond right here. Now to split this bond, we need two things. First, we need a water molecule, H2O. And then we need a catalyst. And as most of you know, in biochemical reactions, a catalyst is usually in the form of an enzyme protein. So the enzyme that catalyzes ATP hydrolysis is ATPase. Any term ending in ASE is an enzyme in biochemistry. So ATPase will catalyze this reaction where this phosphate bond is split open. And as a result of this reaction, the last phosphate is broken off because, again, the bond has been broken. Now, in so doing, you are left with an ADP with only two phosphates, and the phosphate that was just broken off is called an inorganic phosphate, which is symbolized as a P subscript I. Now, what happened to the water molecule? Now, this will be discussed in more detail in the next slide, but as you can see here, we have a positively charged hydrogen ion. And this is simply referred to as a proton. Why? Because we know that hydrogen, based on the periodic table of elements, is in position number 1, indicating that it has one proton. Now, if it was a neutral hydrogen with no charge on it, this would be one proton and one electron. If it was a negatively charged hydrogen ion, it would have one proton and more than one electron. If it is a positively charged hydrogen ion, as we see here, it would be one proton and 0 electrons. And this is why we just simply call a positively charged hydrogen ion a proton. So this proton came off of the water molecule. This proton plays a critical role in acute muscular fatigue, so we will come back to this later. For now, remember, this proton was derived from ATP hydrolysis and it came off of the water molecule. Now, what about the rest of the water molecule, the other hydrogen and the oxygen? As you see here, we have a oxygen plus hydrogen, also known as a hydroxyl group. You can see that it is bonded to the inorganic phosphate that was just split off from ATP. So in the next couple slides, we will discuss how all this happens in much more detail. Now, through this whole process, the energy that was contained in that phosphate bond was released, and this energy can then be used to power cross-burst cycling and thus muscle contractions. Okay, so in summary, ATP hydrolysis is the splitting of the last phosphate bond of ATP, releasing its energy. Now, to resynthesize ADP back to ATP, you need energy from a fuel molecule to be transferred to form a new phosphate bond on ADP, thus forming ATP. And this is what bioenergetic systems do. So please keep in mind, we are still on the refresher course for bioenergetics, and you will see why this is important for you to understand before delving into the science of acute muscular fatigue. So let's discuss ATP hydrolysis in even further detail. Okay, so here on the top left, we have an ATP molecule. We have an adenosine and 3 phosphates. And as I mentioned earlier, the energy is contained within these bonds between the phosphates. And these bonds are referred to as phosphoanhydride bonds, or simply phosphate bonds. Now, it's depicted here using this figure of a battery. Think of ATP as a fully charged battery. Contained in those 2 phosphate bonds between the gamma and beta phosphates and the beta and alpha phosphates, we have about 50 kilojoules per mole total of energy within the ATP molecule. Now, these phosphate bonds, although they are high energy, like I said, they are very weak and unstable. Again, it's kind of like a heavy spring that is compressed, and this is why ATP doesn't exist for very long. Once it is produced, it is almost immediately hydrolyzed. And because these bonds are weak and unstable, it doesn't take a lot of reactions to break it apart. In other words, ATP hydrolysis requires very little reactions. It only requires one reaction and one enzyme, as we discussed in the previous slide. So to reiterate, in order to utilize the energy contained within these phosphate bonds to power cellular processes such as muscle contractions, the energy must be released from these bonds via ATP hydrolysis. So how does ATP hydrolysis work? When ATP is hydrolyzed, the last phosphate bond is broken, and the energy contained in that last phosphate bond is released. Yes, you can hydrolyze ADP as well, but for now, let's just talk about ATP hydrolysis. So as we indicated in the last slide, ATP hydrolysis requires a reaction with a water molecule, H2O, and that is shown right here. Now let's take a closer look at this water molecule. The oxygen atom on H2O contains 2 lone electron pairs, and that is indicated here by these 4 dots. You have 1 electron pair here and another electron pair here. They are considered lone electron pairs because these electron pairs are not used to form a bond with anything in the water molecule. The electron pairs here and here are used to form a bond with hydrogen. Now, lone electron pairs are highly reactive, meaning they want to form a bond with something, and that something in this case is ATP, more specifically with this gamma phosphate, and even more specifically with the phosphorus of this gamma phosphate. So the way hydrolysis works is that this water molecule, more specifically the lone electron pairs on the oxygen, needs to perform what we call a nucleophilic attack on the phosphorus of the gamma phosphate group. I'll say that again. The lone electron pairs on the oxygen needs to perform a nucleophilic attack on the phosphorus of the gamma phosphate group. A nucleophilic attack results in a new bond between the gamma phosphate and the water molecule. And through this process, the phosphate bond between the gamma and beta phosphates is broken. And as a result, energy contained within that phosphate bond is released. As an analogy, the compressed spring is released, and that potential energy within the compressed spring is released. Now, as we said before, in order for any reaction like this to take place, there needs to be an enzyme. And the enzyme that catalyzes ATP hydrolysis is again ATPase. So how does ATPase work and how does it catalyze hydrolysis? So let's turn our attention to this figure down here to the right. On the y-axis, you have a measure of free energy called Gibbs free energy, which is abbreviated by the capital letter G. So this point here is 50 kilojoules per mole, which is the amount of energy contained in ATP within those phosphate bonds. This is the full battery. Now, the point of ATP hydrolysis is to drain the battery to some extent by releasing the energy in that last phosphate bond through the nucleophilic attack. But here's the thing. The nucleophilic attack actually requires a bit of energy. Let's focus on the red line right here. As you can see, the red line increases for a brief period, meaning there is energy input required. This represents the fact that before energy can be released from ATP, there is a need for additional energy to get the hydrolysis, more specifically the nucleophilic attack, started. So why does the nucleophilic attack cost energy? Well, it has to do with the difference in electrical charge. You see, the lone pair of electrons, as you see here on the oxygen of H2O, is negatively charged. They are electrons. The phosphate on ATP, more specifically this gamma phosphate, has an overall negative charge as well. In particular, the oxygens on the phosphate have a negative charge, as you can see by these symbols next to the oxygen. What we all should generally know by now is that negative and negative charges do not attract, they repel. So naturally, the water molecule and the ATP repel one another due to the negative charges on both ends. So this is why it takes a bit of energy to get the water molecule close enough to the ATP so that the lone pairs of electrons on the oxygen can perform the nucleophilic attack on the phosphorus on the gamma phosphate. This is kind of like you taking a negative end of a magnet and trying to get it close to the negative end of another magnet. You know it's going to require some energy to do this. So where does the enzyme ATPase come into play? What ATPase does is make the nucleophilic attack much more energy efficient. What it does is it lowers the energy and thus quote unquote effort required for the water molecule to react with the phosphate. How does it do this? Now, ATPase is an enzyme that can sort of wrap itself around the ATP molecule. Now, ATPase has an overall positive charge. I'll say that again. ATPase has an overall positive charge, and thus by wrapping itself around ATP, it makes ATP less negative, and thus there is less natural repulsion between the lone electron pairs on the water molecule and the gamma phosphate. Thus, less energy is required initially to perform the nucleophilic attack when ATPase is present, and that is represented here by the blue line. You can see that the initial energy requirement for ATP hydrolysis is lower with ATPase activity. So again, the ATPase essentially lowers the initial energy required for the nucleophilic attack and thus hydrolysis. It makes the ATP less negative so that the water molecule is less repulsed from ATP. Now, at this point right here, below the 50 kilojoule per mole line, we see then a drop in energy in ATP. And this is when the nucleophilic attack is completed and thus hydrolysis occurs. This is the point where the phosphate bond is broken and the energy within that bond is released. So this represents the energy that was released from that last phosphate bond upon the nucleophilic attack and hydrolysis. Now, in that phosphate bond between the gamma and beta phosphate, there is about 30 kilojoules per mole of energy. So upon hydrolysis of ATP, about 30 kilojoules per mole of energy is drained from the ATP. And that 30 kilojoules per mole is used to power cellular processes like, again, cross-bridge cycling for muscle contractions. So as you can see, the battery upon that nucleophilic attack and thus hydrolysis is drained about 30 kilojoules per mole. This was the amount of energy that was contained and subsequently released from that last phosphate bond. And again, that energy that was released is then used to power cellular processes like cross-bridge cycling for muscle contractions. Now on the left, you will see the results of ATP hydrolysis. So after the nucleophilic attack, the gamma phosphate is now broken off since the water molecule replaced that phosphate bond between the gamma and the beta phosphates. This is referred to now as an inorganic phosphate, or P subscript I. Please remember this, as this inorganic phosphate is involved in acute muscular fatigue, which we'll talk about later. Now here you can see that one oxygen and one hydrogen from the water molecule, also known as a hydroxyl group, is attached to this free phosphate. And you can also see that one of the hydrogens from the water molecule was freed, and that is shown here as a positively charged hydrogen ion, or in other words, again, as I said before, a proton. And like I also said before, please remember this proton and where it came from, because this proton is implicated in the process of acute muscular fatigue. So at the end of ATP hydrolysis, you are left with an inorganic phosphate and a free proton, both of which, as I said, are implicated in acute muscular fatigue. More on this later. Okay, lastly, going back to this figure here, remember, ATPase is an enzyme that facilitates the nucleophilic attack by making the ATP less negative and thus less repulsive to the lone electron pairs on the water molecule. So ATPase is obviously very important to facilitate ATP hydrolysis and energy release from ATP. So it would be logical to suggest that with greater ATPase levels inside the muscle cells and thus ATPase activity, ATP hydrolysis and thus energy release would be much more efficient. And this is definitely the case. With more ATPase activity in the muscle, the more efficient ATP hydrolysis is on that myosin head, and thus there is more quote-unquote power provided for cross-burst cycling and muscle contractions. How do you think this would translate to muscle force production and exercise performance? It would be favorable. Now, generally speaking, which muscle fiber type has greater contractile force production and also greater contractile speed? If you remember from our exercise physiology course, you will remember that the type 2 muscle fibers, more so the type 2X muscle fibers, have the greatest contractile force and speed. And one of the factors that contribute to this characteristic of type 2 fibers compared to type 1 is that they have much more ATPase levels and ATPase activity. Meaning, in type 2 fibers, more so type 2X fibers, the nucleophilic attack and therefore hydrolysis is much more efficient, thereby making cross-bridge cycling and thus muscle contractions more forceful and faster. Now, to visualize this whole process in the context of muscle contractions, here we have ATP on that myosin head. We need to transfer the energy from the ATP within that last phosphate bond to the myosin head, so the myosin can perform work on the thin filament. So ATP will undergo ATP hydrolysis, which involves a water molecule, more specifically the lone electron pairs on the oxygen of the water molecule, performing a nucleophilic attack on the last phosphate of ATP. Through that process, the water molecule creates a new bond with the gamma, or the last phosphate, which in turn breaks up the phosphate bond. At this point, about 30 kilojoules per mole of energy is released from that phosphate bond and transferred to the myosin head. This would put the myosin head in a high energy configuration. As you can see, as a result of hydrolysis, the ATP is now split to a DP and an inorganic phosphate, which is attached to the hydroxyl group from the water molecule. Also, what is left but not shown here is the proton, which is the positive hydrogen ion which came off of the water molecule. Now, because the myosin has energy now, it can perform work on the actin filament. It can attach to it and slide it across, thus producing the effect we see right here in this animation, the sliding filament process, which will cause the muscle to contract. So moving forward, I want to present to you 3 fundamental questions leading into our discussion on aerobic and anaerobic metabolism and how they both contribute to ATP needs during exercise, especially in response to increasing intensity and thus ATP needs. So first question, how much intramyocellular ATP is stored? So how much ATP is stored inside the muscle cell at any given time? The answer is not very much. ATP is not meant for storage. It is a molecule that contains energy from the oxidation of a fuel molecule for only a temporary period of time until it's hydrolyzed. Thus, ATP hydrolysis occurs almost immediately after it's produced. And this is not just specific to muscle cells, but all cells in the body for whatever cellular processes that require energy. So to put this into simple terms, ATP production is on a per needs basis, meaning the rate at which ATP is produced is based on the rate that it is needed. You also must understand that the body is constantly oxidizing fuel and using up ATP. In other words, the body is constantly in a state of energy transfer in its cells. The reason for this is thermoregulation. Our core body temperature is about 100 degrees Fahrenheit, just about. And we are usually in environments that are below 100 degrees, like room temperature. So if we are standing in a room that is 72 degrees, how does our body maintain a 100-degree core temperature? It generates heat. How is the heat produced then? Heat is a form of energy. And every time energy is released from fuel oxidation or ATP hydrolysis, some of that energy is lost in the form of heat. So yes, we are constantly burning fuel, producing ATP and using ATP. And of course, when we exercise, this will ramp up because ATP needs are elevated. This leads us into the next question. If there is limited ATP in our muscle cells at any given time, how do muscle cells have enough energy support for contraction during exercise? Well, as I said before, our muscle cells through bioenergetic systems generate ATP at a rate that meets the ATP/energy needs of working muscles. ATP production again is on a per needs basis. So when ATP demand increases, like during exercise, the bioenergetic systems ramp up ATP production by quote unquote burning more fuel molecules. So now the last fundamental question. When there is a higher demand for ATP in our muscle cells, like during exercise, how do our muscle cells generate more ATP? It is through our bioenergetic systems, which is a part of human metabolism involving biochemical reactions in dedicated pathways that catabolize and oxidize fuel molecules. Now, fuel oxidation, just a reminder, is a specialized process of catabolism that releases energy from fuel molecules so that it may be transferred to form ATP, which will then contain that energy in the phosphate bond. This is what is generally referred to as the process of quote unquote burning fuel. And these fuel molecules include your nutrients. You have carbohydrate fuel sources, which include glucose and glycogen. You have a fat fuel molecule called fatty acids, and of course, amino acids, which are building blocks of proteins. We also have a small fuel molecule called phosphocreatine, as you might remember. As an important side note, which one of these fuel sources is your muscle cell's primary and most versatile fuel? It is carbohydrate, and more specifically, glycogen, which is the carbohydrate store that is found largely in muscle cells. Keep this important piece of information in mind as we continue on. All right, so now here we have some critical key points for you to understand. And this is something we introduced during our exercise physiology course, but it is important for you to understand this information to understand the concept of muscular fatigue. Our ATP producing bioenergetic pathways can be categorized as aerobic or anaerobic, and we generally know the difference between these two pathways or systems. Aerobic ATP production requires oxygen, and anaerobic ATP production does not. Muscle cells always produce ATP aerobically. And when ATP demand goes up, like during exercise, aerobic ATP production will increase to meet that demand. And please understand that as ATP demand increases and aerobic ATP production increases to meet that demand, we have to know that oxygen demand also increases, since again, aerobic ATP production requires oxygen. So this means that oxygen availability also needs to increase with this increased ATP and oxygen demand. Again, to support aerobic ATP production. But there will be a certain point where the exercise intensity involves an ATP demand that your aerobic system cannot completely fulfill. I will say that again. There will be a certain point where the exercise intensity involves an ATP demand that the aerobic system cannot completely fulfill or satisfy. So what is causing this limitation to the aerobic system? For example, when you do all-out effort exercise, like during the Wingate test, which a lot of you performed during your exercise physiology lab course, why can't the aerobic system completely fulfill the ATP needs of the muscle cells? It's because of two factors. The major factor that limits the aerobic system's ability to meet ATP demand is oxygen delivery and availability. So as the ATP demand and thus oxygen demand increases in the muscle cell with increasing intensity of exercise, there will be a point in which the oxygen delivery and availability can't increase enough to meet the demand. And thus, at this point, there is insufficient oxygen to further support aerobic ATP production to meet this increasing demand. Now, the other factor is the mitochondria, which is where aerobic ATP production occurs. The quote-unquote power of the mitochondria to produce ATP aerobically can limit the ability for the aerobic systems to meet the ATP demands. Now, the first factor and this factor go hand in hand. So if we have limited oxygen delivery and availability, this is going to in turn limit the ATP producing quote-unquote power of the mitochondria, which is the same thing as saying the ATP producing power of the aerobic system. We are also talking about the size and number of the mitochondria, which we know is your cell's power plant. So if mitochondria size and number in the muscle cells is very low, this would of course limit the ATP producing power of the aerobic system. So again, these two things go hand in hand. At the same time, the mitochondria size and number also plays a role in how the aerobic system can be limited, especially at higher demands of ATP. So to put this all together, at the exercise intensities in which the ATP demand cannot be completely fulfilled by the aerobic system due to insufficient oxygen delivery and availability and mitochondrial quote-unquote power, what other system would be able to fulfill the remaining ATP demand that the aerobic system cannot? Yes, it would be the anaerobic system, which does not require oxygen to produce ATP. So it should make sense to you that the muscle cell utilizes an ATP producing system that does not require oxygen, because the whole reason why the aerobic system cannot cover the full ATP demand is mainly due to insufficient oxygen delivery and availability, which obviously would affect the ability for the mitochondria to generate ATP aerobically. So the muscle cell needs to use an ATP producing system that doesn't require oxygen. And again, this is called your anaerobic systems or anaerobic metabolism. So in essence, the anaerobic systems are simply a backup to the aerobic system when the aerobic system cannot fully satisfy the ATP demand of the muscle cells. This also means that the anaerobic system kicks in whenever there is a situation in which the oxygen delivery and availability in muscle cells do not meet the oxygen demand of said muscle cells. Since again, that is the reason why the aerobic system in those muscle cells cannot meet the full ATP demand. What else does this mean? It means that the anaerobic system will kick in during higher relative exercise intensities. Relative meaning intensity is relative to the individual. So high intensity can be walking 5 miles per hour for one person, and the same relative intensity would be, say, 10 miles per hour for another individual who's in better shape. So let's take a look at this concept of aerobic and anaerobic ATP production during exercise of different intensities in a more visual manner. Now, the information presented here is going to clear up many common misperceptions concerning aerobic and anaerobic metabolism. So let's talk about this common misperception so you yourself don't have this misperception. If you have the wrong idea concerning aerobic and anaerobic metabolism, then the rest of this lecture series will be very confusing for you. And so through these visuals, you should be able to understand how the aerobic and anaerobic systems work together during exercise to meet ATP needs in the working muscles. Now, the common misperception regarding aerobic and anaerobic metabolism is represented here on the left in this figure with the giant X next to it. Now, on the y-axis, you will see represents the rate of ATP production and therefore needs. So how fast ATP is being generated by bioenergetic systems collectively inside your muscle cells to meet the muscle cells' ATP needs. Now, on the x-axis, you have general terms to describe how hard your muscles are working. So light effort to max effort, or light intensity to maximum intensity. And in more practical terms, you can say from resting to all-out sprinting. Now, without looking at the colored sections here below the curve, just focus on the actual curve. Now, as you can clearly see, as effort or intensity increases from light to max, so like from walking to sprinting, for example, the rate of ATP production increases since, of course, as we go from light to max intensity, the energy and thus ATP demand increases as well. Remember, ATP production increases when ATP demand increases. So far, this is basic information. Yes, as we exercise harder, the energy and thus ATP demand increases, and thus our bioenergetic systems will increase ATP production accordingly. Now, like I said, our bioenergetic systems are either aerobic or anaerobic. Aerobic requires oxygen to produce ATP, and anaerobic does not require oxygen to produce ATP. Okay, so as I said, this figure here represents a common misperception on how aerobic and anaerobic systems work during exercise. Many people have the understanding that during light efforts or intensities, our muscles rely on aerobic ATP production as represented by this white area under the curve. And as we increase our effort or intensity, aerobic ATP production ramps up, and this is why we consume more oxygen through our cardiorespiratory system. Our heart works harder to get that oxygen to our active muscles to support the aerobic system. This understanding of the aerobic system is correct to some extent. So here, as you can see, the aerobic system up to this point right here is fulfilling 100% of the ATP demand of the muscle cells. Now here's where things get misunderstood. Many people believe that at some certain level of intensity and ATP demand, like right here, for example, in this figure, our aerobic systems completely shuts down and no longer supplies ATP and no longer is contributing to the ATP demand of the muscle. And what would completely take over at this point is the anaerobic ATP production, which is represented by the gray area under the curve. And anaerobic ATP production, which again does not require oxygen, is now providing all the ATP needs at this intensity and above. This is a dichotomous view of aerobic and anaerobic metabolism, meaning this is an either-or type of perception. To be straightforward, this perception of aerobic and anaerobic metabolism is completely false. Hence the big X next to this figure. There is not one single level of intensity and ATP demand where aerobic systems just completely stop producing ATP and stops contributing to the ATP demand of the muscle cells. There is not one point where the anaerobic systems, as shown here, would completely take over the ATP needs of the muscle cells. Now, if this was in fact the case, then think about it. At this point of effort or intensity where the aerobic systems shut down, you would stop consuming oxygen at a high rate, meaning your breathing rate and your heart rate would just go back to resting levels, since by this understanding, your muscles don't require higher oxygen availability since it is now fully dependent on a non-oxygen requiring bioenergetic systems, the anaerobic system. We obviously know this never happens. Imagine running on a treadmill and you are increasing the speed every minute. Imagine as you go higher and higher in speed or intensity, at one point your breathing and your heart rate all of a sudden returns to resting levels and stays that way until you reach maximum sprinting intensity. Again, we clearly know that this does not occur. We know for a fact that as we go from light to maximum intensity exercise, our breathing and our heart rate rises until it maxes out. Why? Because as we go from light to maximum intensity exercise, our oxygen consumption increases until it eventually maxes out. So why do you think our oxygen consumption keeps rising as we increase our intensity of exercise from light to max intensity? Well, it's because our aerobic systems are always, I repeat, always producing ATP. It is always contributing to the ATP needs of the working muscle cells. Aerobic ATP production is the preferred and prioritized means of producing ATP in muscle. It is always active and proportionate to the level of intensity and ATP demand. So let's look at how aerobic and anaerobic metabolism really function during exercise by examining the figure here on the right. Now, as you can see, as effort increases or intensity increases, and thus the rate of ATP demand increases, the aerobic system increases the rate of ATP production, as you can see by this white area under the curve. Now, as you continue to increase exercise intensity and thus ATP demand, at some higher level of intensity and ATP demand, there is a point in which the aerobic system cannot fully satisfy the rate of ATP demand. And that point is referred to as the anaerobic threshold, which is a very important term for you to understand. The anaerobic threshold is simply the level of exercise intensity and ATP demand at which the aerobic system cannot fully satisfy the rate at which the muscle cells need ATP. Now, as intensity increases past this anaerobic threshold, there is a further rise in ATP demand. And the aerobic system, as you see by this white area under the curve, tries very hard to fully satisfy the ATP demand. So as you can see here, the ATP demand is up here on top of the entire curve, but the ATP production via aerobic system is right here on top of the white curve. So the first question is, why can't the aerobic system meet the full ATP demand starting at this point called the anaerobic threshold? It is primarily, again, due to insufficient oxygen delivery and availability, since again, oxygen availability is required for the aerobic system to produce ATP. Now, keep in mind, this does not mean oxygen is not present at all. It just means the rate of oxygen delivery and availability is just insufficient for the aerobic system to fully meet the ATP demand by the working muscle cells. So what fills this gap between the ATP that is needed and the ATP that can be provided by the aerobic system? What satisfies the portion of the ATP demand that the aerobic system cannot satisfy? This is represented by the gray area under the curve. Well, it would make sense that a non-oxygen requiring system would satisfy the ATP demand that the aerobic system cannot cover, since oxygen availability is the limiting factor for the aerobic system. So as we know, this non-oxygen requiring ATP producing system is called the anaerobic system. And as you see in this figure, the anaerobic system becomes more active as you exercise past this anaerobic threshold intensity, because it needs to satisfy the portion of the ATP demand that the aerobic system cannot satisfy. And as you can also see, as we get further away from this anaerobic threshold intensity towards maximum intensity, such as all-out sprinting, there is an increasing portion of the ATP demand that cannot be met by the aerobic system. And so the anaerobic system becomes more and more contributory to the ATP demand. It only covers the portion of ATP demand that the aerobic system cannot cover. So the correct perspective on anaerobic and aerobic metabolism during exercise is that the anaerobic system is simply a backup to the aerobic system during exercise intensities and ATP demands in which the aerobic system cannot fully satisfy the ATP demand of the active muscle cells. Another correct perspective is that there is never, I repeat, never a situation in which the aerobic system shuts off and the anaerobic system completely takes over. During exercise intensities at the anaerobic threshold and above, the aerobic system works as hard as it can with the oxygen that is available and delivered to the muscle cells to meet most of the ATP demand. I'll say that again. During exercise intensities at the anaerobic threshold and above, the aerobic system works hard as it can with the oxygen that is delivered and available to those muscle cells to meet most of the ATP demand. But in addition to the aerobic system, the anaerobic system covers the ATP demand that the aerobic system cannot satisfy. So as you can clearly see, the aerobic system is always active no matter what intensity of exercise, even at all-out sprint efforts. Now, here's some other key terms that you need to understand. The maximum ATP producing capacity of the aerobic system, as you can see here, occurs at maximum intensity. And the maximum ATP producing capacity of the aerobic system is called the aerobic capacity. And this is commonly measured through your maximum oxygen consumption rate. In other words, your VO2 max, which we all measured during your exercise physiology lab course. And it should make sense to you why the maximum oxygen consumption will reflect the maximum ATP producing capacity of your aerobic systems. Because again, the ATP producing power of the aerobic system is reliant on the oxygen availability. So it depends on how much oxygen you can actually consume to provide your muscle cells so that it can produce ATP aerobically. Okay, so moving onward, with all that being said, when someone describes some, say, higher level of intensity of exercise as quote unquote anaerobic, this would be completely false. If I asked one of you to describe anaerobic exercise, you would say something to the effect of high to max intensity exercise like sprinting. Now you understand that describing this type of exercise as anaerobic is a misnomer since, as you can see, this type of exercise is highly and mostly aerobic. Remember, the aerobic system is contributing to most of the ATP demand past this anaerobic threshold. It just can't meet the full ATP demand. So only just a portion, a small portion of the ATP demand at intensities past the anaerobic threshold is fulfilled through anaerobic metabolism. So to call exercise in this zone as anaerobic exercise, again, is a misnomer. Since based on the information that I provided, you can see that all exercise intensities and therefore all types of exercise is aerobic. So there is no real such thing as anaerobic exercise. So this is why I stay away from terms like anaerobic or aerobic exercise or anaerobic or aerobic fitness because they do not make any sense. How can you do exercise that is only anaerobic? There is no way since aerobic systems are always active and increases proportionate to the intensity of exercise. So how can you say anaerobic fitness? How can you be anaerobically fit? There is no way to ever isolate anaerobic metabolism or quote unquote train anaerobic systems. It just does not make any sense from a scientific standpoint. Now, I challenge you to Google anaerobic fitness, and I guarantee you that you will find tons of blog posts and articles written by so-called fitness experts, and I guarantee the information in that article is completely false and not consistent with scientific facts. So lesson here is do not believe everything you read on the internet just because it was written by someone touted as a fitness expert. You need to be smarter than that. And as you grow in knowledge, you will gather the confidence to rebuttal against those who spew misinformation. So to summarize everything we discussed thus far, aerobic ATP production is the preferred and prioritized means of meeting the ATP demand in muscle cells. And as exercise intensity increases, aerobic ATP production also increases proportionately. Now, there's a certain intensity and ATP demand where the aerobic system cannot fully meet the ATP demand of the working muscle cells. This point is called your anaerobic threshold. At intensities at your anaerobic threshold and above, the anaerobic system covers the portion of ATP demand that the aerobic systems cannot. And the maximum ATP producing capacity of the aerobic system is reached at maximum intensity exercise, and that is referred to as your aerobic capacity. So again, as you can see, the aerobic system never shuts off. It never stops contributing to the ATP demand of the muscle cells, regardless of intensity of exercise. And lastly, for some more terminology, instead of describing exercise as either aerobic or anaerobic, since we know now that wouldn't make any sense, it is more accurate to describe exercise as either pre-anaerobic threshold or post-anaerobic threshold. And in this lecture series, we will describe first what's going on pre-anaerobic threshold and what's going on post-anaerobic threshold to help us understand why post-anaerobic threshold exercise is when fatigue occurs. So just to clean this all up, we have the correct visual here of the integration of aerobic and anaerobic ATP production during exercise of varying intensities and ATP demands. And as I emphasized earlier, a key term for you to understand moving forward is the anaerobic threshold. If I asked on the exam what the anaerobic threshold represents, here are the possible answer choices. 1. The anaerobic threshold represents an exercise intensity and ATP demand that the aerobic system cannot fully satisfy. 2. The exercise intensity in which the anaerobic system begins to back up the aerobic system and fulfills only the portion of the ATP demand that the aerobic system cannot. 3. The exercise intensity in which the oxygen availability in the working muscle cells do not meet the required amount of oxygen that is needed for the aerobic system to fully satisfy the ATP demand. So those three answer choices are reflective of the anaerobic threshold. Now let's expound further on that third answer choice because this is an important piece of information for you as we get into discussion on the mechanisms underlying acute muscular fatigue. I said that the anaerobic threshold is also the exercise intensity in which the oxygen availability in the working muscle cells do not meet the required amount of oxygen that is needed for the aerobic system to fully satisfy the ATP demand. So as an example, to further explain this concept, let's take any point on top of this entire curve in the post-anaerobic threshold zone. Let's say this point right here. The top of this entire curve represents the total ATP demand and the amount of oxygen required to fulfill this ATP demand aerobically. However, if you look at box number 2, the top of the white curve represents the oxygen that is available in the muscle cell's mitochondria to support aerobic ATP production. Therefore, the oxygen that is available for the mitochondria and thus the aerobic system is less than the level of oxygen that is required for the aerobic system to cover the full ATP demand. And thus, the aerobic system can fulfill only a portion of the total ATP demand. In simple terms, the aerobic system cannot burn fuel fast enough to provide energy/ATP at the rate that it is needed for that level of muscular work. And this is because the oxygen that is available is insufficient. And remember, the reason why the oxygen availability cannot meet the full oxygen needs is because the rate of oxygen consumption, also known simply as VO2, and therefore oxygen delivery, is not quote-unquote powerful enough to meet the full demand. So this is alluding to the importance of systems like the cardiovascular system, and it is also alluding to improvements in the aerobic system's power that may occur with training. In other words, at this intensity, what if we can provide the mitochondria of muscle cells with more oxygen at a faster rate? How would this white curve change? This is part 2 of this lecture series, so stay tuned for that. Okay, so finally, in box number 3, this gap here between the total aerobic ATP production and the total ATP demand is then fulfilled by what other system? The anaerobic system, which again requires no oxygen and burns fuel fast to provide ATP fast. So what the aerobic system cannot cover, the anaerobic system takes care of. So first, before discussing what goes on inside the muscle cells during post-anaerobic threshold exercise, let's explain what's going on during pre-anaerobic threshold exercise conditions. More specifically, let's discuss what the environment is like within the active muscle cells during pre-anaerobic threshold exercise conditions. So first, what do we know thus far regarding pre-anaerobic threshold exercise? We know by now that the oxygen availability meets the oxygen required by the muscle cells to satisfy the full ATP demand via aerobic ATP production. This is why the area under the curve is completely white in the pre-anaerobic threshold zone, the white representing aerobic ATP production. This means that aerobic ATP production can fulfill the full ATP needs of the muscle cells. Why? Because the level, or more specifically, the rate of oxygen that is needed to meet the full ATP demand aerobically is available for the muscle cells. In other words, the oxygen availability is sufficient for the ATP demand to be fully satisfied aerobically. So what does this mean in terms of fuel metabolism? Remember, fuel molecules provide the energy required to form ATP with, which in turn contains that energy briefly until it is hydrolyzed, which is when that energy can be used to power certain cellular processes like contractions. This process is called oxidation, which is generally described as fuel quote unquote burning. Fuel oxidation is generally describing reactions that break up fuel molecules to release energy, and thus the result of fuel oxidation is ATP production. So what type of fuels do your muscle cells oxidize or burn for ATP production? Your muscle cells can utilize a variety of fuel sources, including carbohydrate, fat, amino acids, and phosphocreatine. But the primary fuel for your muscle cells is carbohydrate. And the two forms of carbohydrate that your muscles can utilize as fuel is 1, glucose, and 2, glycogen. As I taught in exercise physiology, both of these are your endogenous carbohydrate fuel sources. Glucose circulates in the blood, and your muscle cells uptake glucose into the cell to be used as fuel. Glycogen is a large storage form of carbohydrate fuel, largely stored inside muscle cells. Now, you can think of glycogen as multiple glucose molecules bonded together to form a large carbohydrate molecule. Now, the official starting point of carbohydrate oxidation is a molecule called glucose 6-phosphate. That's glucose 6-phosphate. And glucose 6-phosphate is derived from either glucose or glycogen. Glucose undergoes one single fast reaction to be converted to glucose 6-phosphate, while glycogen undergoes a process of degradation called glycogenolysis, lysis of glycogen. And through glycogenolysis, you get multiple glucose 6-phosphates. So in summary, your muscle cells primarily utilize carbohydrates as fuel in the form of glucose or glycogen, which again are sources that provide glucose 6-phosphate, which is the official starting point of carbohydrate oxidation. So when I say carbohydrate oxidation, I am saying glucose 6-phosphate oxidation. Those two terms refer to the same thing. Now in the next slide, you will see a visual of the process of carbohydrate oxidation, but for now, just understand that during pre-anaerobic threshold conditions, because oxygen availability meets the oxygen demand, the glucose 6-phosphate can undergo complete or full oxidation, meaning the glucose 6-phosphate, which again is a carbohydrate, can be completely oxidized, releasing all of its energy. And you get as much ATP out of that glucose 6-phosphate as possible, which is about 32 ATP. This means that full carbohydrate or glucose 6-phosphate oxidation requires aerobic metabolism, which in turn requires, again, oxygen. I'll say that again. This means that full carbohydrate or glucose 6-phosphate oxidation requires aerobic metabolism, which in turn requires oxygen. So if there is sufficient oxygen available, then all the glucose 6-phosphates that are needed can undergo full oxidation. So you can burn the glucose 6-phosphate completely to get as much energy / ATP out of it. So to put this in a very simple example, say you are exercising at a pre-anaerobic threshold intensity that can be placed, say, right here on the curve. And we said that the ATP demand of this intensity can be fully satisfied aerobically, since the oxygen required for this is available. Say to provide the ATP needed, you need 100 glucose 6-phosphates per second. Now, because this is pre-anaerobic threshold conditions and the aerobic system has sufficient oxygen to provide all the ATP needed, all 100 glucose 6-phosphates will be able to undergo complete oxidation. You will get the full 32 ATP complement from each of those glucose 6-phosphates. So what does this imply? This implies that full carbohydrate or glucose 6-phosphate oxidation requires aerobic metabolism. So in summary, during pre-anaerobic threshold exercise, the ATP demand is fully satisfied aerobically, which means that all the glucose 6-phosphates that is needed for the ATP demand can undergo full oxidation. Now, who can remember the first process of carbohydrate oxidation? If you think back to your first lesson on bioenergetics from exercise physiology, you would remember that the first part of glucose 6-phosphate oxidation or carbohydrate oxidation is a short and fast process called glycolysis. Glycolysis is a fast 10 reaction process that occurs outside of the mitochondria in the cell's sarcoplasm and does not, I repeat, does not require oxygen, meaning glycolysis by itself is an anaerobic process. Glycolysis can provide about 2 ATP per glucose 6-phosphate at a fairly fast rate, and this would become important later in discussion. But for now, just know that the major function of glycolysis is to quote-unquote feed the mitochondria with the necessary quote-unquote ingredients for aerobic ATP production. I'll say that one more time. The major function of glycolysis in full carbohydrate oxidation is to feed the mitochondria the necessary ingredients for aerobic ATP production. And those ingredients, which again are the products of glycolysis, are indicated here. And we will review what these are in the next slide. But before moving on, you must understand that full carbohydrate oxidation begins with a short anaerobic process called glycolysis in the sarcoplasm, so outside of the mitochondria, and ends with aerobic processes in the mitochondria. And during pre-anaerobic threshold exercise, all the glucose 6-phosphates that are needed to fulfill the ATP demand can undergo full carbohydrate oxidation. So it will undergo the glycolysis followed by the aerobic process. And this is again only because there is sufficient oxygen available to support that. So here we have an overview of the pre-anaerobic threshold environment in the active muscle cells. First, we have the first part of carbohydrate oxidation, which I indicated was glycolysis, which also I indicated occurs outside the mitochondria and is an anaerobic process. To be specific, this occurs in the sarcoplasm, which is also referred to as the cytoplasm of muscle cells. So because glycolysis is an anaerobic process that occurs outside of the mitochondria, it does not require oxygen. So here we start with the single glucose 6-phosphate, which again is derived from glucose or glycogen. Now this single downward arrow represents the 10 reactions during which time the glucose 6-phosphate is partially oxidized. So this is not full oxidation. This is just partial oxidation. Again, this is called glycolysis. Through glycolysis, you can see that through this partial oxidation, you release energy to form just a couple ATPs. You also have a couple reactions here called redox reactions, and this should be somewhat familiar to you all. This is short for reduction, oxidation, reaction. So to refresh your memories on redox reactions, reduction is when a molecule gains electrons in the form of hydrogens from another molecule. In essence, it steals electrons from other molecules. So when a molecule gains those electrons by stealing it from another molecule, that molecule is reduced. Oxidation refers to when a molecule loses electrons. So reduction and oxidation always go hand in hand. These redox reactions are very important for the oxidation of glucose 6-phosphate. And in order for these reactions to occur, you need an oxidizing agent called NAD, nicotinamide adenine dinucleotide. So NAD will oxidize certain substrates along the glycolysis pathway, meaning NAD will steal electrons in the form of hydrogens from other molecules that are produced along this glycolysis pathway. Specifically, each NAD will gain 2 electrons in the form of 2 hydrogens, each with 1 electron and 1 proton. In so doing, NAD will be reduced to NADH. Now, the reason why the products of this redox reaction is NADH plus a positively charged hydrogen, also known simply as a proton, is because on one of the hydrogens, the electron joins the other hydrogen that is bonded to NAD. So NADH has 2 electrons on the hydrogen, and the other hydrogen is left with no electrons. And this is why we just call this a positively charged hydrogen ion simply a proton, since that is all that it is made up of. So the products of the redox reaction is NADH plus a proton. So during glycolysis, you have 2 redox reactions providing 2 NADH and 2 protons. Now we will discuss in a minute where these go, but for now, let's get to the other products of glycolysis. The end product of glycolysis, as you see here, is pyruvate. A single glucose 6-phosphate ends up as 2 pyruvates. So now, where do these pyruvates go? When we are talking about full carbohydrate oxidation, these pyruvates are quote-unquote fed to the mitochondria for further aerobic oxidation, with the initial reaction converting it to acetyl-CoA. This is a redox reaction, as you can see here, that provides more NADHs and protons. The acetyl-CoA in the mitochondria is then further oxidized through a series of redox reactions in what we call the Krebs cycle. Again, this is all occurring in the mitochondria and thus is an aerobic process. Now, through the Krebs cycle, we get a couple ATPs right here, no big deal, but more importantly, we get more NADHs and protons. And this is again through the series of redox reactions within the Krebs cycle. Now, FYI, the actual number of NADHs is not important for this class. Just know that we get multiple NADHs from the redox reactions through the oxidation of the glucose 6-phosphate, from glycolysis, from the oxidation of pyruvate to acetyl-CoA, from the oxidation of acetyl-CoA through the Krebs cycle. This is where we get a bunch of these NADHs. So now, where do all these NADHs, the electron carriers, and the protons go? They go into the electron transport chain, or the ETC, which is in the mitochondria. And again, this is an aerobic process that requires oxygen. Just keep in mind, anything that occurs inside the mitochondria is an aerobic process requiring oxygen. Now, we won't go over in this class the detailed process of the electron transport chain since we did that in your exercise physiology course, but just know that the NADHs and protons generated from the initial steps of carbohydrate oxidation is fed into the electron transport chain, where it would be used to generate more ATP. For a single glucose 6-phosphate, you get 30 more ATP in the mitochondria through the Krebs cycle and the electron transport chain, which are aerobic processes. So you get 2 from the anaerobic process called glycolysis, plus 30 more here in the mitochondria through aerobic metabolism, providing about 32 total ATP from glucose 6-phosphate. And that is, again, full carbohydrate oxidation. So to summarize, full carbohydrate oxidation starting with the single glucose 6-phosphate, which again is derived from either glucose or glycogen. The first step is glycolysis, which is partial oxidation and does not require oxygen. It is an anaerobic process. Through glycolysis, you get a couple ATP and a couple redox reactions, providing some NADH and some protons. You end up with a couple pyruvates at the end of glycolysis, and with the oxygen that is available, the pyruvates can be further oxidized to acetyl-CoA. Again, with the oxygen that is available, the pyruvates can be further oxidized to acetyl-CoA. The acetyl-CoA can undergo oxidation through the Krebs cycle. And with all the NADHs and protons, the electron transport chain produces the majority of the ATP. Now, as you know by now, once the ATP is produced, whether it's from glycolysis, the Krebs cycle, or the electron transport chain, it is going to be hydrolyzed. Remember, ATP is not meant for storage. It doesn't hang around for a long period of time. Once it's produced, it will almost immediately be hydrolyzed. So in muscle cells during exercise, the ATP could be hydrolyzed on that myosin head to power muscle contractions. As we discussed towards the beginning of this lecture, we indicated that ATP hydrolysis involves the splitting off of that last phosphate through that nucleophilic attack to release the energy contained in that last phosphate bond. And as a product of ATP hydrolysis, you have the inorganic phosphate, which broke off of ATP. You have ADP, adenosine diphosphate, because that last phosphate broke off. And you have a free proton, which came off of a water molecule. And this is why we went over ATP hydrolysis in much detail in the beginning of this lecture series, so you understand where all this comes from. Now, during pre-anaerobic threshold conditions, all these products can be used by the mitochondria. So what we can take away from this overview is that everything is balanced and cycled. All the products from glycolysis, which include NADHs and protons and pyruvate, are all utilized in the mitochondria for aerobic ATP production. All the products of ATP hydrolysis, which include the inorganic phosphate, ADP, and proton, are also utilized in the mitochondria for aerobic ATP production. In essence, nothing is accumulating in the sarcoplasm. The reason why the mitochondria can utilize all these products during pre-anaerobic threshold exercise is because of sufficient oxygen availability. And that again is what characterizes pre-anaerobic threshold conditions. So because there is sufficient oxygen availability, the mitochondria has enough power to take all these products from ATP hydrolysis and glycolysis and use it to form ATP. So this is the pre-anaerobic threshold environment within active muscle cells. Nothing is accumulating and there's sufficient oxygen available so that the mitochondria has enough quote-unquote power to utilize all these products from glycolysis and ATP hydrolysis to be used to produce ATP aerobically. And to reiterate the main points, during pre-anaerobic threshold exercise, the protons released from redox reactions like in glycolysis, the oxidation of pyruvate, and also in the Krebs cycle, as well as protons released through ATP hydrolysis, are all cleared out of the sarcoplasm by the mitochondria. The inorganic phosphates, also from ATP hydrolysis, is also cleared out by the mitochondria, and all of these are used to resynthesize ATP in the mitochondria, primarily through the electron transport chain. During pre-anaerobic threshold exercise, there really is no excess or accumulation of any of these products, and this explains why there is no fatigue during pre-anaerobic threshold exercise. Now, this will all make sense as we move forward, but just know that during pre-anaerobic threshold exercise, we do not experience fatigue. There is no muscle inhibition. Our muscles are not pumping its brakes. This is why we call pre-anaerobic threshold exercise steady state exercise, because we can sustain the intensity of exercise for a long duration simply because we don't fatigue. As I said, you will have a better understanding of this as we move forward. But yes, the fact that free protons and inorganic phosphates specifically do not accumulate in the sarcoplasm and are cleared out by the mitochondria for aerobic ATP production is an important factor to remember. Now that we've discussed the myocellular environment during pre-anaerobic threshold exercise, let's now move on to the post-anaerobic threshold environment. The post-anaerobic threshold zone is highlighted within the red box as we see here. And as we went over earlier, the exercise intensities above the anaerobic threshold involve a situation in which the ATP demand cannot be fully satisfied by the aerobic system. Now, at this point in this lecture series, you should be certain of that knowledge. As you can see in the figure, the aerobic ATP production continues to contribute to the rising ATP demand above the anaerobic threshold. And as intensity increases further past the anaerobic threshold, all the way up to maximum intensity, the aerobic system continues to increase the rate of ATP production as much as possible. At maximum intensity, reaches its maximum ATP producing capacity, and this is referred to as the aerobic capacity. As I mentioned briefly earlier, aerobic capacity is quantified using a measure called VO2 max, or maximum oxygen consumption rate. At this point, this should make sense to you as to why the maximum oxygen consumption rate reflects the maximum ATP producing power of the aerobic system. It is again because aerobic ATP production relies on the availability of oxygen. So consumption of oxygen, which includes delivery and uptake into the muscle's mitochondria, is obviously an important limiting factor for aerobic ATP production. So yes, we can use oxygen consumption rate, or VO2, to indirectly measure aerobic ATP production. And the maximum oxygen consumption rate, or VO2 max, is reflective of the maximum aerobic ATP producing capacity, aka aerobic capacity. So during post-anaerobic threshold exercise, we know that the aerobic system cannot fully meet the ATP demand because the oxygen that is required to do so is not sufficiently available. I will say that one more time. Please take note. During post-anaerobic threshold exercise, we know that the aerobic system cannot fully meet the ATP demand because the oxygen that is required to do so is not sufficiently available. In simple terms, during post-anaerobic threshold exercise, the oxygen availability does not meet the oxygen demand. Now, please keep this in mind as this is related to fatigue. More on that later. All right, so since the aerobic system cannot fully cover the ATP demand of the muscle during post-anaerobic threshold exercise, we said that the anaerobic systems back it up and fulfills the portion of ATP demand that the aerobic system cannot cover. And this is represented by the gray area under the curve. Now, it wouldn't make sense for the muscle cells to use a backup system that requires oxygen when the reason why the aerobic system cannot fully meet the ATP demand is because of insufficient oxygen availability. So this portion of the ATP demand that cannot be satisfied aerobically can be described as the energy void. This is the gap between the energy or ATP that is needed and the ATP that is provided by the aerobic system. Therefore, the energy void is the portion of the ATP demand that is fulfilled anaerobically without oxygen. That is represented by, again, this gray area under the curve. The energy void can also be seen as an oxygen void because, as we said earlier, in the post-anaerobic threshold zone, the oxygen demand is up here on top of the entire curve, but the oxygen availability is represented down here on top of the white curve. So this energy void is the portion of the oxygen demand that is not available. And thus the energy void is filled by the anaerobic systems that don't require oxygen to produce ATP. Now, one of the key characteristics of the anaerobic systems, other than the fact that they do not require oxygen to produce ATP, is that they produce ATP at a very fast rate. They do not require a lot of reactions to generate ATP. Anaerobic ATP production does not occur in the mitochondria, so it does not require processes like the Krebs cycle or the electron transport chain. Anaerobic metabolism occurs in the sarcoplasm, which is, again, the cytoplasm of the muscle cell. Now, the reason why the muscle cell during post-anaerobic threshold situations rely on fast ATP producing systems to back up the aerobic system is because, again, it's not just about the total amount of ATP, but rather the rate of ATP production. So when I say the ATP demand is very high, I am saying that the rate at which ATP is needed is very high. During post-anaerobic threshold exercise, your muscle cells have a relatively high ATP demand, meaning it needs ATP at a very fast rate. So when I say the aerobic system during post-anaerobic threshold exercise cannot fully satisfy the ATP demand, I am essentially saying it can't produce ATP at the rate that it is needed. So this is why the muscle cells initiate fast ATP producing anaerobic systems to back up the aerobic system to cover this energy void, and in essence boost the total ATP production rate to meet the high rate of demand. Now, as a review of exercise physiology, what are your two anaerobic systems? Which two ATP producing systems are your backups to the aerobic system during post-anaerobic threshold exercise? Which two anaerobic systems fill this energy void? The anaerobic systems include the lactic system, aka fast glycolysis, and the alactic system, aka the phosphagen system. These two systems, again, do not require oxygen and produce ATP at a fast rate. And again, they only exist to back up the aerobic systems during situations in which the oxygen availability does not meet the oxygen demand for the aerobic system to fully meet the ATP demand. So here is a sample exam question. Which of the following describes the function of the anaerobic systems? Again, which of the following describes the function of the anaerobic systems? The possible correct answers include 1, to fulfill the portion of the ATP demand that the aerobic system cannot. 2, to help the aerobic system meet the rate at which ATP is needed during post-anaerobic threshold exercise. Or 3, to fill the energy void. I'll go ahead and repeat that. The possible correct answers include 1, to fulfill the portion of ATP demand that the aerobic system cannot. 2, to help the aerobic system meet the rate at which ATP is needed during post-anaerobic threshold exercise. Or 3, to fill the energy void. Essentially, those three answer choices all pretty much say the same thing. Now, before moving forward, I want you to take note of the fact that since we have the anaerobic backup systems, the ATP demand, regardless of exercise intensity, is always fully satisfied. During pre-anaerobic threshold exercise, the ATP demand, as reflected on top of this curve, is satisfied aerobically. As you can see, the area under the entire curve during pre-anaerobic threshold intensities is white, which means the ATP demand during pre-anaerobic threshold intensities is fully satisfied through aerobic ATP production. During post-anaerobic threshold exercise, the ATP demand, again, is fully met, but it's satisfied both aerobically and anaerobically. So there is no situation during exercise when the ATP demand is not met, unless there is, of course, exhaustion, which we defined earlier as when intracellular fuel sources are completely depleted. But this would take obviously a prolonged period of exercise in order for that to happen. But other than that, there are no circumstances when ATP demand is not fully satisfied, regardless of whether you are jogging or doing a full-on sprint. ATP demand is always met. And once again, during post-anaerobic threshold exercise, the full ATP demand is met both aerobically and anaerobically. The anaerobic portion simply backing up the aerobic portion so that the full ATP demand can be met. So what this means is that acute muscular fatigue is not, I repeat, not due to the muscle cell's inability to meet the ATP demand. First, let's again define acute muscular fatigue as we did in the beginning of this lecture series. Acute muscular fatigue is a situation in which muscle contractions and therefore muscular force production is temporarily inhibited due to the presence of a stressor during heavy exercise or high to maximum intensity exercise. In other words, during post anaerobic threshold exercise intensities. To put it into simple terms, acute muscular fatigue is observed when you are running and you cannot sustain a certain speed for very long, no matter how hard you try, because something is inhibiting your muscles. You feel as if something is pumping your muscles' brakes. We obviously experience this anytime we perform an all-out sprint. We cannot sustain peak speed during a sprint for very long, maybe only a couple seconds until we start to decline in speed. That subsequent decline in speed is due to acute muscular fatigue. When you perform some bicep curls, you know that as you perform one rep after another, the curls become harder and harder. You know that the force production after each rep declines until you can't lift that dumbbell anymore. This is acute muscular fatigue. So again, acute muscular fatigue is the decline in muscular contractility and force production during post-anaerobic threshold exercise. So simply put, acute muscular fatigue is therefore observed during exercise intensities within this zone highlighted within the red dotted lines. Now, how else can you frame this? Acute muscular fatigue is observed when there is an energy void, or acute muscular fatigue is observed when the anaerobic systems are required to help the aerobic system meet the full ATP demand. And lastly, and most critically, acute muscular fatigue is observed when the oxygen availability does not meet the oxygen required for the aerobic system to fully satisfy the ATP demand. I'll say that last one more time. Acute muscular fatigue is observed when the oxygen availability does not meet the oxygen required for the aerobic system to fully satisfy the ATP demand. Of course, that describes the post-anaerobic threshold environment. Remember, the ATP demand is up here on top of this entire curve, but the oxygen availability is down here represented on top of the white curve. In other words, this situation, post-anaerobic threshold, where the oxygen availability does not meet the oxygen demand, is a hypoxic situation. Once again, this situation, post-anaerobic threshold exercise, where the oxygen availability does not meet the oxygen demand, is a hypoxic situation. This is hypoxia within the muscle cells. Most people would say that hypoxia is when there is zero oxygen. That is true, but not entirely accurate. Hypoxia is when the oxygen availability does not meet the oxygen demand. So say we are resting and we know our cells inside our body are producing ATP to keep the cells functioning and our body alive. And as I emphasized earlier, the body's primary bioenergetic or ATP producing system is aerobic metabolism. Not every cell in the body has anaerobic metabolism, and this is why we need oxygen to survive. So during resting situations, we have resting oxygen consumption, and the cells of our body have sufficient oxygen to produce ATP aerobically. Now, what happens if we say block oxygen consumption? Say we are choking on something and our airway is restricted and oxygen is not getting into our blood circulation. Now, at this point, the cells of our body that rely on aerobic ATP production to function and to essentially stay alive are deprived of oxygen. So then what happens? Well, the oxygen available to our cells do not meet the oxygen that is required for the cells to meet its resting ATP needs. So if this continues too long, what happens? The cells eventually die because they cannot produce ATP since there is insufficient oxygen. In other words, the cells die due to hypoxia. So would you say that hypoxia is a stressor to the cells and to the body in general? Of course it is, since hypoxia is conducive to death. Now let's apply this concept to a situation in which one is exercising above the anaerobic threshold. Let's just say one is performing a Wingate test, which is a test involving 30 seconds of all-out effort cycling. During the all-out test, the subject is clearly in the post-anaerobic threshold zone. We know that the aerobic system is trying as hard as it can to meet the ATP demand of the muscle cells, but it can't because the oxygen that is required to do so is insufficiently available. Meaning there is a very high oxygen demand at this point, but there's only this much oxygen available to support aerobic ATP production. So in summary, the oxygen availability in those muscle cells do not meet the oxygen demand of those muscle cells. This is also hypoxia. Just like how the body's cells experience hypoxia when one's airway is restricted, the muscle cell during this sprint exercise is undergoing a similar situation. Obviously, the hypoxia is under different circumstances, but it is the same conceptually. The oxygen availability does not meet the oxygen demand. When you are choking and the airway is restricted, the cells in the body do not have enough oxygen to meet the demand for aerobic ATP production. When you are exercising post-anaerobic threshold, like during the Wingate test, your muscle cells do not have enough oxygen to meet the demand for aerobic ATP production. So in other words, it doesn't matter what the oxygen demand is. If the oxygen availability does not meet that demand, there is hypoxia. I'll say that one more time. It doesn't matter what the oxygen demand is, whether you are resting or you are sprinting. If the oxygen availability does not meet that demand, there is hypoxia. Now, whether this was a part of human evolution or not, skeletal muscle is unique in that it has anaerobic metabolism. Our muscle cells have the ability to back up the aerobic system and produce ATP without oxygen. Regardless, as I emphasized earlier, our muscle cells never, ever rely on anaerobic metabolism for its full ATP needs. There is never a situation in which your muscle cells can survive off of anaerobic metabolism. Why? Again, because the aerobic system is the primary energy system for all cells in the body, including your muscle cells. So with that said, during a post-anaerobic threshold situation, the muscle cells are therefore under hypoxia. In other words, the active muscle cells during post-anaerobic threshold exercise are under a hypoxic stress, stress being the key word. Active muscle cells during post-anaerobic threshold exercise are under a hypoxic stress. So the stress of post-anaerobic threshold exercise is not that the ATP demands are not met. As I said, the ATP demands are fully met because we have anaerobic backup systems to help out the aerobic systems. So if I ask, which of the following describes the stress that muscle cells experience during post-anaerobic threshold exercise, the answer is not the inability to meet the full ATP demand. Again, the answer is not the inability to meet the full ATP demand. The correct answer is hypoxia. The oxygen required for aerobic systems to meet the full ATP demand is not fully available. Now, what do we know about the human body and its physiology? Our physiology is made to survive. This means when there is a stressor present, the body, more specifically the cells experiencing the stress, will respond in a way to mitigate that stress. In other words, our cells have acute or immediate stress response mechanisms. So if you are exercising at a post-anaerobic threshold intensity, such as during a Wingate sprint, what do you think would be the acute stress response to mitigate the stress of hypoxia? You don't even need to think too hard about this. What would be the most logical way for the muscle to deal with this hypoxic stress? It would be to decrease the ATP demand and thus the oxygen demand of the muscle cells. How would it do that? By pumping its brakes, by slowing it down. In other words, by inhibiting the process that requires ATP during exercise, which is muscle contractions. What is this referred to as? It's referred to as fatigue, more specifically, acute muscular fatigue. Acute muscular fatigue is essentially an acute stress response to hypoxia. Through fatigue, the muscle gets itself out of the post-anaerobic threshold zone and thus gets out of hypoxia. Essentially, what fatigue does is take you from a post-anaerobic threshold workload and forces you into a pre-anaerobic threshold workload. I'll say that one more time. Essentially, what fatigue does is take you from a post-anaerobic threshold workload and forces you into a pre-anaerobic threshold workload through the inhibition of muscle contractions. By the muscle inhibiting its own contractions, the muscle is ultimately reducing the ATP demand and thus oxygen demand so that the muscle can be in an environment in which the oxygen availability meets the oxygen demand, a non-hypoxic environment. So based on this figure, if you are exercising here, fatigue is going to force you to exercise here. And as you know, fatigue overrides your effort, meaning no matter how hard you are trying to sustain the peak workload of an all-out effort sprint, fatigue will force the muscle to reduce the workload to a pre-anaerobic threshold, no matter how much effort you are putting out. So if you remember the Wingate test, as you ramp up, you will reach peak RPMs within about 5 seconds. And after 5 seconds, your power output starts to decline due to fatigue as an acute or an immediate response to a hypoxic stress. Now, if you remember, RPMs during this 30-second test was measured. And within that 30 seconds, you fatigue and your RPMs rapidly decline. But then the RPMs eventually stop declining and you are cycling at a constant RPM for the last few seconds of the test. Why isn't the RPMs continually declining? Well, because at this point, you are now fatigued down to a pre-anaerobic threshold intensity. And now there's no more hypoxia and thus no more fatigue. In essence, fatigue forces you into a steady state exercise intensity or workload, which is right at or before your anaerobic threshold. Again, fatigue is taking you from here and forcing you here through muscle inhibition. All right, so in summary, acute muscular fatigue is a forced reduction in exercise workload during post-anaerobic threshold situations through the inhibition of muscle contractions in a response to a hypoxic stress in the active muscle cells. In so doing, the muscle reduces the ATP and thereby oxygen demand of the muscle, thus diminishing hypoxia. Acute muscular fatigue can be described as an acute stress relief mechanism. What this also means is that fatigue during post-anaerobic threshold exercise workloads is not due to the depletion of fuel or ATP. Fuel does not deplete fully within the few seconds you are sprinting or performing resistance exercise reps, and ATP demand is always met even during post-anaerobic threshold exercise, since we have anaerobic backup systems to help out the aerobic systems. So if I asked a true and false question on the exam to the effect of an acute decrease in muscular force output during all-out effort exercise is due to insufficient ATP, this is a false statement. If the statement said instead that it is due to an inhibition of muscle contraction in response to a hypoxic stress, then it would be true. Now the question is, what is the mechanism connecting this hypoxic myocellular environment to the inhibition of the muscle cells during post-anaerobic threshold exercise workloads? To answer this, we need to understand how fuel metabolism changes during post-anaerobic threshold and thus hypoxic situations. Earlier, we discussed the nature of carbohydrate oxidation during pre-anaerobic threshold exercise. And I said that glucose 6-phosphate, which again is derived from your two carbohydrate sources, glucose or glycogen, can undergo full oxidation because full oxidation requires aerobic metabolism. And since during pre-anaerobic threshold exercise there is sufficient oxygen availability to meet the demand, the glucose 6-phosphate needed to meet the ATP demand is fully oxidized, and you get as much ATP out of it as possible. But what if now oxygen demands are so high that the oxygen availability is insufficient and your muscle cells are now under a hypoxic situation? How does carbohydrate oxidation change during these situations? And how is it linked to acute muscular fatigue? Now in this slide, we can see the changes in carbohydrate oxidation from a more visual perspective. So first, let's turn our attention to the labeling on the left. As you see here, full carbohydrate oxidation, so full oxidation of glucose 6-phosphate, initially involves an anaerobic process that provides 2 to 3 ATP from the partial oxidation of glucose 6-phosphate, meaning through this initial step from the breakdown of glucose 6-phosphate all the way to pyruvate through roughly 10 reactions, 2 to 3 ATP's worth of energy is released, and thus 2 to 3 ATP are formed. This initial step right here is called glycolysis, and glycolysis is an anaerobic system and thus occurs outside the mitochondria and does not require oxygen for these reactions to occur. So the 2 to 3 ATP that is produced during glycolysis is done so anaerobically. So during glycolysis, again, the glucose 6-phosphate is partially oxidized down to pyruvate. At this point, there is a junction. The pyruvate can be further oxidized in the mitochondria through aerobic metabolism, or it can be simply converted to lactate. Pyruvate, as indicated earlier, is a partially oxidized carbohydrate, meaning it's a product of glycolysis, which is a partial oxidation of glucose 6-phosphate. This means pyruvate still has energy remaining to form ATP with. So how is this energy harnessed for more ATP production? It is harnessed through further oxidation in the mitochondria, through aerobic metabolism. And through aerobic metabolism in the mitochondria, there is 30 more ATP produced. This means this pyruvate has about 30 ATP's worth of energy still remaining. Thus, if you do the simple math, a single glucose 6-phosphate, which is derived from glucose or glycogen, your two endogenous carbohydrate sources, you get a total of 32 to 33 ATP through its full oxidation, which again initially involves glycolysis, an anaerobic process, then aerobic metabolism in the mitochondria. So once aerobic metabolism is completed, the glucose 6-phosphate is then completely oxidized. So here is the first fundamental question. During pre-anaerobic threshold exercise, which of the following describes carbohydrate oxidation? The answer will be something to the effect of the glucose 6-phosphate required for the ATP demand can undergo full oxidation, which involves again glycolysis followed by aerobic metabolism in the mitochondria. If there was a follow-up to this question asking why, it would be because there is sufficient oxygen available to support the full oxidation of the glucose 6-phosphates, since full oxidation involves aerobic metabolism. Okay, so now what about during post-anaerobic threshold exercise? How does this dynamic of carbohydrate oxidation change? At this point, during post-anaerobic threshold situations, carbohydrate oxidation is ramping up to help meet the ATP demand that is rising. But here is the thing. Now there is insufficient oxygen to allow for all the glucose 6-phosphates to undergo full oxidation. But which part of this entire pathway is affected by this? Glycolysis is not affected meaning glucose 6 phosphate can undergo glycolysis and not be limited by oxygen availability because, again, it is an anaerobic process. So you can get 2 to 3 ATP per glucose 6 phosphate through glycolysis without the need of oxygen. The part that is affected during post-anaerobic threshold hypoxic situations is the second step of carbohydrate oxidation, which is aerobic metabolism, since now this is limited by the oxygen that is available. So as glucose 6 phosphate undergoes this entire full oxidation pathway from glycolysis to aerobic metabolism, the mitochondria gets to a point where it does not have enough oxygen to quote unquote process further pyruvate, meaning the mitochondria doesn't have enough quote unquote power to process all this pyruvate coming out of glycolysis, which at this point during post-anaerobic threshold exercise is ramping up. So now there is more pyruvate than the mitochondria can handle due to the insufficient oxygen availability. Remember, the mitochondrial power is dependent on the oxygen that is available. This, in essence, creates a quote-unquote bottleneck effect with pyruvate accumulating right before the mitochondria. Now, most of the pyruvate undergoes further oxidation in aerobic metabolism, but an increasing portion of the pyruvate cannot undergo further oxidation in aerobic metabolism. So what happens to that pyruvate? That portion of pyruvate that cannot get further oxidized in the mitochondria is simply diverted into another pathway, which is its conversion to lactate through one single reaction. Now, this pathway from glucose 6 phosphate all the way to lactate is partial oxidation and is referred to as the lactic system or fast glycolysis. So if glycolysis ends with lactate, meaning pyruvate is diverted to its conversion to lactate instead of further going oxidation in the mitochondria, this is referred to as fast glycolysis. This again is an anaerobic process, so it is not affected by insufficient oxygen availability. Now let me put this into a hypothetical yet practical example to help you visualize all this, because I know this can be a bit complex. Now please take note of this example. Say you are exercising at a pre-anaerobic threshold workload. Say to meet the ATP demand, your muscles needs to fully oxidize 100 glucose 6 phosphates per second. Because the oxygen availability meets the oxygen required to fully oxidize the 100 glucose 6 phosphates per second, all 100 glucose 6 phosphates are fully oxidized per second. And you get 32 to 33 ATP out of each of those glucose 6 phosphates per second. Now, let's say you increased your exercise workload and now your muscles are in a post-anaerobic threshold state and thus a hypoxic state. And now the ATP demand is a lot higher, and now you need to fully oxidize 300 glucose 6 phosphates per second to meet that ATP demand. However, the oxygen that is available does not meet the required amount of oxygen needed to fully oxidize 300 glucose 6 phosphates per second. Your muscle cells only have enough oxygen to fully oxidize 200 glucose 6 phosphates per second. So now there is an energy void, and your muscle cells need to rely on an anaerobic system to cover the remaining ATP demand. So now the muscle cells ramp up glycolysis even further to help meet the full ATP demand. And it is able to because glycolysis, again, is not limited by oxygen. And because the mitochondria at this point are overwhelmed, the additional pyruvate coming out of glycolysis that cannot enter aerobic metabolism is simply converted to lactate. Now, in addition to this, the muscle cell also utilizes other anaerobic systems to help out, like the phosphagen system. So you have fast glycolysis And you have the phosphagen system helping out the mitochondria meet the full ATP demand of the muscle cells during post anaerobic threshold exercise workloads. So, in summary, if I ask the question, which of the following is characteristic of carbohydrate oxidation during post anaerobic threshold exercise, you have all of these answers being correct. 1. Increasing full carbohydrate oxidation with greater increases in partial oxidation. This means 2. There is increased fast glycolysis or lactic system activity. And thus, 3, there is increased lactate production. So, in summary, during post anaerobic threshold conditions, most of the carbohydrates, so most of the glucose 6 phosphates available, undergoes full oxidation. So they go through this entire pathway with the oxygen that is available. But a portion stops with glycolysis. It stops right here and ends here because the aerobic system, aka the mitochondria, is overwhelmed, again, due to insufficient oxygen availability. So the question is now, why lactate? Why does the additional pyruvate that cannot be further processed through aerobic metabolism convert to lactate? Now, the reason I want to talk about lactate is because I want to dispel common myths associated with lactate, namely lactate being simply a metabolic waste product and causes fatigue when it accumulates. So let's discuss the purpose of lactate formation and what it is used for. But before moving forward, keep this in mind. Lactate is simply a result of carbohydrate or glucose 6 phosphate oxidation cut short, meaning lactate is a result of a glucose 6 phosphate molecule oxidized to a point where only 2 to 3 ATPs worth of energy was released. This means that lactate still has a lot of energy left in it. More specifically, each lactate has roughly about 30 ATPs worth of energy remaining. Now, how do we get that 30? Well, if pyruvate did not convert to lactate and was further oxidized in the mitochondria through aerobic metabolism, you would get 30 more ATP out of it, like we said earlier. So if lactate reconverted back to pyruvate and pyruvate was further oxidized in aerobic metabolism, 30 more ATP would be produced. Therefore, lactate has about 30 ATPs worth of energy still remaining. Now, just FYI, it's not exactly 30 ATP, but it's about 30 ATP. Now, either way, this should make sense to you when I say that lactate is not a metabolic waste product. It is an actual usable fuel molecule. It's a partially broken down carbohydrate molecule. So what we know generally about biological organisms is that fuel is the key to survival. And us, as sophisticated and energy costly organisms, our body will not waste fuel. This is why we love to store excess fuel, especially in adipose. And this is why we get fatter when we overfuel our body, because our body is storing that excess fuel. So if lactate is a usable fuel from a biological perspective, do you think your body will see it as simply a waste product and get rid of it? Absolutely not. Your body will always find a way to either store or utilize fuel molecules. It will not just waste it and throw it away. That will not be conducive to survival. In fact, about 80% of the lactate that is formed during exercise is used eventually as fuel for ATP production in the mitochondria after it is converted back to pyruvate. The other 20% is used to recycle back to glucose 6 phosphate and eventually back to glucose, and this occurs in the liver. This is called the Cori cycle, which we talked about in your exercise physiology course. So no, lactate is not simply a waste product. It is a usable source of fuel. So I wanted to dispel that myth first. The next thing I want to discuss is why lactate. Why does pyruvate that cannot undergo further aerobic metabolism convert to lactate? So again, the fundamental question we want to answer is, why do we form lactate during post-anaerobic threshold exercise? As we indicated earlier, and is important for you to know, most of the glucose 6 phosphate undergoes full oxidation. They undergo glycolysis and provide 2 to 3 ATP and a couple pyruvates. The pyruvates then undergo further oxidation down to acetyl-CoAs, which then proceed for further oxidation in the mitochondria through aerobic metabolism. This is the entire full oxidation pathway. And again, most of the glucose 6 phosphate during post-anaerobic threshold exercise undergo this full pathway with the available oxygen. Now pay attention to this reaction right here. Part of the reactions involved in glycolysis are redox reactions, reduction oxidation reactions, which we introduced earlier. Again, the redox reactions involve NAD oxidizing substrates which are produced along the glycolysis pathway. In so doing, NAD gains electrons from those substrates in the form of 2 hydrogens, thereby being reduced to NADH. The electron from one of the hydrogens, as I said earlier, goes onto the hydrogen on NADH. And thus, that hydrogen is left being a positively charged hydrogen ion and simply called a proton. Now, also, as I said earlier, the NADHs and the protons are utilized in the mitochondria for aerobic ATP production. But what I didn't mention earlier was that this redox reaction is necessary for glycolysis to occur. This means there needs to be an adequate supply of NAD to keep glycolysis going. Normally, the NAD is provided by the mitochondria after the NADH is oxidized back to NAD. However, during post-anaerobic threshold and thus hypoxic situations, the mitochondria and thus the aerobic system is overwhelmed to the point it cannot process any more of the NADHs, the protons, and the pyruvates, or the acetyl-CoA that is derived from the pyruvates. This again is largely due to limited oxygen availability and therefore the limited ATP producing quote-unquote power of the mitochondria. And as I said earlier, the muscle cell will therefore ramp up glycolysis so it can get the 2 to 3 ATP very fast from the glucose 6 phosphates that are available to help meet the ATP demand. But in order for glycolysis to increase its activity, there needs to be a fast supply of NAD because, as I said, those redox reactions here are necessary for glycolysis to proceed, especially at a fast rate. Now remember, I said in the prior slide that the main provider of the NAD is the mitochondria after it oxidizes NADH back to NAD. But now the mitochondria can't take in any more of the NADH that is produced from this redox reaction. And so the question is, where does the muscle cell get the NAD from at the fast rate it needs it? It gets it from this reaction right here. Pyruvate can be reduced by NADH, meaning it can pull the electrons from NADH and thus NADH is quickly oxidized back to NAD. And the quick supply of NAD through this reaction can be used to keep glycolysis going so that the ATP demand can be met. So when the pyruvate is reduced, it is converted to lactate. And this is catalyzed by the enzyme lactate dehydrogenase. And this ultimately is the main reason why lactate is formed during post-anaerobic threshold exercise. Now, another interesting aspect of lactate is that it has been shown to be a buffer, albeit a weak buffer, to rising proton levels within the cell's sarcoplasm. So it also helps neutralize the pH in the sarcoplasm as protons start to build up in the sarcoplasm, which can cause acidification. Now, the reason I wanted to bring this up is because many people believe that lactate is what causes acidification and therefore fatigue. And many people associate lactate buildup with that burning sensation you feel during heavy exercise. As I alluded to before, this is false. And with the information I just provided, we see that lactate is actually adverse to acidification, not conducive to acidification. It actually absorbs the protons as opposed to releasing protons and causing acidification. So when people say something to the effect of lactic acid buildup causes fatigue and pain during heavy exercise, you should know now that is not factual. We will discuss this a little bit further as we proceed along. So to reiterate why lactate is formed during post-anaerobic threshold exercise, it is 1, to provide NAD at a fast rate to support ATP production via glycolysis, more specifically fast glycolysis or the lactic system. 2, because the mitochondria cannot process more pyruvate and thus acetyl-CoA, therefore a large portion of pyruvate is reduced to lactate, which is related to the first point because the reduction of pyruvate to lactate is what provides the NAD for glycolysis to keep going. 3, lactate serves partly to buffer the rising levels of protons in the sarcoplasm and thus helps neutralize muscle pH. And 4, lactate is a fuel molecule that essentially contains the remaining energy until it can be used later. Remember, it is a usable fuel molecule with about 30 ATPs worth of energy still remaining. That 30 ATP can be later harnessed when lactate is reconverted back to pyruvate, and pyruvate can get further oxidized to acetyl-CoA, and acetyl-CoA can undergo further oxidation through aerobic metabolism. So with that said, we know that lactate is not what connects the hypoxic stress during post-anaerobic threshold exercise to the fatigue that is experienced during post-anaerobic threshold exercise. Many people believe that lactate is the direct cause of fatigue just because it is produced during situations when you fatigue. As we know now, lactate is simply produced because pyruvate is reduced to lactate to provide the NAD that is needed to keep glycolysis going so that glycolysis can help meet the ATP demand of the muscle and help satisfy the portion of the ATP demand that the aerobic systems cannot. So if it is not lactate buildup that causes fatigue, what then is the mechanism that connects the hypoxic stress during post-anaerobic threshold exercise to acute muscular fatigue? In other words, what is stimulating the muscle inhibition during a hypoxic post-anaerobic threshold situation? So first, let's refresh our memories on the state of the muscle cells during a pre-anaerobic threshold state, a non-hypoxic state. Remember, during pre-anaerobic threshold exercise, there is sufficient oxygen available to allow for full oxidation of glucose 6-phosphate. The mitochondria has sufficient oxygen to fully process all the products of glycolysis, which include the pyruvates, the NADHs, as well as the protons. It also has the ability to process all the products of ATP hydrolysis, which includes the ADP, inorganic phosphates, and the protons again. As you can see, nothing is accumulating in the sarcoplasm, especially the protons and the inorganic phosphates. The mitochondria has enough quote-unquote power to clear all of it out of the sarcoplasm to be used for aerobic ATP production. But what happens to all of this during post-anaerobic threshold hypoxic situations, like during heavy to max intensity exercise? So as I emphasized earlier, during post-anaerobic threshold exercise, the oxygen that is available does not meet the rising oxygen demand. So therefore, the mitochondria and thus aerobic systems do not have enough power to meet the full ATP demand. This means that the mitochondria is quote-unquote tapped out and cannot process all the products coming out of the sarcoplasm. First, we have glycolysis ramping up to produce 2 to 3 ATP quickly to help meet the ATP demand. And as we said earlier, the excess pyruvates from glycolysis cannot be further oxidized in the mitochondria because, again, the mitochondria is quote-unquote tapped out due to the limited oxygen that is available. It doesn't mean that none of the pyruvates are getting through. It's just that an increasing portion of the pyruvates cannot go through because of the limited oxygen availability. So the excess pyruvate, as you see here, is instead reduced to lactate. And this is why lactate levels start to rise at your anaerobic threshold intensity and rises exponentially as you increase your intensity further past the anaerobic threshold. Now, as we said earlier, this reaction involves the quick oxidation of NADH back to NAD to keep glycolysis running at a fast rate. Now, because the muscle cells rely on glycolysis to help meet the very high ATP demand, this redox reaction is also occurring at a very high rate. And as we said, one of the products of this redox reaction is the free proton. Now, lactate can absorb this free proton, but only to an extent. At intensities right at your anaerobic threshold, the lactate can sufficiently absorb these protons and buffer it adequately. However, as intensity increases further away from the anaerobic threshold towards maximum intensity exercise, lactate cannot absorb and buffer the rapid production of these free protons coming from the rapid redox reactions. Therefore, as you get towards maximum intensity exercise, these protons start to accumulate within the muscle cell's sarcoplasm. Remember, normally these protons would be cleared out by the mitochondria and used in the electron transport chain. But because there's insufficient oxygen for the mitochondria cannot clear out the protons at the rate they are being produced. And as I said, the lactate can only absorb so much of it. So again, because of all that, they begin to accumulate as intensity increases post-anaerobic threshold. Also during post-anaerobic threshold exercise, ATP hydrolysis is occurring at a very high rate since there is high muscle contraction activity. ATP hydrolysis can proceed at a high rate because, again, we have rapid ATP production from your anaerobic systems, which include fast glycolysis and the phosphagen system. Not to mention the ATP still coming out of the mitochondria from aerobic metabolism, which at this point is at its capacity. This rapid ATP hydrolysis would result in rapid release of inorganic phosphates and protons, since these are the products of ATP hydrolysis. Most of these are again cleared out by the mitochondria, but not at the rate that they are being produced. So again, the protons as well as the inorganic phosphates start to accumulate within the muscle cell's sarcoplasm. So in summary, because the mitochondria does not have enough power to process all these products, especially from glycolysis and ATP hydrolysis, due to limited oxygen availability, these products, namely the protons and inorganic phosphates, start to accumulate in the cell's sarcoplasm. So for the exam, you need to understand the contents of this slide and know what the state of the muscle cell is during post-anaerobic threshold hypoxic exercise situations. So how does the accumulation of protons and inorganic phosphates cause fatigue? How does it cause the muscle inhibition to relieve the muscle of this hypoxic stress during post-anaerobic threshold exercise? Let's go ahead and take a look at this figure here. So here we have post-anaerobic threshold exercise. There is an energy void and an increase in anaerobic ATP production to fill that void to help the aerobic system meet the full ATP demand. The reason why the aerobic system cannot meet the full ATP demand is because the oxygen required to do so is not sufficiently available. Thus, the muscle cells are under a hypoxic stress. So now the muscle needs to respond and pump its own brakes to get itself out of this hypoxic stress by reducing the ATP demand and thus the oxygen demand. It will do this by inhibiting muscle contractions. So how does it do this? So during this situation, there is a ramping up of ATP hydrolysis due to the rapid production of ATP, namely through the anaerobic systems. Fast glycolysis, which is partial carbohydrate oxidation, as well as the phosphagen system, not to mention the ATP that is produced still by the aerobic system. There is also rapid redox reactions that are necessary to keep glycolysis running. As a result, there is a rapid release of protons and inorganic phosphates in the sarcoplasm as we depicted in the last slide. Now again, the mitochondria at this point cannot process all the excess protons and inorganic phosphates at the rate they are being produced, so they start to accumulate rapidly in the muscle cell's sarcoplasm. So first, the accumulation of protons affects muscle cell pH. It decreases the pH and thus acidifies the muscle cell. The proton accumulation therefore causes acute or temporary metabolic acidosis, which in turn causes acute muscular fatigue. We will discuss in a bit how the acute metabolic acidosis actually causes the muscle inhibition. How does it actually inhibit muscle contractions? We will also discuss later that proton accumulation also stimulates pain, which we often refer to as the burning sensation we typically experience during heavy post-anaerobic threshold exercise. And I will also discuss the concept of pain during post-anaerobic threshold exercise and how it is also a means of stress relief. If you think about it carefully, pain is telling you to stop doing what you're doing. So more on that later. Now also, as you see here, is the accumulation of inorganic phosphates, which come from the rapid ATP hydrolysis. This also stimulates fatigue, but not through acute metabolic acidosis. Now in a few moments, we will discuss the mechanisms connecting proton and inorganic phosphate accumulation to fatigue in much more detail. We will also talk about how they directly inhibit muscle contractions. Now, another takeaway from this slide is that lactic acid, or lactate, is not the cause of acute muscular fatigue or even pain. It does not contribute to acute metabolic acidosis. Remember, lactate does the opposite. It tries to buffer the acidosis. As you see here, and as emphasized earlier, the lactate that is produced during post-anaerobic threshold exercise is primarily used as a fuel molecule for future ATP production. It is also used as a substrate for glucose production in the liver. It is not a waste product, nor does it accumulate in the muscle cell and cause acidosis. You may hear people talk about lactic acidosis as the cause of fatigue during heavy exercise, but again, this is not evidence-based information. Lactate is not the bad guy here. So in summary, the stimulus for acute muscular fatigue is 1, proton accumulation and the resulting acute metabolic acidosis, and 2, inorganic phosphate accumulation. And again, please understand that they accumulate in the sarcoplasm because the mitochondria is overwhelmed due to limited oxygen availability and thus cannot clear out the protons and inorganic phosphates at the rate they are being produced by the rapid ATP hydrolysis and redox reactions. So next, let's talk about how the proton accumulation as well as the inorganic phosphate accumulation connects to acute muscular fatigue as well as pain. The question now is how? How does proton accumulation and acute metabolic acidosis as well as inorganic phosphate accumulation cause acute muscular fatigue during post-anaerobic threshold hypoxic situations? In other words, how do they cause muscle inhibition? First, let's discuss proton accumulation and the resulting acute metabolic acidosis. Now, if you remember, I indicated that fatigue is simply a situation in which the muscle inhibits itself as a way to relieve itself from a hypoxic stress. So this means that there is some mechanism associated with hypoxia that interferes with the process of muscle contraction. If muscle contraction processes are interfered with, how would that affect the ATP and thus oxygen demand of the muscle cells? It would clearly decrease the demand for ATP and thus oxygen because the muscle is quote-unquote slowing itself down. This would in turn mitigate hypoxia in the cell. Again, the muscle inhibition that characterizes fatigue is an acute stress response. More specifically, an acute stress relief response. So how does proton accumulation in muscle cells during a hypoxic post-anaerobic threshold state interfere with the processes of muscle contractions? When protons accumulate and your muscle cells undergo acidification, particularly to a pH below 7, several processes that are critical for muscle contractions are inhibited. First, as glycolysis ramps up to help meet the ATP demand during post-anaerobic threshold exercise, as we discussed earlier, the ensuing proton accumulation will in turn slow glycolysis down, again for the sake of slowing down the muscle. How would inhibiting glycolysis in turn inhibit muscle contractions? It would inhibit ATP production, which would in turn inhibit muscle contractions. Remember, glycolysis is one of the important anaerobic processes that backs up the aerobic systems during post-anaerobic threshold exercise to help meet the ATP demand. So in essence, the more glycolysis ramps up, the more it will eventually be inhibited, because when glycolysis ramps up further, that means you are further in the post-anaerobic threshold hypoxic zone. In summary, glycolysis contributes to proton accumulation, and proton accumulation will eventually inhibit glycolysis, thereby inhibiting muscle contractions. So how does proton accumulation cause an inhibition of glycolysis? It slows down glycolysis through the inhibition of the enzyme phosphofructokinase, which is the rate-limiting enzyme of glycolysis. A rate-limiting enzyme is an enzyme that controls how active a metabolic pathway is, such as glycolysis. In other words, when phosphofructokinase is highly active, glycolysis is highly active, and vice versa. So proton accumulation, more directly the resulting acidification, is adverse to phosphofructokinase activity. It suppresses its activity, which in turn will slow glycolysis down. As a result, the rate of ATP production will slow down, which will in turn, quote unquote, slow down muscle contractions, thereby reducing the ATP and oxygen demand of the muscle. This will then translate to a forced decrease in muscular performance, putting you into a pre-anaerobic threshold non-hypoxic workload. Second, proton accumulation and the resulting acute metabolic acidosis can inhibit cross-bridge formation between the thick and thin filament. Remember, cross-bridge formation is a critical process in muscle contractions. Now, this will require a bit of memory of your exercise physiology course, but if you remember, in order for the thick and thin filament to form a cross-bridge connection, the binding sites for myosin to attach to must be exposed on the thin filament. And if you remember, in order for the binding site to be exposed, tropomyosin, as you see here as a long linear protein, must be removed off of these binding sites. Tropomyosin is removed off of the binding sites by another protein called troponin. And troponin knows to remove tropomyosin off of the binding sites when calcium binds to it. So if calcium can't bind to troponin, the tropomyosin can't be removed off of the binding sites, and myosin cannot form a cross-bridge with the thin filament. This would in turn interfere with the sliding filament process and thus muscle contraction. The calcium binding sites on troponin is sensitive to pH, in that when pH decreases, like during post-anaerobic threshold hypoxic situations, the binding potential with calcium is reduced significantly. So in summary, proton accumulation and the resulting acute metabolic acidosis reduces overall muscle contractility by 1, slowing down glycolysis after it ramps up, thereby reducing the rate of ATP production, and 2, by inhibiting cross-bridge formation by reducing the binding of calcium to troponin. Now, how else can proton accumulation in the muscle inhibit muscle contractions? How else can it cause fatigue? Another way is not through a direct inhibition of muscle contraction processes like presented here in these 2 bullets, but also indirectly through the stimulation of pain. We often describe this pain during high-intensity exercise as burning pain. We've all experienced this. Fundamentally speaking, what is pain and what is the purpose of pain? The biological purpose of pain is protection. It is sensory information indicating that there is a stressor present. For example, if you pinch your skin lightly, you sense touch. This is called a mechanical stimulus. But if you pinch harder and harder, this mechanical stimulus increases. Eventually, that pinch will induce pain. This is when the mechanical stimulus exceeds a certain threshold, at which point it becomes a noxious stimulus, Noxious, a noxious stimulus, which means pain stimulus. In other words, the sensation of touch becomes the sensation of pain. The process by which the sensory nervous system, through sensory neurons, like those in your skin, detects a noxious stimulus and induces pain is called nociception, nociception. Nociception is in essence the process of pain. So as you pinch your skin harder and harder and you sense pain, what happens? What does the pain do? Pain causes you to stop pinching your skin. So why do you think you pinching your skin at a certain intensity causes pain? Because the pinching at a certain degree was perceived as a threat to your body, particularly your skin cells. That mechanical stimulus turned into a mechanical stress and thus into a noxious stimulus to tell you that the pinch has reached a level that may damage your skin. The pain caused you to stop pinching your skin, and thus the stress is removed. As you can see, pain is an acute stress response mechanism, more specifically, an acute stress relief mechanism. When you place your hand on the hot stove, there is pain because the thermal stimuli exceeded a certain threshold, making that thermal stimuli a noxious stimuli causing nociception or pain. This causes you to remove your hand from the hot stove immediately, thereby relieving your skin from the stress and protecting your skin from damage. So how does this relate to the pain you feel during heavy exercise? And how is it related to the hypoxic stress that you experience during post-anaerobic threshold exercise? Let's go ahead and answer these questions through this figure. Starting on the left, you have a subject exercising at a post-anaerobic threshold workload. To keep it simple, let's just say the subject is performing an all-out set of weighted back squats. As we discussed earlier, due to the rapid ATP hydrolysis and redox reactions and the limitation of the mitochondria due to the hypoxic state, there is an accumulation of free protons. When the protons accumulate to a certain degree or threshold, they can become a noxious stimulus. Again, a pain stimulus. I'll say that one more time. When the protons accumulate to a certain degree or threshold, they can become a noxious stimulus, which again is a pain stimulus. You see, surrounding your muscles, you have tons of sensory neurons that detect a variety of stimuli. And many of these sensory neurons are what we call nociceptors, which have the ability to detect noxious stimuli and transmit those signals to the CNS. Now, as I said earlier, the proton accumulation can be a noxious stimuli, just like the pinch on the skin. There are specialized receptors on these nociceptors that detect a variety of stimuli, such as mechanical, thermal, and chemical stimuli. One of the chemical or chemoreceptors on these muscle nociceptors is called ASICs, which stands for acid sensing ion channels. ASICs are triggered when protons accumulate beyond a specific level. Protons can bind to the ASICs and cause them to depolarize the nociceptor. The signals are then transmitted towards the central nervous system in the afferent direction. Afferent meaning towards the CNS from the periphery being the muscle. When these signals reach the CNS, they can cause the sensation of pain, or that burning sensation, if you will, in your muscles. So then what does this pain do? It stops you from doing whatever it is that is causing the pain, which in this case would be exercising at that certain intensity, which in this example was that all-out weighted back squat. Pain is a biological stop signal. It is telling you to stop doing what you're doing because it is causing stress to your muscles, which can eventually damage your muscles. Pain is trying to protect your muscles. So what is that stress the pain is protecting the muscle against? It is again hypoxia and also the resulting acute metabolic acidosis. So as you can see, the pain during heavy exercise as well as the fatigue or muscle inhibition work together to relieve your muscles from this hypoxic stress. This is the very same concept as the pain you experience when you pinch your skin hard enough or you accidentally touch a hot stove. Now, also to add, the degree of pain is directly related to the degree of stress. As you know, the pain is more severe if you pinch harder. This is because there's more noxious stimuli. So as exercise intensity increases towards maximum intensity, the noxious stimuli in the form of these protons becomes more severe, and thus the pain becomes more severe. So think of the types of exercises that burns the most. Next time you experience that burn, go over in your head what is happening physiologically as described here in this slide. Now, in a couple slides, I will discuss why you experience this nociception more severely during, say, resistance exercise. Stay tuned. So in summary, pain or nociception due to the proton accumulation becoming a noxious stimuli is part of the acute stress response mechanism to relieve your muscles from that hypoxic stress and the resulting acute metabolic acidosis. And pain and fatigue go hand in hand, meaning they both serve the same purpose during post-anaerobic threshold exercise. It is stress relief. Now, as a side note, remember lactate, or what people refer to as lactic acid, again, not the same thing, is not the cause of this pain because it is not the cause of the mechanisms underlying the pain, which is the proton accumulation and the resulting acidosis. Thus, lactic acidosis or lactic acid buildup is not the cause of that burning sensation during fatiguing exercise. At this point of this lecture series, you should now understand why. Now, other than proton accumulation, I also indicated that the accumulation of inorganic phosphates in the sarcoplasm also contributes to acute muscular fatigue and is a part of this stress response mechanism. However, keep in mind that inorganic phosphate accumulation does not contribute to acute metabolic acidosis, but it contributes to fatigue. So how does inorganic phosphate accumulation cause fatigue? More specifically, how does it inhibit muscle contractions? The effect of inorganic phosphate accumulation on muscle contractions is very direct, meaning it directly interferes with processes critical for muscle contraction. First, as a brief review of exercise physiology, we should know that once a neural signal reaches the muscle fiber from the motor neuron, the muscle fibers depolarize, meaning action potentials are produced. As you see in this figure, the action potential propagates or travels across the muscle fiber and down the transverse tubule, or T-tubules, which is a passageway for the action potential that dips into the muscle fiber. From there, the dehydropyridine receptors are activated upon detecting the action potential. This would essentially open up the bottle cap of the sarcoplasmic reticulum, which houses the intracellular calcium inside muscle cells. This would allow calcium to exit the sarcoplasmic reticulum. As I mentioned earlier, the calcium would then bind to troponin on the thin filament, which would then remove tropomyosin off of the cross-bridge binding sites. This would allow for cross-bridge formation and thus contraction. So when inorganic phosphates accumulate to a certain degree, this entire mechanism is interfered with, particularly in two areas. One, inorganic phosphate, when accumulated, can prevent calcium release by the sarcoplasmic reticulum by precipitating with calcium, forming calcium phosphate. This prevents calcium from exiting the sarcoplasmic reticulum and thus binding to troponin. Second, inorganic phosphates, when accumulated, can block calcium from also binding with troponin. So in summary, inorganic phosphate accumulation interferes with the role of calcium during contraction, thereby inhibiting muscle contraction. So in summary, proton accumulation and inorganic phosphate accumulation both inhibit muscle contractions by interfering with the contraction process. Proton and inorganic phosphate accumulation prevents binding of calcium to troponin and thus inhibits cross-bridge formation. Inorganic phosphate accumulation also inhibits the release of calcium from the sarcoplasmic reticulum. Proton accumulation also stimulates nociception or pain, which can also indirectly inhibit muscle contractions. These are the mechanisms connecting the stress of hypoxia during post-anaerobic threshold exercise to acute muscular fatigue. So this is a very important summary for you to understand before moving forward. Now remember this important fact: fatigue overrides effort, meaning no matter how hard you are trying, fatigue will force your muscles to slow down. And we can see this from a physiological perspective as well. Let's go ahead and look at this figure again for an example. The muscle cells during fatigue are receiving plenty of neurostimuli since neurostimulation of the muscle is voluntary. Since you are putting forth as much effort as possible, the neurostimuli is very high. So even though your muscle cells are neurally stimulated sufficiently, the muscle's force output is decreasing because of fatigue. This is because the mechanisms of fatigue involving proton and inorganic phosphate accumulation occurs after the neural stimulation of the muscle cell. As you see here, neural stimulation is up here. The effects of inorganic phosphate accumulation is down here. And same with the effects of proton accumulation. So no matter how much neural stimulation there is, the machinery for muscle contractions are being inhibited by proton and inorganic phosphate accumulation. This is why no matter how hard your voluntary efforts are, say like during an all-out sprint, fatigue will occur and force you into a lower work output. Remember, fatigue is a forced reduction in muscle contractility, again, due to a hypoxic stress. Now, when I ask students during what type of exercise do you experience the most fatigue and the most pain, most of the time students will say resistance exercise. We generally know that resistance exercise is very fatiguing. When you contract against a heavy resistance, you know that you cannot sustain the same force production from the first rep through multiple repetitions. You know that eventually, rep after rep, the movement will slow down, and eventually you won't be able to move that weight. You also know that one of the reasons why you eventually stop your reps during a set is not just because of fatigue, but also because of that burning pain. So the question is, why is resistance exercise so acutely fatiguing? Why is it that during a set of resistance exercise, you experience the fatigue most severely? By now you understand that proton and inorganic phosphate accumulation are the mechanisms causing fatigue through inhibition of muscle contractions and sometimes pain. We also know that the proton and inorganic phosphate accumulation occurs during a hypoxic state. So this means exercise in which fatigue effects are most severe, like during resistance exercise, the hypoxic stress is most severe. And this is the case for resistance exercise. During high tension and high force exercise, like when you perform a set of dumbbell curls, there is a severe hypoxic stress in the active muscle cells. And so why is the muscle cell under such severe hypoxic stress? Is it because there is a very high oxygen demand in the muscle? Partly yes, but not so much. It is because the oxygen delivery to the muscle cells is impaired when the muscle is undergoing high tension contractions, more specifically, high tension sustained contractions. You see, during high tension contractions like during dumbbell bicep curls, the bicep muscles are in a constant contracted state throughout the entire repetition, unless of course you relax your muscle at the end of each rep, which most people don't. Usually your muscles remain contracted even at the bottom of the rep and at the top of the rep. When muscles contract, there is compression of blood vessels, including arterial blood vessels that deliver oxygen to the muscle cells. Therefore, when there is a constant state of high tension contraction, like during a set of dumbbell bicep curls, the oxygenated blood flow to the bicep muscle cells are being restricted, and thus the oxygen delivery is restricted. And if the oxygen delivery is restricted, then the oxygen availability is reduced. And thus, aerobic ATP production becomes more limited, as we see reflected in the figure to the right compared to the figure on the left. The anaerobic threshold and aerobic capacity are drastically reduced, and thus the biceps during the dumbbell curl set is in a very post-anaerobic threshold and hypoxic state. And thus the bicep muscles experiences inorganic phosphate accumulation as well as proton accumulation and acute metabolic acidosis more severely. This is why as you increase the resistance, there is more fatigue because there's more tension and more restriction of arterial blood flow in addition to the increased oxygen demand. In other words, the more resistance, the more hypoxic stress. An easy experiment to test this effect would be for you to raise your arms up to shoulder level and just hold it there. Your shoulder muscles, like your deltoids and trapezius muscles, are under constant tension to keep that arm up. Eventually, you know it will become harder and harder to keep that arm up. In other words, you will fatigue, and no matter how hard you are trying, the arm is going to lower itself. Also, you would likely experience the burning pain in your shoulder, which we said was an indication that there is a lot of hypoxic stress and acute acidosis in the muscle cells. Eventually, you'll be forced to drop your arms. You see, simply lifting your arm up is not high intensity. It is not a post-anaerobic threshold workload. But when you hold your arm up, that constant tension is restricting flow of oxygenated blood to your shoulder muscles, which are responsible for holding your arm up. So the shoulder muscles in this situation have a reduced anaerobic threshold due to severely limited oxygen availability. Thus, there would be high amounts of proton and inorganic phosphate accumulation in those muscle cells, and thus fatigue would be very severe. Well, we can quantify this effect using a muscle oximeter, which is similar to a pulse oximeter that you can put on your finger to measure oxygen saturation in your blood. A muscle oximeter measures oxygen saturation in muscle blood vessels. If I place this device on your shoulders during this experiment, you will see that the muscle oxygen saturation will decrease, which indicates that the delivery of oxygen is less than the rate of consumption and utilization. In other words, the oxygen runs out. At the point where the oxygen saturation dips too low, this is the point where you will experience the fatigue and possibly pain, because you know now this is when the hypoxic stress occurs. So again, in summary, because of the high tension sustained contractions during resistance exercise, aerobic ATP production in those active muscle cells will be severely restricted. And this is because of restricted arterial blood flow, the flow of oxygenated blood. And this explains why resistance exercise is so acutely fatiguing. Now, up to this point, we have discussed the mechanisms underlying acute muscular fatigue, and we should recognize that these mechanisms occur in the muscle itself. The hypoxia is in the muscle, the proton and inorganic phosphate accumulation is in the muscle, and this inhibition of muscle contraction is in the muscle. And the pain we experience sometimes is initiated in the muscle. This mechanism of fatigue is referred commonly as peripheral fatigue. So acute muscular fatigue is peripheral fatigue. Muscle is a peripheral tissue, meaning it is outside of the central nervous system, or CNS. And so when we describe fatigue as peripheral fatigue, as we have discussed thus far, it is indicating that the mechanisms underlying the fatigue is occurring in the muscle itself. So now the question is fatigue simply regulated only at the peripheral level, meaning only at the muscle? Is there anything outside of the muscle that can fatigue the muscle? Again, fundamentally as a means of protection. Yes, there is. And this is referred to as central fatigue. The term central is in reference to the central nervous system, which is the ultimate command center that controls the peripheral tissues like muscle. We know that in order for you to produce a muscle contraction, the CNS initiates the process by transmitting signals to the muscle you want to contract. So if those signals from the CNS to the muscle are inhibited, then how would that affect muscle contractility? It would suppress it and manifest in fatigue. So how does central fatigue work, and in what circumstances does it come into play? Now, the easiest way to explain central fatigue is by using an example of a resistance exercise bout. During a resistance exercise session, you have multiple sets across multiple exercises per muscle. As we know, during each set, the muscles experience acute muscular fatigue. In other words, as just described earlier, it experiences peripheral fatigue. And again, this is due to the hypoxic stressor and the ensuing acute acidosis, as well as the inorganic phosphate accumulation, resulting in the inhibition of muscle contractions and oftentimes pain. This occurs within each set, and we know fatigue is expressed by reduction in performance from rep to rep. We know that within each set, the efficiency of each repetition decreases one after another. Now, after you perform a set of reps, you rest. And during the rest, the muscle is completely relieved of the hypoxic stress. So you would expect that the next set will be performed to the same level as the previous set. And the third set and the fourth set and so forth would just all be performed the same. But you know that's not the case. You know that one set after another, the performance declines overall. It is common for you to perform less repetitions from one set to the next. In other words, from one set to the next, you fatigue faster during each set. Then you go to the next exercise for that same muscle, and you know you can't perform the reps as efficiently. You may say that the fatigue from the previous exercise is negatively affecting the next exercise. This is a very common circumstance during a resistance exercise bout. So why is this the case? Why does fatigue overall accumulate in the muscle throughout repeated sets of exercise? One of the explanations is central fatigue, and this is largely mediated from feedback from the muscle to the CNS. Now, during each set, there is hypoxia in the cells, and thus there is acute metabolic acidosis. The acidosis eventually makes you stop the set through fatigue. Then you rest and the hypoxia and thus the acidosis will go away. And then you start your next set and the cycle continues. So during each set, during which time your muscle cells are hypoxic and under acidosis, I mentioned that the protons accumulating can trigger sensory neurons. And sometimes when the protons accumulate to a certain degree, pain can be triggered through those sensory neurons. These sensory neurons are referred to as the group 3 and group 4 afferents, which is another way to describe sensory neurons since they transmit signals away from the peripheral tissue and towards the CNS, the afferent direction. Now, these group 3 and 4 afferents are stimulated in the presence of proton accumulation and thus acidosis, regardless of whether there is pain or not, meaning any level of proton accumulation can stimulate these neurons. So as you perform one set after another, these group 3 and 4 sensory neurons are stimulated. And thus, throughout your entire exercise bout, you could say that these sensory neurons are constantly stimulated since each set would cause proton accumulation. When there is perpetual constant stimulation of these group 3 and 4 sensory neurons, there is in turn inhibition at the central nervous system. More specifically, these group 3-4 sensory neurons, when perpetually stimulated, can in turn inhibit motor neurons, which are responsible for carrying signals towards the muscle to contract it. This inhibition occurs before the motor cortex, where the activation of motor neurons are initiated. In other words, group 3 and 4 sensory neurons can inhibit motor neurons upstream of the motor cortex. Thus, when the motor neurons are suppressed, there is less stimulation of the motor neurons. The muscle receives less neural drive or stimulation. In turn, the total number of muscle fibers stimulated and thus performing contractions during the exercise is overall reduced. So in summary, there is less neural or motor drive to the muscle as the muscle experiences repeated exposures to acute metabolic acidosis from one set to another. This is again a protective mechanism. It is a negative feedback loop. The muscle is telling the CNS through these sensory neurons that it is undergoing repeated exposures of hypoxic stress and acidosis. So the CNS in turn then reduces the neural drive to the muscle to reduce muscle contractions and to protect it from this hypoxic stressor. And this is why, as you proceed through your exercise bout, the muscle becomes more and more fatigued after repeated exposures to hypoxia and acute acidosis. So a sample exam question related to central fatigue could be, which of the following explains the overall reduction in muscular force production across an entire bout consisting of repeated sets of post-anaerobic threshold exercise? The answer would be central fatigue. And what explains the reduction in force production across repetitions during a single set of post-anaerobic threshold exercise? It would be peripheral fatigue or acute muscular fatigue. Now, this slide explains the buffering systems that our body utilizes to deal with situations of acute metabolic acidosis, like during post-anaerobic threshold exercise. Buffering systems are utilized to neutralize pH during situations in which protons accumulate. Essentially, they serve to reduce the level of free protons in a solution, whether it is the muscle sarcoplasm or the blood. Of course, there are times when the buffering systems are overwhelmed and we experience fatigue and sometimes pain. This is usually the case during post-anaerobic threshold exercise, especially maximum intensity exercise. Acute muscular fatigue is the last line of defense during a state of acidosis. The buffering systems are the first line of defense. I'll say that again. Acute muscular fatigue is the last line of defense during a state of acidosis. The buffering systems are the first line of defense. When acidosis overwhelms the first line of defense, the muscle will inhibit itself to reduce the acidosis, i.e. fatigue. So what are the first lines of defense? What are the buffering systems your body utilizes during exercise to deal with acidosis? We can categorize the two buffering systems as intramyocellular and extramyocellular. In other words, inside the muscle cell and outside the muscle cell, respectively. The intracellular buffering system is the carnosine buffering system. And the extracellular system is called a bicarbonate buffering system. Let's discuss. First, we have the carnosine buffering system, which I indicated was the intramyocellular buffering system. So this system tackles proton accumulation inside the muscle cell. Now, carnosine is a dipeptide which is comprised of two amino acids, beta-alanine and histidine. Carnosine is quite abundant in skeletal muscle cells because it is a good buffer since it has a similar pKa as the cell's sarcoplasm, which is 6.8, meaning they are weak acids and absorb protons as opposed to releasing them. So when the sarcoplasm pH decreases, carnosine serves to help neutralize the pH by absorbing free protons. Of course, when the proton accumulation exceeds the rate at which carnosine can buffer them, there will be a state of acute metabolic acidosis and fatigue. So there may be a natural question of whether increasing carnosine levels in the muscle cells can improve performance by improving the buffering capacity inside muscle cells. And there is research evidence to suggest that this is possible. Evidence indicates that by increasing carnosine in muscle cells, there can be better defense against acidosis. Well, this is very much observed in single-cell experiments where we can put the cells in an acidic environment and manipulate the carnosine levels in the cell. And we see that when we put more carnosine inside the muscle cells, there is better neutralization of the pH. But how can we do this in muscle cells in an intact organism like the human body? Can we increase muscle carnosine levels? Yes, we can. One way we can do this is nutritionally. We can increase muscle carnosine levels through increased consumption of one of its amino acid constituents. This is beta-alanine. Beta-alanine is the rate-limiting amino acid for carnosine synthesis, meaning carnosine production is regulated by the availability of intramyocellular beta-alanine. This is not the case with histidine, meaning more histidine in muscle cells do not affect carnosine synthesis. So this gave rise to the idea of supplementing the diet with beta-alanine. So we see tons of different supplement products out there, especially sports supplements with beta-alanine in it. The research shows that with beta-alanine supplementation, there's increased muscle carnosine levels. But does this translate to improved performance and less fatigue? This is where the evidence is mixed. But when we look at the overall body of evidence on the effects of beta-alanine supplementation, it appears that the effects are more substantial during exercise intensities right around your anaerobic threshold, meaning it can help minimize fatigue at your anaerobic threshold. So this means you may be able to improve your steady state muscular power output, whether it's running speed or cycling speed and so forth. So this means you may be able to improve your steady state muscular power output, meaning perhaps you can run a little bit faster at steady state. Beta-alanine supplementation, on the other hand, does not appear to be beneficial during intensities significantly above your anaerobic threshold, like during all-out sprint efforts or high-intensity resistance training. This is because the metabolic acidosis during those exercise situations will always exceed the buffering capacity of the carnosine system, regardless of whether you have slightly more carnosine in the muscle cells due to beta-alanine supplementation. Now, the issue here is that the individuals who supplement beta-alanine the most are those who lift weights and perform high to max intensity intermittent exercise. Again, beta-alanine supplementation may improve muscle carnosine levels, but this will not translate to improved performance and less fatigue during exercise significantly above the anaerobic threshold. So during what situations would beta-alanine supplementation be most beneficial? During steady state exercise that is right at your anaerobic threshold intensity. So if your anaerobic threshold is at a running speed of 6 miles per hour, and so your steady state running speed is at 5.5 miles per hour, increasing muscle carnosine levels through beta-alanine supplementation may allow you to run at 6 miles per hour without fatigue. And again, that is simply because your intracellular buffering capacity has improved. So with that said, what population of athletes would beta-alanine supplementation be most beneficial for? Endurance athletes who need to do as much muscular work across a prolonged period without fatigue. Therefore, if you have friends who consume beta-alanine but only do maximum intensity exercise like resistance exercise, CrossFit, high intensity interval training, sprint interval training, it would most likely not provide any benefit since this type of exercise will always result in acute acidosis that will always exceed the buffering capacity of the carnosine system, regardless of whether there is increased levels of carnosine inside your muscle cells. Now, later on, we will also discuss that carnosine levels in muscle cells naturally increase in response to training, especially training that stresses the carnosine buffering system, meaning this is an adaptive response to exercise that is above the anaerobic threshold. Now, in part 2 of this lecture series, we will discuss this a lot further. Okay, so now what about the extracellular buffering system? This buffering system is described as systemic, meaning it is the buffering system for your entire body. And it is mainly there to neutralize blood pH, since blood affects all cells in the body. As I indicated earlier, your extramyocellular buffering system is called the bicarbonate system, and it is regulated through sensory receptors and breathing. We know as we increase intensity, our breathing rate increases. In other words, ventilation increases. This is primarily due to the need to consume more oxygen. However, at a certain higher intensity, our ventilation increases exponentially, meaning we begin to hyperventilate. The point at which you begin to hyperventilate is your anaerobic threshold, and this point is also referred to as the ventilatory threshold. So anaerobic threshold and ventilatory threshold go hand in hand. So now we know that this increased hyperventilation response is related to the anaerobic threshold. At the anaerobic threshold, we know that protons are going to accumulate and create an acute state of metabolic acidosis. This will also negatively affect the pH of blood because there will be an increase in free protons released to the blood. So this decrease in blood pH will be detected by receptors called chemoreceptors. And this in turn will tell the CNS to increase breathing rate or ventilation. So why does an increase in protons in the blood cause this reflexive increase in ventilation? It is because by increasing ventilation, there is increased exhaustion of carbon dioxide. So the hyperventilation observed at the anaerobic threshold is not because of an exponentially increased need for oxygen consumption, but rather an exponentially increased need to get rid of carbon dioxide from your blood. So how does this affect blood pH and counter acidosis? To explain this, let's turn to this figure here. Here in the middle, we have carbonic acid. And on both sides of carbonic acid, we have reactions. On this side, we have carbon dioxide plus water. And on the other side, we have a proton and bicarbonate. Now, the basics of blood chemistry is that the body strives to maintain chemical equilibrium in the blood. In other words, the body strives to keep both sides of carbonic acid at equilibrium. This reaction and this reaction at balance. It wants to make sure all of this is balanced out. However, during post-anaerobic threshold exercise, the muscle is releasing more free protons into the blood. So now the right side of the formula is increasing, particularly here. And in so doing, chemical equilibrium is disrupted. So how does your body reestablish equilibrium? Like I said, the increase in free protons will decrease pH will be detected by chemoreceptors, and this will cause hyperventilation. Through hyperventilation, you are exhausting more CO2. In other words, hyperventilation will decrease CO2 in the blood. So in accordance to this figure, it will decrease the left side. To create equilibrium here, the reaction shown here will be pushed to the left to bring CO2 back up. And in so doing, the protons will decrease. The protons will be absorbed by bicarbonate to form carbonic acid, and the carbonic acid will be used to form CO2 and H2O. In so doing, this will raise CO2 back up to reestablish chemical equilibrium. So hyperventilation during post-anaerobic threshold exercise is to exhaust more CO2 to reduce CO2 in the blood, to force this reaction from the right to the left so that the protons can be absorbed by bicarbonate. And this is how increasing levels of proton and reduced pH is buffered systemically. Since proton accumulation triggers this response, we can use ventilation as an indicator of the anaerobic threshold. As I mentioned earlier, the hyperventilation response occurs when pH decreases, which occurs at your anaerobic threshold. So the point at which you begin to hyperventilate is the point at which the anaerobic threshold is observed. This is why we use ventilatory threshold as a way to measure one's anaerobic threshold. They go hand in hand. So if I ask the question, which way does this reaction go during hyperventilation at one's anaerobic threshold? It would be to the left. And if I ask, which of the following explains why we have exponential increase in ventilation at the anaerobic threshold? It is because we want to exhaust more CO2 to reduce CO2 in the blood, not because we need to consume more oxygen. This now concludes part 1 of this lecture series focused on acute muscular fatigue. Now that we have a deeper understanding of acute muscular fatigue and the whys and hows, we can now have an advanced understanding of how to reduce fatigue. More specifically, how to perform greater amount of muscular work without fatigue. We understand that the fundamental aspect of reducing fatigue at higher workloads is to increase the power of the aerobic system so that the aerobic system can meet the ATP demand to avoid the energy void and hypoxia. It's all about the aerobic system. As I highly emphasized earlier in this lecture series, anaerobic fitness is not a thing. It does not make any sense. Why would you want to be more reliant on your anaerobic system? This just means that your aerobic system's ATP producing power is very low. So ultimately, conditioning is largely predicated on improving the aerobic system, mainly through better delivery of oxygen and greater aerobic power plants, the mitochondria. So in part 2, we will discuss the fundamental science behind conditioning programs and address the question of how do we become less fatigued at greater muscular workloads. Again, to reiterate, the reason why it is so important for you to understand acute muscular fatigue because it is the most limiting factor to human performance and general physical functioning. Experiencing fatigue doing simple tasks like walking can reduce the overall quality of life. Experiencing fatigue at a running speed of 6 miles per hour while your opponent experiences the same fatigue at running speeds of 8 miles per hour is going to limit your performance in sports. Fatigue will be relevant to whatever fields you go into, whether it is physical therapy, occupational therapy, strength and conditioning, fitness, and so forth. So now that you have an advanced understanding of fatigue, you now have a strong foundation to learn about conditioning and the fundamental basis of any effective conditioning program. Now that you have an advanced understanding of acute muscular fatigue, you should have a good understanding of its implications to human performance. To reiterate, acute muscular fatigue is the most limiting factor to performance or general physical functioning. For example, if you experience acute muscular fatigue at relatively low workloads, like say walking 2 miles per hour down the street, this could certainly affect one's overall function and quality of daily living. The implications of acute muscular fatigue are obviously much clearer in sporting situations. The athlete who is able to do more work with less fatigue is usually the athlete that performs the best. You can be the fastest sprinter or highest jumper, but acute muscular fatigue will negate these abilities. These are the reasons why acute muscular fatigue is an important target of intervention for exercise training programs. Specific training programs that are intended to improve the level of muscular work with less acute fatigue are referred to simply as conditioning or endurance training. Some other terms that have been used to describe this type of training is metabolic training. So any of those terms are really referring to the same type of training that is intended to improve your ability to perform at greater workloads without or with less acute muscular fatigue. So in part 2 of this lecture series, we will take concepts from part 1 regarding the science of acute muscular fatigue and the variables involved, such as anaerobic threshold and aerobic capacity, and apply it to training. In other words, we will answer the question of how we can improve the level of muscular work with less fatigue and what are the underlying mechanisms. We will also highlight the fundamental factor underlying training adaptations, meaning what is required to stimulate changes in the muscle as well as the body in general for you to be able to eventually do more muscular work with less or no fatigue. So first, as always, let's go ahead and make sure we are clear on some key terminologies. So as I said earlier, conditioning, aka endurance training, aka metabolic training, is a long-term exercise training program that is specifically designed to improve muscular endurance. So what is muscular endurance? Well, many believe muscular endurance is simply how long we can exercise for, or how long or far we can run, for example. This is not muscular endurance. Muscular endurance is not about simply how far you can run, for example. It's about the running speed or muscular work you can achieve and sustain. The operative word there being sustain. Let's put this into a race perspective. Is performance in a race about how far you can run, or is it about how much speed you can produce and sustain without fatigue? The latter is what determines performance, and the latter concept is what is most practically significant. There's very little practical incidences where one is required to simply do some level of muscular work for as long as possible. What's more practically relevant is the ability to sustain greater muscular work, like running faster for a longer period. That is muscular endurance. So what is the limiting factor for you to be able to sustain as much muscular work as possible? That limiting factor is acute muscular fatigue. Acute muscular fatigue, as detailed in part 1, is going to be what determines the amount of muscular work one can achieve and sustain. Sustain is, again, the important term here. So for instance, say you had two athletes running the 800 meter, and you instructed them to achieve a workload of 12 miles per hour. One athlete is able to achieve a 12 mile per hour running speed and sustain it for a majority of the 800 meter run. The other athlete is able to achieve 12 mile per hour running speeds, but can only sustain it for, say, 200 meters due to acute muscular fatigue. Who will win the race? Who performed better? The first athlete, right? The first athlete has greater muscular endurance. That first athlete was able to produce a workload of 12 miles per hour in running speed and sustain it for the majority of the 800 meter run, while the other athlete was only able to achieve that same workload for only 200 meters because of acute muscular fatigue, muscle inhibition in response to a stressor. And so because muscular endurance is predicated on the ability to achieve and sustain greater muscular work because of less fatigue, muscular endurance can be simply described as fatigue resistance, in that the greater the muscular endurance, the greater resistance to fatigue. So to reiterate, fatigue resistance, and thus muscular endurance, is the ability to achieve and sustain greater muscular work. And this is both continuously, like during a race, or intermittently, like during a soccer match or basketball game. Now, as it relates to sporting scenarios, here are some examples of what it means to improve muscular endurance and thus fatigue resistance. Being able to run at a faster pace during a race. Being able to hike at a faster pace and finishing the trail earlier. Being able to move faster throughout a soccer match and not be overly limited by fatigue. Completing high loads with greater volume during a CrossFit workout of the day. So before delving into the science of conditioning, let's recap the mechanisms underlying acute muscular fatigue. By now, you should be familiar with this figure and what it represents. This figure displays the integration of aerobic and anaerobic metabolism in meeting the rate of ATP demand during exercise from light to maximum intensity exercise. Now, we can categorize exercise workloads based on whether they are pre-anaerobic threshold or post-anaerobic threshold. Pre-anaerobic threshold exercise workloads are workloads in which the rate of ATP demand can be adequately fulfilled aerobically because there is adequate oxygen delivery and availability for that to occur. Post-anaerobic threshold exercise workloads are workloads in which the rate of ATP demand cannot be adequately or fully satisfied aerobically because the oxygen required to do so is not sufficiently available, and thus a non-oxygen requiring ATP producing system called the anaerobic system works to fulfill only the portion of the ATP demand that the aerobic system cannot. And remember, this portion we refer to as the energy void. So with that said, we can clearly see that the anaerobic systems are never the sole contributor to the ATP needs. The aerobic system never shuts off. It is always contributing to the ATP needs, and its activity increases with increasing intensity and ATP demand. So the anaerobic threshold, as indicated here, is simply the exercise workload or intensity at which the aerobic system cannot fully meet the ATP demand of the active muscles, and anaerobic systems kick in to help out. As the workload increases further past the anaerobic threshold intensity, the aerobic ATP production continues to rise, but there is a greater reliance on your anaerobic systems to help meet the rising ATP needs. The aerobic capacity, as indicated here, is the ATP producing capacity of the aerobic system, meaning it describes the maximum capability of the aerobic system. And as I mentioned in part 1, the variable that is used to quantify or measure aerobic capacity is maximum oxygen consumption rate, or VO2 max. And this should make sense to you all by now, since we know that the ability for the aerobic system to produce ATP is completely dependent on the rate oxygen is consumed and available. So the maximum oxygen consumption rate accordingly reflects one's aerobic capacity. I will reiterate this later on. So let's go ahead and continue this refresher and discuss acute muscular fatigue in relation to this figure here. First, acute muscular fatigue occurs within this zone of workloads or intensity, again described as post-anaerobic threshold workloads or intensity. Oftentimes, the post-anaerobic threshold workload is described as quote-unquote anaerobic exercise. But as you can see, this is a misnomer, since during post-anaerobic threshold workloads, the aerobic system is still the primary ATP producing system, and the anaerobic system is simply filling a portion of the ATP needs that the aerobic system cannot. It is simply a backup support system. So I don't like to describe exercise as either aerobic or anaerobic. Exercise, no matter whether you are walking or sprinting with all-out effort, is always aerobic. So because the term anaerobic exercise is inaccurate, I like to use the term post-anaerobic threshold exercise or pre-anaerobic threshold exercise. So post-anaerobic threshold intensities or workloads are fatiguing and can be sustained only for a brief period of time. Why? Because of acute muscular fatigue, which again is the involuntary inhibition of muscle contractions in response to a hypoxic stress. And as we discussed in part 1, that hypoxic stress results in acute metabolic acidosis and of course the inorganic phosphate accumulation, which disrupts the process of muscle contractions. Remember, fatigue is simply a negative feedback system that forces your muscles from being in a post-anaerobic threshold hypoxic state to a pre-anaerobic threshold non-hypoxic state. Essentially, fatigue forces your muscles to demand less ATP and thus less oxygen by quote-unquote slowing it down, by pumping its own brakes. So fatigue is simply an acute or immediate stress relief system. That stress, again, being hypoxia, which is what characterizes the post-anaerobic threshold condition. So post-anaerobic threshold exercise and hypoxia are synonymous. So remember, post-anaerobic threshold is characterized by the oxygen consumption and availability not being able to meet the oxygen required for the aerobic system to fully meet the ATP demand. This is a hypoxic situation. All right, so once your workload or intensity exceeds the anaerobic threshold, fatigue starts to set in because there is the presence of hypoxia in the active muscle cells. Now, as you increase the workload or intensity further past the anaerobic threshold and towards maximum intensity, the hypoxia becomes more and more severe, and thus the fatigue becomes more and more severe. And thus the exercise workload or intensity becomes more and more difficult to sustain. And we know this because we generally understand that an all-out sprinting speed is less sustainable because it is much more fatiguing than running slightly slower. Finally, let's focus on the third box in the bottom right-hand corner, which will segue into our discussion on the science of conditioning. Now let's think about the fundamentals of training adaptations. Let's say that a workload of a 10 mile per hour running speed is a post-anaerobic threshold workload and can be placed, say, like right here on this curve. It's not maximum intensity, but it is post-anaerobic threshold, and therefore it is fatiguing because there's obviously a hypoxic stress in the muscles. So what needs to happen in order for you to be able to exercise at this 10-mile-per-hour workload without fatigue? Obviously, you need to train, but what needs to happen physiologically? Well, if hypoxia is what's causing the fatigue at this 10-mile-per-hour workload, changes need to occur so that the 10-mile-per-hour workload goes from being a post-anaerobic threshold hypoxic workload to a pre-anaerobic threshold non-hypoxic workload. So in reference to this figure, what needs to change? The white area under the curve needs to increase. More specifically, the anaerobic threshold point needs to shift to at least a 10-mile-per-hour workload so that the 10-mile-per-hour workload is non-hypoxic and non-fatiguing. Now in the next slide, we will discuss this further, but for now, we have to understand that the fundamental aspect of training is that adaptations in the body or parts of the body like muscle occurs in response to a constant or frequent stressor. If there is no stressor present, there is no adaptation and therefore no positive changes. So training without sufficient stress is not training at all, since no changes will occur. There will be no reason for the body to undergo adaptations and changes because there will be nothing to adapt to. When there is a constant, frequent stressor, the body or parts of the body that are undergoing that stress will adapt to the stress, so the stress becomes no longer stressful eventually. In so doing, the body or parts of the body that has undergone adaptation is quote-unquote enhanced. So adaptation can be viewed as the long-term stress relief system. So in order to reduce the stress of this 10-mile-per-hour run and thus the fatigue associated with the 10-mile-per-hour run, what should be the fundamental basis for the training program? The training program should frequently impose a hypoxic stress to the muscle. So when this hypoxic or metabolic stressor is imposed on the muscle consistently, the muscle will eventually adapt to it. This is a form of a survival mechanism. The muscle recognizes that this hypoxic stress is frequent and is a threat to the muscle. So it undergoes processes to adapt to the stress, thereby making it more, quote-unquote, enhanced. It will adapt to the frequent hypoxic stress, so eventually that 10-mile-per-hour workload is no longer associated with the hypoxic stress. You have undergone adaptations to the hypoxic stress associated with that 10-mile-per-hour run, and thus enhancements, so that the 10-mile-per-hour workload is quote-unquote easier, and you are able to sustain it because there is no hypoxia to induce fatigue. What this means is that training should be at or above the anaerobic threshold in order to impose appropriate metabolic/hypoxic stress on the muscles so that the muscles have a reason to adapt and to move this white curve and thus the anaerobic threshold and aerobic capacity upwards. So let's take a look at how these curves change over time with training in that post-anaerobic threshold zone and what type of adaptations are taking place. So as you engage in a conditioning program consisting of repeated bouts of post-anaerobic threshold exercise and your muscles undergo frequent hypoxic stress, adaptations to this stress will occur. So what type of adaptations will take place in response to this frequent stressor? The adaptations that take place in response to post-anaerobic threshold type training are aerobic adaptations, not anaerobic adaptations, because that would make no sense, as you all should know by now. There is only aerobic fitness. There's no such thing as anaerobic fitness. You are trying to become less reliant on anaerobic systems at higher workloads, and you want the aerobic systems to become more reliable, or in other words, more quote-unquote powerful, to better meet the ATP needs of higher workloads. In summary, the result of training will be reflected by the changes you see here in this upper right figure compared to this baseline figure. So this is pre-training and this is post-training. As you can see, the white area under the curve, which represents aerobic ATP production, is much larger compared to baseline. You see two variables of aerobic ATP production improving with training. You see an improvement in anaerobic threshold as well as aerobic capacity. Now let me put this into the context of that 10-mile-per-hour workload example. Say at pre-training, that 10-mile-per-hour workload was a post-anaerobic threshold workload and can be placed right about here on this curve. At pre-training, that 10 mile per hour workload induced a hypoxic stress to the muscle and fatigue occurred. You were not able to sustain that 10 mile per hour run for very long. Now with training, you experience these changes reflected by the figure here on the right, where the anaerobic threshold and aerobic capacity increases. And that 10 mile per hour workload is placed on the same area of this curve as it was on the other curve for the baseline. You see now, because the aerobic system has improved, the 10 mile per hour workload is now a pre-anaerobic threshold workload. This means there is no more hypoxic stress and no more fatigue. This means that you are now able to sustain a 10 mile per hour workload without fatigue. So in summary, the improvement of muscular endurance and thus fatigue resistance as defined earlier is a result of aerobic adaptations, which is reflected by two measures: improved aerobic capacity and improved anaerobic threshold. An improvement in aerobic capacity, as you see here, can be quantified by an improvement of VO2 max. Again, maximum oxygen consumption rate. This means that if you experience an increase in VO2 max during a VO2 max test taken before and after months of training, that would reflect an improvement in aerobic capacity. Next, an improvement in anaerobic threshold can be quantified by an improvement in lactate or ventilatory threshold. Remember, the anaerobic threshold is when lactate production increases due to increased fast glycolysis, and ventilation rate increases due to metabolic acidosis. The lactate threshold is the workload or intensity where lactate levels in the blood increase exponentially, and ventilatory threshold is the workload or intensity where ventilation rate increases exponentially. These are reflective of the anaerobic threshold and can be measured to quantify the anaerobic threshold. So say if your lactate or ventilatory threshold occurred at 7 miles per hour pre-training, and after months of training, your lactate or ventilatory threshold now occurs at 11 miles per hour, that means aerobic adaptations have occurred. That means your anaerobic threshold has improved. So altogether, an improvement in aerobic capacity and anaerobic threshold are what results from the aerobic adaptations to post-anaerobic threshold training or conditioning. Now, as a side note, improving anaerobic threshold is more significant for well-trained individuals, as well-trained individuals do not experience significant improvements in aerobic capacity or VO2 max because they have likely already achieved the highest potential aerobic capacity possible. So usually well-trained individuals focus more on anaerobic threshold and get tested for lactate or ventilatory thresholds as opposed to VO2 max. Now, collectively, the anaerobic threshold and aerobic capacity is referred to as aerobic power. So the factors constituting aerobic power are anaerobic threshold and aerobic capacity. So the goal of conditioning programs is to improve aerobic power, which can be simply described also as an improvement in aerobic fitness. So if I ask the question, an improvement in aerobic power following training is reflected by which of the following? It would be an improvement in anaerobic threshold and aerobic capacity, the two factors that constitute aerobic power. Now, other potential answer choices could be an improvement in VO2 max and lactate or ventilatory threshold, which I said were measures of aerobic capacity and anaerobic threshold, respectively. Now, later on in this lecture series, we will discuss what these aerobic adaptations are that contribute to this increase in aerobic power. I'm sure you can already guess that the adaptations involve greater oxygen supply and improved mitochondrial power or function. So more on this later. For now, just understand that the aerobic adaptations that improve aerobic power are multifactorial and involves multiple organ systems, meaning it's not just about changes in the muscle, but in other organ systems as well. So what about the other side of the coin? What happens when you get out of shape, when you detrain or become sedentary? The figure here down to the left represents those changes compared to baseline. As you can see, aerobic power decreases, and that would be reflected by a decrease in anaerobic threshold as well as aerobic power. So let's again use that 10 mile per hour workload again. So let's say at baseline, a 10 mile per hour running workload was placed right here on this curve, right above the anaerobic threshold. And therefore, the 10 mile per hour run was slightly fatiguing. But you decided to just become completely sedentary. Now, because there's absolutely no metabolic hypoxic stressor imposed on the muscle may undergo deadaptation, which again is reflected by a decrease in aerobic power. So now let's place that 10 mile per hour workload on the same area on this curve. You just put it down here. Now you can see that because the aerobic power decreased, that 10 mile per hour run is now way above the anaerobic threshold and is even more fatiguing because there is more hypoxic stress. In simple terms, you have become out of shape. You become less aerobically fit. Okay, so in summary, training with post-anaerobic threshold intensities and frequently imposing that hypoxic stress to the muscle will result in aerobic adaptations, which will increase aerobic power, which is reflected by an increase in anaerobic threshold and aerobic capacity. Therefore, workloads that used to be a post-anaerobic threshold workload and thus fatiguing may now be a pre-anaerobic threshold workload and non-fatiguing. Again, even if you are doing sprint interval training, no matter how many people describe it as anaerobic training, there is no such thing as anaerobic fitness. So later on, we will discuss this myth a bit further and discuss how even sprint interval training can be a great mode of training to improve aerobic power. Now, to better understand the practical implications of improved aerobic power, I want to present some experimental data here that I collected on elite marathoners and also recreationally active individuals, the quote-unquote average Joe, if you will. Now, the test we had both of these groups undergo was a 13.1 mile per hour run to failure treadmill test, and this test involved just what the description implies. We had subjects in both groups run at a constant speed of 13.1 miles per hour on a treadmill, and we simply tested how long they could sustain that workload for. A few days before this test, we also tested the subject's anaerobic threshold as well as VO2 max. The anaerobic threshold was measured via ventilatory threshold testing, and VO2 max was tested to quantify the aerobic capacity. So why 13.1 miles per hour? Well, this is an important test for marathoners because a 13.1 miles per hour average run pace equates to a sub 2-hour race time, which is the ultimate goal for competitive marathoners. So we wanted to see how long the elite marathoners can sustain a 13.1 mile per hour workload and compare that to the average Joe. Now, I'm sure you can imagine how fast a 13.1 mile per hour run is on a treadmill. It's pretty fast. To some of us, 13.1 miles per hour is an all-out sprint speed. So let's take a look at the results. So first, let's look at how long the subjects that were average Joes could sustain this 13.1 mile per hour run. So subject 1 was able to sustain that 13.1 mile per hour run for 1 minute and 30 seconds. Subject 2, 54 seconds. Subject 3, 1 minute and 12 seconds. Subject 4, only 25 seconds. Subject 5, 1 minute and 20 seconds. And on average, the average Joes was able to sustain that 13.1 mile per hour run for only 1 minute and 4 seconds. Now let's take a look at the elite marathoners and see how long each subject was able to sustain a 13.1 mile per hour running workload. Subject 1 was able to sustain that 13.1 mile per hour running workload for 1 hour, 57 minutes, and 32 seconds. Subject 2, 1 hour, 42 minutes, and 10 seconds. Subject 3, 2 hours, 2 minutes, and 14 seconds. Subject 4, 1 hour, 58 minutes, and 16 seconds. Subject 5, 2 hours, 1 minute, and 18 seconds for an average of 1 hour, 56 minutes, and 18 seconds. So just imagine that, running 13.1 miles per hour on a treadmill and being able to do that for almost 2 hours straight. So as you can clearly see, the elite marathoners did not experience acute muscular fatigue running at 13.1 miles per hour, while the average Joes clearly did. The only reason these marathoners stopped was not because of fatigue after 2 hours of running, but because of exhaustion, which we defined in part 1 as simply running out of fuel. More specifically, their muscle glycogen was completely depleted. Now, remember, muscular endurance is about not only achieving a higher workload, but sustaining that workload. So, yes, the average Joe can achieve 13.1 miles per hour, but can they sustain it? No, they cannot. This data also demonstrates that simply achieving 13.1 miles per hour is possible for many. But sustaining 13.1 miles per hour without fatigue is a whole nother story, right? Achieving and sustaining such a high workload is exemplary of muscular endurance. So the important question is why? Why is the elite marathoner able to maintain a 13.1 miles per hour workload for a significantly longer time than the average Joe? The obvious answer is that the average Joe during the 13.1 miles per hour run experienced acute muscular fatigue, while the elite marathoner did not. But what explains this from a physiological perspective? Let's take a look at the measures of aerobic power from the other exercise lab tests and compare between groups. First, the average Joe. Let's take a look at their VO2 max, which again is maximum oxygen consumption rate and is a quantification of aerobic capacity, which is the maximum ATP producing capacity of the aerobic system. So for the average Joes, subject 1 had a VO2 max of 60 milliliters per kilogram of body weight per minute. Subject 2 had a VO2 max of 54. Subject 3 with the VO2 max of 58. Subject 4 with the VO2 max of 42. And 58 for subject 5, with the average VO2 max for the average Joe group being 54 milliliters per kilogram of body weight per minute. Now, in all honesty, compared to norm values, these numbers are not terrible. They are somewhat average for the general population within their age range. Now, here's what's important. Let's look at the intensity at which the anaerobic threshold is observed. The intensity is reflected as a percent of VO2 max. So this is relative intensity, intensity relative to the VO2 max. So for subject 1, their anaerobic threshold was at a relative intensity of 60% VO2 max, 58% VO2 max for subject 2, 62% of VO2 max for subject 3, 50% of VO2 max for subject 4, 58% VO2 max for subject 5, and the average anaerobic threshold for the average Joes was 58% VO2 max intensity. Now, during the 13.1 miles per hour run, we hooked up the subjects to a metabolic cart and we measured their oxygen consumption rate, their VO2. And we calculated the intensity of that 13.1 mile per hour run relative to their VO2 max, so as a percentage of their VO2 max. So for subject 1, the 13.1 mile per hour run was a relative intensity of 80% of their VO2 max. So was that 13.1 mile per hour run for subject 1 a workload above or below their anaerobic threshold? Well, that 13.1 mile per hour run for subject 1 was an intensity of 80% VO2 max, and their anaerobic threshold was at an intensity of 60% VO2 max. So yes, that 13.1 mile per hour run was significantly above subject 1's anaerobic threshold. So this means subject 1 was under a hypoxic stress and the active muscles were undergoing acute metabolic acidosis from proton accumulation as well as inorganic phosphate accumulation. Thus, muscle contractions were self-inhibited during this 13.1 mile per hour run. In other words, the hypoxic metabolic stress of the 13.1 mile per hour workload induced fatigue to slow the muscles down to reduce the ATP and oxygen demand of the muscle to get down to a pre-anaerobic threshold workload. And this all occurred within 1 minute and 30 seconds. Fatigue forced the subject to stop at 1 minute and 30 seconds. So in summary, subject 1 was only able to sustain the 13.1 mile per hour workload for 1 minute and 30 seconds because that workload was at a relative intensity above their anaerobic threshold. And you should be confident in understanding that the fatigue subject 1 experienced was in response to that hypoxic stress. Now let's look at subject number 2. Same story as subject 1. The 13.1 mile per hour run workload was at a relative intensity of 82% of their VO2 max, which is higher than the intensity at which their anaerobic threshold was observed, which was 58% of their VO2 max. This means subject 2 was way above their anaerobic threshold during that 13.1 mile per hour run. Thus, conceivably, subject 2 may have experienced greater hypoxic stress and thus fatigue compared to subject 1, and that is reflected in subject 2's performance. Subject 2 was only able to sustain that run for 54 seconds. Now, if you look at subjects 3, 4, and 5, we have the same situation. If you look at subject 4 in particular, you can see that he performed the worst out of the group. He could only sustain that 13.1 mile per hour run workload for only 25 seconds. Now let's look at the relative intensity of that 13.1 mile per hour run for this subject. It was 90% of his VO2 max, which means that the 13.1 mile per hour workload is near maximum intensity for this subject. And what was the intensity of his anaerobic threshold? It was pretty low at 50% of his VO2 max. So subject 4 was way above his anaerobic threshold during that 13.1 mile per hour test. And thus the hypoxic stress was comparatively more severe in subject 4, and thus fatigue was comparatively more severe in subject 4 compared to the others. And this was reflected by a very poor performance. Okay, now let's take a look at the elite marathoners. We obviously know that they were able to sustain that 13.1 mile per hour run for significantly longer, for almost 2 hours. And I think you all can guess by now that the 13.1 mile per hour workload was at an intensity below their anaerobic threshold. So let's take a look at their testing data. Of course, as you can expect, their VO2 max, which again is a measure of aerobic capacity, was significantly greater than the average Joe's. On average, the VO2 max for the elite marathoner group was 81 milliliters per kilogram of body weight per minute, which is again significantly greater than the 54 average for the average Joe's. But as we know, VO2 max is only one factor of aerobic power, meaning it's not the only important factor for endurance performance. Let's take a look at the elite marathoners' anaerobic threshold, again as reflected by a percentage of their VO2 max. Subject 1 had an anaerobic threshold intensity of 78% of their VO2 max. Subject 2 had an anaerobic threshold of 80% of their VO2 max. Subject 3, 79% VO2 max. Subject 4, 87% of their VO2 max. Subject 5, 84% of their VO2 max. And on average, for the elite marathoners, anaerobic threshold was 82% of VO2 max. You can clearly see that the elite marathoners had a significantly greater anaerobic threshold intensity compared to the average Joe. You could say that since VO2 max, aerobic capacity, as well as anaerobic threshold, was significantly greater in the elite marathoners versus average Joes, you can say that the elite marathoners altogether had greater aerobic power. But what's more important here is the relative intensity of that 13.1 mile per hour run for the elite marathoners. So let's first take a look at subject number 1. The 13.1 mile per hour run was a relative intensity of 75% VO2 max. So was this intensity post or pre-anaerobic threshold? It was pre-anaerobic threshold. The intensity of the 13.1 mile per hour run was lower than the intensity at which the anaerobic threshold was observed. Therefore, subject 1 did not experience hypoxic stress in their muscles during that 13.1 mile per hour run. Therefore, subject 1 did not experience acute metabolic acidosis or inorganic phosphate accumulation, and thus there was no stimulus or reason for fatigue, again, because there was no stress. If you look at subjects 2, 3, 4, and 5, we have the same exact situation. The intensity of their 13.1 mile per hour run was slightly less than their individual anaerobic threshold intensity. Subject 2, that 13.1 mile per hour run was a relative intensity of 78%. Their anaerobic threshold intensity was 80%. So at 13.1 miles per hour, they were slightly below their anaerobic threshold. Same thing with subject 3, 4, and 5. And on average, the 13.1 mile per hour run for the elite marathoners was a relative intensity of 78% VO2 max. And the average anaerobic threshold intensity was 82% VO2 max. So that 13.1 mile per hour run was a pre anaerobic threshold non-hypoxic exercise workload or intensity. And again, this is why the elite marathoners were able to sustain that running workload for almost 2 hours straight. They did not experience acute muscular fatigue. So from this data, you can clearly see the importance of aerobic power in endurance performance, which again is being able to achieve and sustain greater muscular work. And just as a reminder, aerobic power is a function of both VO2 max and anaerobic threshold. Now, as I alluded to earlier for the elite marathoner, VO2 max at this level of competition and training is not the most important factor, because for these individuals, due to their extensive training history, they have achieved the highest possible VO2 max that they can ever achieve, meaning they've achieved the highest possible aerobic capacity that they can ever achieve. And this is genetically determined. As you can see, their VO2 max scores are very high compared to norm values. So for very well-trained individuals such as these elite marathoners, the goal outcome of their training is yes, to improve aerobic power, but more specifically through anaerobic threshold improvements. They want to get that anaerobic threshold intensity as close to 100% VO2 max as possible. Now keep in mind, there's never a case where anaerobic threshold intensity is at VO2 max. I believe the record for anaerobic threshold was somewhere around 90% VO2 max. 82% VO2 max is pretty high, and this is why they are elite marathoners. So to put it all into a visual perspective, let's compare the aerobic anaerobic curves of the average Joes versus the elite marathoners. So on the x-axis, you have running speed as a measure of exercise workload, and that 13.1 mile per hour run is placed here on the x-axis on both figures, exactly in the same spot. So as you can see with the average Joe, their aerobic power is significantly lower than the elite marathoners. More importantly, if you look at the 13.1 mile per hour workload for the average Joe, you can clearly see that it is at an intensity above their anaerobic threshold, which means that the 13.1 mile per hour workload is a post-anaerobic threshold workload, and thus it is fatiguing as demonstrated by the data in the previous slide. For the elite marathoner, you can see that the 13.1 mile per hour workload is slightly below their anaerobic threshold, which means it is a pre-anaerobic threshold workload, and thus non-fatiguing. So obviously, the elite marathoner will be able to not only achieve 13.1 mile per hour, but sustain it for a prolonged duration until they experience exhaustion, until they run out of fuel, in other words. And the thing is, this 13.1 miles per hour is right below their anaerobic threshold. And that is the point for marathon performance, right? They want to run at a pace right before their anaerobic threshold intensity, because that is the maximum workload that they can sustain without experiencing fatigue. This is also referred to as your maximum steady state workload. It's the maximum workload that you can sustain without fatigue. Maximum steady state workload. So for these elite marathoners during that test, that 13.1 miles per hour, since it was slightly below the intensity of their anaerobic threshold, you could say that 13.1 miles per hour is their maximum steady state workload. Now, you could also perceive this comparison between the average Joe and elite marathoner as a comparison within a single individual from pre to post training. So say you were an average Joe at one point, and then you trained for however many months and you became a competitive marathoner. So view the left side as your pre-training curves and the right side as your post-training curves. The difference between these two figures reflect the changes you could expect with training. So to reiterate, with proper training, the 13.1 mile per hour run can become quote unquote easier and perhaps less fatiguing or even non-fatiguing because of an improvement in aerobic power, which again is a function of anaerobic threshold and aerobic capacity. Now, as a recap question, if improvements in aerobic power is critical to experience improvements in endurance and fatigue resistance, which of the following measurements must improve? The answers would be lactate or ventilatory thresholds, which are measures of anaerobic threshold, and VO2 max, which is a measure of aerobic capacity. And as a side question, Which of the following measures of aerobic power would an elite marathoner expect to improve with training? It would be the measures of anaerobic threshold, which again include lactate or ventilatory thresholds, because again, their VO2 max has likely already reached its peak. So let's talk a little bit more about aerobic power. Again, aerobic power is a function of aerobic capacity as well as anaerobic threshold, and thus a function of VO2 max and lactate or ventilatory thresholds, respectively. As I emphasized moments earlier, anaerobic threshold becomes more and more the critical physiological measure of aerobic power as you become more well trained. And this is because there is a genetically determined ceiling for how much your VO2 max can increase, and therefore how much your aerobic capacity can increase. So as demonstrated in the content in the previous slides, the key to improving performance or endurance, in other words, is to be able to stay below the anaerobic threshold with higher workloads, so that you can achieve these higher workloads and also sustain them for a longer duration due to the absence of metabolic stress and thus fatigue. This concept is reiterated in bullets 4 and 5. And again, to address the fundamental aspect of endurance training or conditioning programs, in order for one's anaerobic threshold to improve and thus aerobic power to improve, the training intensity must elicit the appropriate stress, which again is that hypoxic/metabolic stress during post-anaerobic threshold exercise. This means that during your training, there needs to be frequent, high-volume post-anaerobic threshold exercise bouts so that your muscles are frequently exposed to this metabolic hypoxic stressor. Now, because of the frequent stress imposed on the muscles, there is a reason or stimulus for your muscles to undergo adaptation. And those adaptations are again collectively termed aerobic adaptations, which makes a given workload that was once post-anaerobic threshold and hypoxic into one that is pre-anaerobic threshold and non-hypoxic. This is achieved through an improvement in aerobic power, which includes an improvement in the anaerobic threshold intensity. Now, you can think of the anaerobic threshold as the red line on a tachometer. If a car engine is pushed past the red line, the engine will undergo a lot of stress. Now imagine over time, as you keep running that engine past this red line day in and day out, the engine starts to get stronger and stronger, and it adapts to the stress frequently imposed on it. And that red line eventually increases from 7,000 RPMs as shown here to say 8,000 RPMs. This means you can now run your car engine past 7,000 RPMs and up to 8,000 RPMs without any stress. This analogy illustrates the concept of endurance training or conditioning. The anaerobic threshold is your muscle's red line, and if you exercise at a workload or intensity past this red line, there is a hypoxic stress, and that intensity or workload is fatiguing. Now, as you continue to train at intensities at or above your red line, imposing frequent stress to your muscles will eventually adapt and increase your red line so that you can do more work without stress and thus fatigue. So if I ask the question, which of the following is required for a training program for one to experience an increase in aerobic power, which includes anaerobic threshold and aerobic capacity, the answer would be frequent exposures to hypoxic stress. Other potential answer choices that would essentially say the same thing would be anaerobic threshold to post-anaerobic threshold exercise bouts or fatiguing exercise bouts. So by now, you should be very clear that any improvements in endurance performance and fatigue resistance is due to solely aerobic adaptations, which improves aerobic power. So even if you're doing so-called anaerobic training, quote unquote, which people often describe training like high intensity to sprint interval training, improvements in endurance and fatigue resistance is again due to aerobic adaptations and improved aerobic power or fitness. So to bust some common myths out there that even strength and conditioning professionals disseminate, there is never a situation in which the anaerobic system is isolated as depicted in the figure to the left. There is no situation in which the anaerobic system can quote unquote improve to enhance performance. The changes from left to right is what many people believe contributes to increased performance with quote unquote anaerobic training, with training highly above the anaerobic threshold, in other words. This type of change does not happen because this is not how aerobic and anaerobic systems function. There is no such thing as anaerobic fitness or enhancement in anaerobic metabolism. As shown in the bottom left figure on slide 4, if you are more reliant on anaerobic metabolism to meet your ATP needs, then that means you are out of shape. That means your aerobic system, your primary bioenergetic system, is not powerful enough to meet ATP demands at higher workloads. So the correct perspective of the adaptations that occur in response to your so-called quote-unquote anaerobic training is reflected here as shown in the previous slides as well. Again, many people consider exercise post-anaerobic threshold as quote-unquote anaerobic exercise, but as you can see, it is very, very aerobic. Only a portion of the ATP demand is fulfilled anaerobically. Anaerobic systems, again, only support the aerobic system when the aerobic system does not have enough oxygen or power to meet the full ATP demand. So yes, even when you do max intensity exercise training and sprint interval training or CrossFit or max effort circuit training, the improvements in endurance performance and fatigue resistance is again due solely to improvements in aerobic power as shown here from left to right. So if you do sprint interval training and you notice that you are more fit and able to do more work without fatigue, do not attribute it to quote unquote anaerobic fitness or anaerobic adaptations. It is due to aerobic adaptations, which improve aerobic power, which include an improvement in anaerobic threshold and aerobic capacity. So is there any research data and evidence that is currently published that supports this? Yes, in fact, there is. There is a lot of evidence, and this figure here summarizes results of numerous studies that have tested the effects of maximum effort sprint interval training programs on aerobic power, more specifically on aerobic capacity, as measured by VO2 max. So if the myth, as presented 2 slides back, was factual, then sprint interval training would not have any impact on aerobic variables like VO2 max. But what does all the research studies show? Now, these are the results of 38 trials across 34 studies which examine the effects of sprint interval training on aerobic capacity or VO2 max. The red bars represent sprint interval training programs that included 20 to 30 second sprints with 2 to 3 sprint repetitions. The gray bars represent sprint interval training programs that consisted of 10 to 30 second sprints with 4 to 6 sprint repetitions. And the white bars represent sprint interval training programs that consisted of 10 to 30 second sprints with more than 6 sprint repetitions. And what does each study demonstrate? Well, each study showed that sprint interval training improved aerobic capacity and thus improved aerobic power, which indicates that sprint interval training induces aerobic adaptations. The analysis of all these studies show that the greatest improvements in VO2 max or aerobic capacity resulted from longer sprints with low volume or repetitions. However, only a few studies tested this type of sprint interval training program. Most studies, as you can see by the number of gray bars, were done on 10 to 30 second sprints for 4 to 6 sprint repetitions. So although longer sprints of lower repetitions show the greatest improvement in anaerobic capacity, the strength of evidence was greatest with moderate repetitions of 10 to 30 second sprints. So if I ask the question, which of the following sprint interval training programs have the greatest strength of evidence in terms of its effects on improving aerobic capacity, the answer would be 10 to 30 second sprints with moderate repetitions such as 4 to 6 sprint repetitions. However, overall, generally speaking, across all these studies, we can clearly see that low volume training using maximum intensity exercise can be an effective method to elicit aerobic adaptations and thus improvements in aerobic power and thus endurance performance, being able to achieve and sustain greater muscular work due to the mitigation of stress and thus fatigue. Now, there is even a growing body of evidence that demonstrates these types of effects on aerobic capacity with high-intensity resistance training, such as CrossFit, for example. Now, from a practical perspective, this may be of benefit for many people as it is a time-efficient way to improve aerobic fitness. Now, going back to the fundamentals, Why would sprint interval training elicit aerobic adaptations to improve aerobic power? It is because sprinting is maximum intensity and thus as far above the anaerobic threshold as possible. And thus, sprint interval training involves repeated exposures to the most hypoxic stress in the muscles that are involved in sprinting. Remember, stress equals adaptation. So sprint interval training, more specifically sprinting, would ensure that your muscles are under the appropriate stress to cause adaptation and improvements. Now, here's the thing, because sprinting is so fatiguing, repetitions are very short in duration. And this is why you need to do multiple intervals of sprints to expose the muscle to the hypoxic stress sufficiently to induce adaptations. For someone who is very untrained, you could expect improvements in aerobic power even with doing one all-out sprint per day. But as you become better trained, we all know that training must be progressive and thus modifications must be made, such as increasing the volume or reps of each sprint interval training bout. Now, if you're wondering how long the rest intervals were between each sprint repetition, it was roughly about 5 minutes, and it was active rest. So they were just doing pre-anaerobic threshold light exercise in between each sprint. Okay, so now you understand that in order to experience aerobic adaptations and improvements in aerobic power, your training must impose sufficient and frequent stress, and that stress is experienced during post-anaerobic threshold exercise. So what types of training would be optimal for improving aerobic power and thus endurance? Well, generally, training can be described based on the relationship to the anaerobic threshold. So pre-anaerobic training is steady state training, meaning you can sustain the level of intensity continuously for a prolonged duration because there is no fatigue. So far below the anaerobic threshold intensity, you have low intensity steady state training. And low intensity here is about 40 to 50% VO2 max or heart rate max. Now, keep in mind the workload at a particular intensity is relative to the individual. For example, 50% VO2 max intensity for you may be a 5 mile per hour jog, while 50% VO2 max intensity is a 2 mile per hour walk for another less fit individual. So please keep that in mind. Next, you have moderate intensity steady state training, which is roughly about 50 to 60% VO2 max or heart rate max. And again, this is steady state because you are still way below your anaerobic threshold. This is not fatiguing exercise. Then you get to intensity right below or at your anaerobic threshold. This is on average about 60 to 70% VO2 max or heart rate max. Now, of course, the anaerobic threshold intensity differs among individuals, but this is just an average figure here. Now, again, this is referred to as high intensity steady state training. The exercise bouts are still steady state because it is not exceeding the anaerobic threshold, but you do have some metabolic stress to your muscles at these intensities. Now, as you train above your anaerobic threshold, the exercise bouts go from steady state to intervals. Why? Because since the exercise bout is above the anaerobic threshold intensity, it is not sustainable due to the presence of hypoxia and thus fatigue. Therefore, the exercise bout is split into post-anaerobic threshold intervals of exercise, separated by intervals of rest or pre-anaerobic threshold exercise as a recovery interval. So right above the anaerobic threshold intensity, you have high intensity interval training or HIIT training as most of you refer to it as. And this type of training involves exercise intensities at around 70 to 80% VO2 max, which for the average individuals is slightly above the anaerobic threshold. Again, these relative intensities differ among individuals depending on the level of fitness. I'm just presenting norm values here, in other words. Also keep in mind, I will not ask questions on the exam regarding the relative intensity as a percentage of VO2 max or heart rate max for each of these training modes, since again, it is all relative to the individual. So as you get further above the anaerobic threshold and closer to maximum intensity and thus closer to your VO2 max or aerobic capacity, you have max effort interval training, otherwise known as sprint interval training. This type of training incorporates exercise bouts at a relative intensity of 90 to 100% VO2 max or heart rate max. So basically it is all out efforts during the exercise intervals. Now, the exercise intervals within sprint interval training bouts would obviously be shorter compared to high intensity interval training because they are much more fatiguing. Now, as shown in the studies presented in the previous slides, sprint intervals can be anywhere from 10 to 30 seconds, followed by a rest or low intensity interval, usually around 5 minutes. So, with everything that has been discussed thus far regarding the fundamentals of training adaptations, which of the training modes listed here would be optimum for aerobic adaptations and improvement in aerobic power? It would be the training modes that are at or above the anaerobic threshold. So, anaerobic threshold or post anaerobic threshold exercise bouts. These training modes, since they are at or above the anaerobic threshold, involve frequent exposures to hypoxic stress to the muscles involved, and thus there is a stimulus for change, for adaptations, which are collectively referred to as aerobic adaptations. Now, many people believe that in order to be more aerobically fit, you need to do low intensity steady state to moderate intensity steady state, super long duration exercise bouts. However, that is not the most optimum approach because there is low exposure to hypoxic stress to the muscles, and thus the stimulus for adaptation is minimal. So, to improve endurance and thus fatigue resistance by aerobic adaptations, you must train at or above your anaerobic threshold intensity. And as you get in better shape, you must adjust your workloads so that you stay at or above your anaerobic threshold. For instance, say your anaerobic threshold at pre-training was at a workload of 6 miles per hour. You performed high intensity interval training with an average workload of 8 miles per hour for 4 months, and your anaerobic threshold improved to a workload of 9 miles per hour. So now the 8 mile per hour workload is insufficient for high intensity interval training because it is now a pre-anaerobic threshold workload, and it is too easy, in other words. It does not impose much stress anymore, and thus you won't adapt further and experience further improvements. So what do? Well, you adjust your HIIT workload to say 10 miles per hour so you are staying above your anaerobic threshold workload of 9 miles per hour. This is a simple example of progressive overload. So what do I mean by progressive overload? The concept of progressive overload and why it is necessary to experience continual adaptations and improvements is based on the general adaptation syndrome. The general adaptation syndrome describes the stages that an organism, like a human being, undergoes when exposed to a new stressor, such as exercise, and the changes that occur when that stressor is frequently imposed on the body. So, as I emphasized earlier, the most important aspect of endurance training to improve muscular endurance is to train at or above a post-anaerobic threshold intensity or workload. The reason for this is because only during these intensities would there be an appropriate stressor to elicit adaptations for the ultimate purpose of increasing aerobic power and muscular endurance. So what is that stressor associated with post-anaerobic threshold exercise intensities or workloads? It is the hypoxic stress that we discussed in part 1 that causes acute muscular fatigue. Remember, the presence of acute muscular fatigue is an indication that there is a stressor present since fatigue is the acute stress relief response. Fatigue is again a muscle self-inhibition for the purpose of relieving itself from this hypoxic stress by reducing the ATP demand from the aerobic systems and thus the oxygen demand. So remember, the adaptations are specific to the stressor because the whole purpose of adaptations is to minimize the stress from that stressor. So what type of adaptations will reduce the hypoxic stress and thus the fatigue associated with a particular workload that was once a post-anaerobic threshold workload? Adaptations that would improve the oxygen availability in the muscle, as well as adaptations that improve the aerobic system's ATP producing power. I am alluding to enhancements in the cardiovascular system as well as the muscle itself. More on this later. These adaptations would reduce the hypoxic stress and thus less fatigue would occur at a workload that used to be hypoxic and fatiguing. So as an individual begins a training regimen, they undergo stages leading to aerobic adaptations and thus improvements in muscular endurance as defined earlier. First, we have the alarm phase. During this stage, the first few exercise bouts that is at or above the anaerobic threshold is performed. These first few exercise bouts are at intensities that the individual is not accustomed to. Thus, these initial exercise bouts will induce what we call an overload stress. An overload stress is referring to that hypoxic stress associated with the post-anaerobic threshold exercise workload. Now, because the individual is not accustomed to this stress yet, meaning they have not adapted to this stress obviously causes fatigue and a temporary decrease in performance. This temporary decline in performance is referred to as shock and can be characterized with increased muscle damage, prolonged central and peripheral fatigue, and may require active recovery strategies. Now, naturally, as you continue your training at that particular intensity or workload, the body begins to adapt to it, first by compensation, which is essentially bringing your body back to baseline levels. At this point, no positive net gains have been achieved. As you continue your training and impose that overload stress continuously and frequently, your body will continue to adapt and eventually undergo what we call supercompensation, which describes the phase when adaptations have resulted in improved muscular endurance. And the workload that elicited the overload stress here is now less stressful and thus less fatiguing. It becomes easier, in other words. As you can see here, compensation and supercompensation is within the adaptation stage. So just to recap, if I asked, when a particular post-anaerobic threshold workload becomes less fatiguing and sustainable for a longer period, which of the following has occurred? If one of the answer choices is supercompensation, that would be the correct answer. Now, as the individual adapts and supercompensates, there will be a point when the adaptations sufficiently overcome the initial overload stress associated with your initial training workload. At this point, the overload stress is no longer an overload stress because you have adapted to it and have become accustomed to it. You got used to it, in other words. At this point, therefore, the exercise workload that once elicited this overload stress now does not elicit an overload stress, meaning it is too quote-unquote easy and does not provide much hypoxic stress and is not fatiguing. At this point, supercompensation stops, meaning adaptations no longer occur because that exercise workload does not provide enough stress to stimulate further adaptations. You could say that the hypoxic stress of a particular workload will diminish as you adapt to it. That is the whole point of adaptations after all. Now, this point when supercompensation or adaptation ceases is called the plateau stage. So to summarize a plateau, it is simply a situation in which the initial training workload that used to elicit an overload stress no longer provides a sufficient stress to stimulate further adaptations, to stimulate further supercompensation. Simply put, a plateau means you have sufficiently adapted to the initial training workload. And you have to understand that every athlete that undergoes training will experience this plateau stage. And it is the quality of the training program that will determine whether the athlete continues to adapt and therefore improve muscular endurance further. Now, from a conceptual standpoint, we know that in order to stimulate further adaptations, we need a new overload stress. The workload that elicited the initial overload stress for the initial adaptations or supercompensation is now no longer adequate to stimulate adaptation. And thus, something needs to be modified in the training so that an overload stress can be imposed on the body. This usually is achieved by simply increasing the exercise workload. So, for example, if a 5-mile-per-hour run workload caused the initial overload stress for the initial adaptations, and you have adequately adapted to that 5-mile-per-hour workload, and further improvements in muscular endurance have stopped, you would increase that workload to say 8 miles per hour so that you can reintroduce a new overload stress to stimulate further adaptations and improvements in muscular endurance. Now, many athletes who have poor training programs without any consideration of the general adaptation syndrome and without any progressive modifications may undergo a prolonged period of plateau, which is referred to as stagnation. And this could also lead into a decline in performance, which is referred to as overtraining. Now, a program that incorporates modifications to introduce new overload stress in a timely manner to avoid stagnation is described as a periodized program. Periodization simply means that there are intentional, systematic modifications placed within the program to maximize adaptations. So, as a new overload stress is imposed by progressively modifying the training program, for example, by increasing the training load from 5 miles per hour to 8 miles per hour, you would experience a new shock, just like you did initially when you were not accustomed to that 5 mile per hour run. This would lead to compensation, followed by supercompensation, and further improvements in muscular endurance. As you sufficiently adapt to this new 8 mile per hour workload, you would eventually experience another plateau, as you see here. And at this point, further progressive modifications must be implemented, like a further increase in exercise workload. Now, this concept of applying progressive modifications is referred to as progressive overload. The term overload is referring to the overload stress. Without an overload stress, there is no reason or stimulus for adaptation. And so as you adapt to an initial overload stress, you need to apply progressive modifications to continue to impose overload stress to the body to elicit continual adaptations and therefore improvements in muscular endurance. Now, this here is a training week of a sample conditioning program that incorporates exercise at or above the anaerobic threshold. Now, on Monday, you have your sprint interval training, which is small in volume but very high in intensity. On Wednesday, you have your high intensity steady state exercise, which is right at your anaerobic threshold or just below your anaerobic threshold. This again is the maximum steady state intensity, meaning the maximum intensity that you can sustain without fatigue. On Friday, you have your high intensity interval training bout, which incorporates intervals at intensities above your anaerobic threshold, but just slightly below your max intensity. You see, in this example, there's quite a bit of variation here, and this would be more appropriate for someone who is well trained as opposed to someone who is a novice or has been previously sedentary. But this is just an example of how a training week would look like in a periodized conditioning program. And as you adapt to these stressors imposed week after week after week, and these training bouts become essentially less stressful, you have to apply progressive overload to your training program to experience continual adaptations and improvements in muscular endurance. All right, so as I discussed, the training modes that are at and above the anaerobic threshold, as listed as these three right here, are the training modes that will elicit the appropriate overload stress required for adaptations. But what about the pre-anaerobic threshold training workloads or intensities? Does a low to moderate intensity steady state training elicit aerobic adaptations and improve aerobic power? The answer to this is yes. These pre-anaerobic threshold training modes can improve aerobic power, but to a limited extent, and I do mean limited extent, and only if it is sufficient in duration. Meaning a 30-minute low-intensity bout a few times a week will not induce significant adaptations, if any. For example, if you are a pretty well-trained individual and say your maximum steady-state running speed is 10 miles per hour, meaning 10 miles per hour is your anaerobic threshold workload, walking at 2 miles per hour for 30 minutes 3 times a week is not going to cause any improvements in aerobic power because there is no overload stressor. In fact, exercising at these lower workloads would likely cause de-adaptations and lower your anaerobic threshold. But what if you were doing a 2 mile per hour hike for 5 to 6 hours, say 3 times a week? Could this then help improve aerobic power? Possibly yes, but again to a limited extent. There is a reason why these low intensity workloads need to be performed for a prolonged duration to elicit some adaptations. And that is because fuel depletion is a stimulus for adaptation among other stimuli. So since low to moderate intensity steady state exercise depletes fuel, particularly muscle glycogen, at a comparatively low rate, it would take a longer period to have sufficient depletion. Now keep in mind that fuel depletion is only one of the many stimuli for adaptations, which we will discuss in detail in a few moments. Many of the stimuli for aerobic adaptations are not present with low to moderate intensity steady state exercise, as they are more associated with the hypoxic stress that occurs with post-anaerobic threshold exercise. but fuel depletion is one stimuli that is still possible with low to moderate intensity steady state exercise as long as it is prolonged in duration. So yes, if you are performing low to moderate steady state intensities and expect to experience increases in aerobic power or fitness, these exercise bouts need to be very prolonged, which can be impractical for many people. Many of us don't have hours within the day to exercise. As a concluding statement for exercise programming, anaerobic threshold to post-anaerobic threshold training modes would be the most optimal for improving muscular endurance. So for example, in those elite marathoners we introduced earlier, that 13.1 mile per hour workload was right below their anaerobic threshold. That means these marathoners would not benefit much from training at a workload at, like, say, 5 miles per hour, for example. 5 mile per hour training wouldn't be training at all for them since it would not cause enough stress or overload stress to stimulate any kind of adaptations. Now, as far as practical conditioning program principles are concerned, the number one factor is stress, particularly overload stress. Stress is why we adapt, more specifically, frequent stress. So if your training program does not impose that hypoxic stress associated with the post-anaerobic threshold workload frequently, then there will be no adaptations. There will be no supercompensation because an overload stress is required for that to occur. Now we have to understand that the desired adaptations is specific to the stress. So in the case of improving muscular endurance and thus fatigue resistance, adaptations such as improved oxygen availability and mitochondrial power are adaptations specific to reducing the stress of hypoxia associated with the given workload or intensity. These adaptations again will be discussed shortly in much more detail, but just know for now that the improvement in aerobic power, which again is improved anaerobic threshold and aerobic capacity, is a result of adaptations that occur to deal with this hypoxic stress of a particular exercise workload. This means that endurance training is really anaerobic threshold training or post-anaerobic threshold training, meaning training using workloads at or above your anaerobic threshold is how you introduce the specific stress to improve aerobic power, to improve muscular endurance. This means that in order to effectively impose the appropriate stress to the body, you need to know what your anaerobic threshold is. In other words, you need to know your anaerobic threshold intensity, such as a percent of your max heart rate, or anaerobic threshold workload, like running speed or cycling power, that is at your anaerobic threshold. Now, as indicated here, random training intensity selection will lead to random outcomes. Meaning if you select intensities without knowing where it is in relation to the anaerobic threshold, you wouldn't know what outcomes to expect. There are plenty of people who do high intensity interval training, but their high intensity intervals are pre-anaerobic threshold intensities. They just don't know it because they don't know what their anaerobic threshold intensity is. This means data on the individual's aerobic power, which again include aerobic capacity and anaerobic threshold, would be critical for training programming and intensity selection. So for aerobic capacity, we said the measurement is VO2 max, maximum oxygen consumption rate. However, VO2 max is not easily measured because it requires equipment, lab equipment. You need to perform a graded maximal exercise test while hooked up to a metabolic cart that measures VO2 to test your VO2 max. This means you need access to lab equipment, which most of us don't have. There are, however, procedures, more specifically field testing procedures, that allow you to predict VO2 max using heart rate instead, since heart rate and VO2 have a direct relationship. Many of the new heart rate monitoring technologies have algorithms to predict VO2 max and changes in VO2 max, but the reliability and accuracy of these devices are always in debate. As I emphasized earlier, the anaerobic threshold is one of the most critical factors of aerobic power. I mentioned that the two measurements of anaerobic threshold is the lactate and ventilatory thresholds. The lactate and ventilatory threshold tests are performed during a maximal graded exercise test. This test requires you to exercise, usually running or cycling, at multiple stages of increasing intensity. Lactate or ventilation will be measured at each stage, and eventually, as you increase intensity, lactate and ventilation will increase exponentially, marking the point at which the aerobic system requires anaerobic backup because the oxygen availability and the mitochondrial power is insufficient to fulfill the full ATP demand. This is when fast glycolysis, your anaerobic system, increases in activity and thus lactate production increases. This is when acute metabolic acidosis increases and ventilation increases to activate the bicarbonate buffering system. Thus, this is the point at your anaerobic threshold, and the intensity as a percent of heart rate max or VO2 max at your anaerobic threshold can be determined. Now, as you can tell, lactate and ventilatory threshold testing can also be impractical and requires equipment. Lactate threshold testing is also invasive as blood draws are required. However, knowing the intensity as a percentage of heart rate max, for example, at which your anaerobic threshold is observed, can be very useful if you want to train with multiple modes of exercise. For example, if your anaerobic threshold intensity is at 80% your heart rate max, you can apply this in cycling, swimming, rowing, running, circuit training, etc. to determine the training intensity for each of these modes as long as you can monitor heart rate. But what if you are just a single mode athlete who just cycles, for example? Is there another more practical route to anaerobic threshold testing? What if there is a test where you could simply determine the workload, such as cycling power, at which your anaerobic threshold is observed? In other words, your maximal steady state workload, the maximum workload you can sustain without fatigue. Luckily, there is such a test, and this is called the 3-minute all-out test. This test, as the name implies, is an all-out effort test, just like that Wingate test, to determine the maximal steady state workload, also known as critical workload or critical power, if the measurement of workload is power. This is your anaerobic threshold workload. When you figure out your critical workload, then you can determine the appropriate training workloads. Again, this is specific to the mode of exercise or task. So if you determine your critical cycling power using the 3-minute all-out test, you can't apply this to running or rowing. You would have to do the 3-minute all-out test for each mode of exercise to determine the anaerobic threshold workload for each mode. Now, this testing protocol has been recently popularized for athletes because it is simple and a practical way to determine the anaerobic threshold workload for a particular exercise mode. It also doesn't require hefty lab equipment. So let's go ahead and discuss the concept underlying the 3-minute all-out test. The 3-minute all-out test is based on the concept that when you perform exercise with a post-anaerobic threshold workload, fatigue will slow your muscles down to a workload that is slightly below your anaerobic threshold to reduce the hypoxic stress. Therefore, fatigue will essentially force you to bring your workload down to one that is without hypoxic stress and thus sustainable. During the test, the subject will cycle, run, or row at maximal effort for 3 minutes straight. Within about the first 150 seconds, depending on the subject, fatigue will bring the workload or power output down to the point that is not hypoxic. It will bring you down to a workload that can be performed at steady state, right below your anaerobic threshold workload. This again is referred to as your maximal steady state workload or critical workload / power. For this procedure, the average workload or power within the last 30 seconds of the test is used to calculate the maximal steady state workload, aka critical workload, aka workload at the anaerobic threshold. So let's say this is for cycling. The subject will be cycling with maximal effort and will ramp up the cycling power output to say 800 watts, which would be peak power. As you can imagine, the subject would not be able to sustain peak power for very long because of fatigue, caused by the severe hypoxic stress associated with maximal intensity exercise. So due to fatigue, the power output will decline. It will decline to a power output to say 300 watts. And this power output is sustained for the remainder of the test, even with maximal effort. So whatever the average power output within that last 30 seconds of this test is, that will be considered your maximal steady state power or workload, again also known as critical power. Now let's just say, for example, 300 watts was the maximal steady state power, and thus 300 watts is the workload at which your anaerobic threshold is observed. This means that if you're going to do high intensity steady state training, your workload should be around 300 watts. If you're going to do high intensity interval training, your workload for your high intensity intervals should be, say, 600 watts. And for sprint interval training, your workload for your max intensity intervals would be your peak power, which is about 800 watts. Now, you can apply this protocol to other modes of exercise as well, such as running and rowing, and this protocol is practical and can be done with minimal equipment. So, to summarize, the concept of the 3-minute all out test is for an individual to exercise at maximum all out effort, so at maximal intensity. And acute muscular fatigue caused by the severe hypoxic stress will force that individual to reduce his or her workload all the way down to a workload that is slightly below the anaerobic threshold. And this will occur towards the end of the test. And that last 30 seconds of that test, the average power within that time frame will be used to determine critical power, the maximal steady state power output or workload. Now, moving forward, let's go ahead and continue our discussion on endurance training adaptations, answering the question of what explains improved aerobic power with endurance training, meaning what is physiologically occurring that improves anaerobic threshold and aerobic capacity with training. First of all, many attribute the improvements in anaerobic power or fitness to changes in the cardiovascular system, and this is why conditioning is often referred to simply as cardio. In our exercise physiology course, we talked about the cardiovascular adaptations to endurance training, and these adaptations function overall to improve oxygen supply to muscles at a higher rate during exercise. But the thing is, it's not all about cardiovascular adaptations. That's only one piece of the puzzle. Conditioning or endurance training improves aerobic power, which again is an increase in anaerobic threshold and aerobic capacity, through multiple mechanisms, both in the cardiovascular system as well as in the skeletal muscle itself. This is why earlier I indicated that the adaptations to endurance training that contribute to improved aerobic power is a function of a multi-organ system adaptation. This is why we can compartmentalize the underlying mechanisms or adaptations to endurance training as cardiovascular adaptations and metabolic adaptations, all leading to improved aerobic power and thus muscular endurance or performance. Now, everything under cardiovascular adaptations we discussed in our exercise physiology course, so we're not going to spend too much time in this lecture series going over the details concerning the cardiovascular adaptations to endurance training. Briefly, as a result of endurance training and the frequent hypoxic stress imposed on the body, there is an increase in maximal cardiac output, which is the maximum rate of oxygenated blood flow pumping out from the heart. There is also an increase in muscle capillary density. If you remember your basic anatomy, capillaries are microvessels where oxygen exchange occurs between the blood and the tissues, like muscle. Finally, you have increased oxygen carrying capacity in blood and increased oxygen extraction by muscle. Now, altogether, these individual cardiovascular adaptations improve oxygen availability. This would contribute to an increase in aerobic power directly and also indirectly by increasing the ATP producing power of the mitochondria, which, as you know, is where aerobic metabolism occurs. Now, take careful note of this. These cardiovascular adaptations and the resulting increased oxygen availability enhances aerobic power specifically through the enhancement of VO2 max, or in other words, aerobic capacity, meaning the cardiovascular adaptations primarily impact aerobic capacity, which is again measured by VO2 max. And this should make sense because the cardiovascular adaptation increases oxygen availability for muscle. In other words, it increases the maximum oxygen consumption in muscle, which is measured by VO2 max. So if I ask the question, in which of the following ways do the cardiovascular adaptations to endurance training improve aerobic power, the answer would be an increase in aerobic capacity, which can also be stated as an increase in VO2 max. This means the cardiovascular adaptations to endurance training have only minimal impact on the other factor of aerobic power, which is anaerobic threshold. It doesn't mean it has zero impact on anaerobic threshold, it just means it has a little impact on anaerobic threshold. So what about the other side of this figure? Metabolic adaptations refer to the adaptations that occur in the skeletal muscle itself. So the skeletal muscle adaptations to endurance training are collectively referred to as metabolic adaptations, and they usually occur concurrently with the cardiovascular adaptations during training. Now again, these metabolic adaptations are changes that the skeletal muscles that are being trained are undergoing, leading to improvements in aerobic power. Now keep in mind, under metabolic adaptations, I have three major specific adaptations that occur. But just understand that there are multiple changes or adaptations that occur within the muscle in response to training. Arguably, the primary and most impactful metabolic adaptation to endurance training is mitochondrial biogenesis. And just like the term implies, this is a process of forming new mitochondria within the skeletal muscle cells, which we know is the powerhouse of the cell where aerobic ATP production occurs. So without overthinking it, if mitochondria are the aerobic powerhouses of the muscle cell, how would forming more mitochondria and increasing the overall number or quantity of mitochondria within the muscle cells impact aerobic power of the muscle cells? It would obviously improve aerobic power. Accompanying mitochondrial biogenesis is also an increase in mitochondrial size and density. However, we typically describe mitochondrial genesis as not only an increase in the number of mitochondria, but also in the size and density of each mitochondria, because all three of these outcomes occur together. So overall, mitochondrial biogenesis increases mitochondrial power, quote unquote, which is alluding to the ATP producing power of the mitochondria overall. And this improvement in the mitochondrial quote unquote power would directly improve aerobic power. Now, we will emphasize this further in the following slide, but mitochondrial biogenesis impacts aerobic power primarily by increasing anaerobic threshold and has just some impact on VO2 max or aerobic capacity. So if I asked on the exam which of the following factors of aerobic power is impacted most by mitochondrial biogenesis, the answer would be anaerobic threshold. Some other metabolic adaptations in skeletal muscle in response to endurance training include fuel supercompensation and buffering capacity. Now let's talk about fuel supercompensation briefly. Now remember, the term supercompensation simply just means more than what you had before. So fuel supercompensation is simply having more fuel stored and available in muscle. It is essentially the process of increasing the fuel tank within skeletal muscle cells. So what fuel molecules are we talking about? We are mainly talking about muscle glycogen, which is a stored carbohydrate molecule, and this is the muscle's most versatile and primary fuel substrate. If glycogen runs out, performance stops, and this is referred to again as exhaustion. This occurs despite having other fuel molecules available, such as fat or lipids. So increasing the amount of muscle glycogen stored within the skeletal muscle cells would obviously be conducive to performance and muscular endurance. It would also be conducive to aerobic power, but not so much directly as mitochondrial biogenesis. Remember, aerobic ATP production is not only dependent on oxygen availability and mitochondrial power, it is also dependent on fuel availability because you can't produce ATP without fuel. So in addition to glycogen supercompensation in muscle, you also have fat supercompensation in muscle, meaning an increase in intramyocellular fat or lipids. This has very minimal impact on aerobic power, but it can surely contribute to aerobic power because fats can only be oxidized aerobically. Fat supercompensation in muscle may be more beneficial for prolonged bouts of exercise as it can help preserve muscle glycogen. Now, more on this in a few moments. Lastly, we have increased buffering capacity, and this is mainly referring to the intramyocellular buffering system, which is the carnosine buffering system, which we discussed in part 1 of this series. As an adaptation to post-anaerobic threshold training or endurance training, especially at higher intensity, so for example sprint interval training, there is evidence that as an adaptation to frequent exposures to high metabolic acidosis, there is an increase in muscle carnosine levels. Now, this would not have any direct impact on aerobic power, but it can surely impact performance by improving sustainability of workloads slightly above the anaerobic threshold. So altogether, this figure demonstrates the multitude of adaptations, both cardiovascular and metabolic/skeletal muscle, that contribute to improved muscular endurance with training. And as a summary, the cardiovascular adaptation primarily contribute to increased oxygen availability and thus maximum oxygen consumption rate, which means it impacts mainly aerobic capacity as opposed to anaerobic threshold. On the other hand, the primary metabolic adaptation in muscle is mitochondrial biogenesis, which increases the ATP producing power of the mitochondria within muscle cells. This would have a direct impact on aerobic power by improvement of anaerobic threshold, with some impact on aerobic capacity or VO2 max. Now, as indicated earlier, we already discussed the cardiovascular adaptations in your prior exercise physiology course, and thus for the remaining portion of this lecture series, we will focus mainly on the metabolic adaptations, starting with mitochondrial biogenesis, which again is the primary metabolic adaptation to endurance training in muscle. So, to reiterate, mitochondrial biogenesis is technically an increase in mitochondria number, but this is accompanied always by an increase in mitochondrial size as well as density. So, what connects mitochondrial biogenesis with enhancement in aerobic power? Now, as we know, the mitochondria is the powerhouse of the cell and is where aerobic ATP production occurs. So it's simply an increase in the quantity of mitochondria. This would increase overall aerobic power in the cell because altogether there's increased amount of aerobic enzymes catalyzing aerobic processes such as the Krebs cycle and the electron transport chain. But also in individual mitochondria, there are positive changes which result in both an increase in size and density of the mitochondria. The increased size of individual mitochondria is attributable to simply more stuff inside the mitochondria, and the stuff are important to aerobic metabolism and the overall quote-unquote power of the mitochondria. Now, going along with an increase in mitochondrial size is an increased density of individual mitochondria. This means that within a given volume of mitochondrial space, there's just more stuff packed inside. The main contributor to an increase in mitochondrial density, as we will discuss in the next slide, are these inner membrane folds called the cristae, which is where much of the aerobic ATP production occurs via the electron transport chain. More on this in a minute. So overall, this figure here represents mitochondrial biogenesis. Not only do you see an increase in the number of mitochondria, you see an increased size in the mitochondria and also increased density of the mitochondria. Again, the size increase is attributable to simply more stuff related to aerobic ATP production contained within the mitochondria, thereby making it more powerful. And the increased density is attributable to an increase in the inner membrane folds, which is where most of the aerobic ATP production occurs via the electron transport chain, also improving the power of the mitochondria. So altogether, mitochondrial biogenesis improves overall aerobic ATP production in the muscle cells. This means the cell has the greater ability to process products such as pyruvate from glycolysis, NADH and protons from redox reactions, as well as ADP, inorganic phosphates, and free protons released during ATP hydrolysis. Now, in a few moments, I will provide a visual representation of this. But for now, just understand that with an increase in mitochondrial power within each individual muscle cell, there is less stimuli for fatigue at higher workloads, and this is why mitochondrial biogenesis primarily affects the anaerobic threshold. So if a question is presented with these two figures and I ask which of the following contributes to an increase in anaerobic threshold, the answer would be mitochondrial biogenesis. Or I could also ask this question in a more practical scenario. For example, at pre-training, a 10 mile per hour workload was slightly above the anaerobic threshold and thus fatiguing. After months of training, the 10 mile per hour workload is now pre-anaerobic threshold and non-fatiguing. Which of the following adaptations is the primary contributor to this outcome? If one of the answer choices is mitochondrial biogenesis, then that will be the correct answer. Now, if one answer choice was mitochondrial biogenesis, another answer choice was metabolic adaptations, and another choice was an increase in mitochondrial power, then the answer in this case would be all of the above, since all three of those answer choices refer to the same thing. Let's go ahead and discuss a little bit more about mitochondrial density. Remember, mitochondrial density is characterized by an increase in the inner membrane folds, again called the cristae. And this change in cristae in response to training, or rather the stress of training, is referred to as cristae plasticity. Plasticity is a term to just simply describe the ability to adapt. Like I said earlier, the cristae is where most of the aerobic ATP production occurs in the mitochondria via the electron transport chain. And thus, having more cristae in each individual mitochondria is indicative of greater aerobic ATP producing power of each of those mitochondria. We can understand cristae plasticity and how it changes with training by comparing the mitochondria between sedentary subjects and highly endurance trained subjects. Now, in this particular study, researchers obtained muscle biopsies from a group of sedentary obese individuals and a group of individuals that were cross-country skiers. And they compared the muscle mitochondria between these two groups of subjects. And as you can see in the images in the upper right-hand corner, you can see the mitochondria circled in red. Now, this type of analysis is referred to as histology, which is basically the analysis of cells or cellular structures like mitochondria using imaging technologies like a light microscope or electron microscope as used here. So the mitochondria of the sedentary obese individual can be reflective of the state of muscle mitochondria before training, and the mitochondria of the cross-country skier subjects can be reflective of the state of muscle mitochondria after training. Now, obviously, these differences are on an extreme end because you have two groups on the very opposite ends of a training spectrum, but differences you see here between these two groups can provide some visual of what you would experience as you engage in endurance training. Now, if you compare the mitochondria between the two groups, you can clearly see a difference. The lines that you see within the mitochondria are the inner membrane, and you can see that the inner membrane is much more abundant in the cross-country skiers versus the sedentary obese individuals. This means that the mitochondria of the cross-country skiers are significantly more dense than the sedentary obese individuals, and this is attributable to increased cristae density, meaning in the cross-country skiers, there is a greater density of inner membrane folds, which again is where the electron transport chain occurs. And therefore, this increase in cristae density is associated with greater aerobic power. Now, if you focus on the graph to the bottom left, the y-axis represents mitochondrial volume, so essentially the size of the mitochondria. And you can see compared to sedentary individuals, there is significantly greater mitochondrial volume in those who are trained. And part of this increased volume is the increased density of cristae. So how would these changes in the muscle mitochondria affect muscular endurance? How would it affect performance at a particular workload that was once unsustainable because of fatigue? Let's use this figure here that I presented in part 1 to describe the mechanism underlying acute muscular fatigue. Imagine at pre-training, running at 10 miles per hour was a post-anaerobic threshold workload and thus fatiguing. It was a workload that you could not sustain for very long. And this figure represents the state of the muscle during a 10 mile per hour workload. Before you started training, the mitochondria were too quote-unquote weak to handle the stress of this workload. Thus, there was increased proton accumulation leading to acute metabolic acidosis and inorganic phosphate accumulation, both of which lead to the inhibition of muscle contractions and thereby fatigue. Now, after a period of training, we know that one of the major metabolic adaptations is mitochondrial biogenesis, which not only involve an increased number of mitochondria, but more powerful mitochondria reflected by increased cristae density and size of mitochondria. So now, after some period of training, running at 10 miles per hour is a pre-anaerobic threshold workload and non-fatiguing, because now the state of the muscle cells look like this at this 10 mile per hour workload. There is less or no hypoxic stress, and thus no accumulation of protons and inorganic phosphates inhibiting muscle contractions. Why? Well, one reason is because of increased mitochondrial power. They are bigger, greater in number, and more dense because of more cristae density. The muscle mitochondria is now able to reduce the level of proton and inorganic phosphate accumulation during that 10 mile per hour run, and thus the 10 mile per hour workload is less fatiguing. It is now a pre-anaerobic threshold workload because the anaerobic threshold improved with mitochondrial biogenesis. Now, to dive a little bit more into mitochondrial biogenesis, I want to discuss the factors that connect training and the stresses of training to mitochondrial biogenesis. And if you look at this flowchart to the right, you would see certain factors that are associated with training, which again include low to moderate intensity steady state training that is prolonged, high intensity steady state training, which is at or slightly under your anaerobic threshold workload, high intensity interval training, and sprint interval training, which are your training modes that are significantly above your anaerobic threshold. Now, of course, the fundamental stimulus for mitochondrial biogenesis, along with all the other adaptations, is frequent hypoxic stress exposures. But what are other factors associated with this stress during training that stimulate the processes leading to mitochondrial biogenesis? One of the most potent stimuli for mitochondrial biogenesis is muscle glycogen depletion. This means that to optimize adaptation, there needs to be some level of fuel depletion present following each exercise bout. In other words, exercise bouts should have some level of exhaustion. Along with glycogen depletion, other stimuli include increased ADP/AMP levels, which occur with elevated ATP hydrolysis. Others include increased calcium flux, which is associated with muscle contractions that are repeated for a prolonged period. And lastly, increased epinephrine, which is associated with sympathetic activity, which occurs with exercise. Now, all these factors activate a particular enzyme in muscle cells called AMPK, adenosine monophosphate kinase. Thus, each of these four factors are considered upstream effectors of AMPK. AMPK is a multifunctional enzyme that regulates a lot of metabolic processes. But when chronically activated, like during training, AMPK can stimulate increased synthesis of mitochondrial proteins through a specialized molecule called PGC1 alpha. Now, I won't go into too much detail as to what PGC1 alpha is, but just understand that with increased activation of PGC1 alpha from AMPK activation, there's increased synthesis of mitochondrial proteins, which include aerobic enzymes, electron transport chain proteins, and other proteins that are critical for aerobic ATP production. Eventually, with increased mitochondrial protein synthesis, you get mitochondrial biogenesis. Now that we've covered the main metabolic adaptation to endurance training in mitochondrial biogenesis, let's now discuss the other adaptations, next being fuel supercompensation. Now, as mentioned briefly, the primary fuel source for muscle is glycogen, and it is one of the fuel stores that supercompensates with endurance training. But first, let's talk about intramyocellular fat supercompensation. Now, to preface this topic, I want to ask, between these two individuals, who has more fat storage in their body? The sedentary obese subject or the endurance athlete? Now, most of you will clearly say that it is the sedentary subject on the left that has more fat storage in their body. But the thing is, the sedentary obese subject may have more adipose mass, but similar fat mass as the endurance athlete. Remember, fat is a fuel molecule. It circulates in the form of fatty acids, and it is stored in the form of triglycerides in various cells in the body, especially adipose tissue, but also in areas like muscle. In the sedentary individual, much of the fat is stored in adipose, and therefore they have large adipose tissue mass, as you can clearly see in the CT scan. But the endurance athlete may have just as much fat stored within the body, but just in different tissues, for example, like skeletal muscle. In fact, there are studies that show that highly endurance-trained individuals have increased fat storage within their body, but within muscular compartments as opposed to adipose when compared to sedentary obese individuals. And this has shown to be an adaptation in response to endurance training and is referred to as intramyocellular fat supercompensation, which again is essentially increasing fat storage within muscle cells. This just means you have more available fuel for your muscles to utilize during exercise that is localized to the muscle, that is immediately available in the muscle. Meaning the muscle does not have to rely on fat being broken down in, say, adipose cells and circulated as fatty acids and having to be taken up by muscle cells to be utilized as a fuel. It can just utilize the fat fuel molecules that are directly available in its cells. Now, intramyocellular fat supercompensation doesn't directly affect performance through improved aerobic power, like mitochondrial biogenesis does, but rather it supports performance that is super prolonged, whether it's continuous, like marathon running, or intermittent, like, say, during a soccer match. And it does this by increasing the preservation of the muscle's primary fuel source, which again is glycogen. Fat supercompensation in muscle does not mean that there is simply more alternative fuel and therefore the athlete can perform longer. When glycogen runs out, performance will stop even if there is plenty of intramyocellular fat available. So in summary, intramyocellular fat supercompensation may improve performance during prolonged bouts of exercise by simply increasing the capacity to preserve or spare muscle glycogen, which again is the muscle's primary fuel source. So let's take a look at some evidence of this adaptive response to endurance training as it relates to intramyocellular fat supercompensation. Now, this study represented here is also a histological analysis of muscle cells, and researchers applied a lab technique called oil red O staining, which incorporates a chemical to the muscle sample that stains lipid molecules, particularly triglycerides. And after the stain is applied, the density of the color red is assessed, and the darker the red stain, the more indicative of greater intramyocellular fat content. So here we have images that are representative of their findings. On the left column here, we have two images from pre-training. Image A are stained muscle cells at rest, and image C are stained muscle cells after a single bout of exercise. Again, these are images obtained prior to the training program. The second column here are images taken after 6 weeks of endurance training. Image B is at rest, and image D is following a single bout of exercise. Now, generally speaking, we can visually observe a darker red stain in the post-training column versus the pre-training column, indicating that with training, there is increased intramyocellular fat storage. Now, where we find the darkest red stain is in image D, which again is post-training muscle samples after a single exercise bout. Now you can see in image D versus C that there is greater fat content within the muscle cells following a bout of exercise after 6 weeks of endurance training. This means that not only is there greater resting fat storage in muscle, but there is greater mobilization of fat to muscle during exercise. Essentially, what this data is indicating is that as an adaptation to endurance training, there is greater storage of fat in muscle cells, in addition to greater mobilization of fat to muscle cells during exercise. So overall, this data demonstrates the increased storage and mobilization of fat to muscle following endurance training, and again, this accompanies improvement in glycogen sparing or preservation, especially during prolonged bouts of exercise, whether continuous like during a marathon or intermittent like during a soccer match. Now, briefly, as I don't want to spend too much time on this, the glycogen preserving effects of fat supercompensation in muscle is connected to an increased capacity for fat oxidation, and this is driven not only by an increase in intramyocellular fat storage, but also increased fat mobilization from other tissues in the body like adipose, as well as increased mitochondrial biogenesis, since fat oxidation occurs in the mitochondria and involves aerobic metabolism. Now, the data shows that endurance training can increase fat oxidation rates in muscle, and thus there is increased ability to utilize fat as a fuel, especially at higher intensities of exercise. This again would benefit prolonged exercise performance through the preservation of muscle glycogen, and not so much through fatigue resistance or improvements in the aerobic power. So now that we've discussed fat supercompensation in muscle, what about the supercompensation of the muscle's primary fuel source, glycogen? So muscle glycogen supercompensation simply means an increased capacity for muscles to store its primary fuel, glycogen. Also to reiterate, remember glycogen is a endogenous carbohydrate store. You can think of it simply as a bunch of glucose molecules, which is also an endogenous carbohydrate, bounded together to be stored up as a large glycogen molecule for later use, like during exercise. So as I described earlier, muscle glycogen supercompensation is a training adaptation. So it's a specific adaptation to training programs that challenge glycogen stores consistently, or exercises within a training program that have a high utilization of glycogen, which is almost any type of exercise that you might do that is high intensity in nature and exhausted to some degree. So as you can see here, muscle glycogen supercompensation is multifactorial. So as I mentioned before, this figure is just the underlying mechanisms of muscle glycogen supercompensation as a result of training. So the two factors that contribute to this is one, increased muscle glucose uptake, which would in turn increase glucose availability to form glycogen with. Number two is increased rate of glycogenesis. So if you don't remember this term, make sure you know it before the exam. Glycogenesis, formation of new glycogen. And so what's mediating this process of increased rate of glycogenesis is an increased glycogenic enzyme activity. What else this figure shows is that all this is largely mediated by increased insulin sensitivity, which simply means there is much more efficient interaction between insulin and its respective receptor, the insulin receptor. So with more efficient interaction, you have more efficient signaling. And these signals result in increased glucose uptake through the GLUT4 transport system and increased glycogenic enzymes, which again are the enzymes that catalyze glycogenesis. So all together, increased insulin sensitivity as a result of chronic training results in muscle glycogen supercompensation. So with that being said, it is clear that endogenous carbohydrate storage and availability is integral for exercise performance, in particular endurance performance. And with that, we also have to understand exogenous carbohydrate intake, or carbohydrate nutrition, in other words, is also very important, because the exogenous carbohydrates that you consume, say on a daily basis, influences how much glycogen is being stored within the muscle. So training only increases the size of the fuel tank, but the gas within the fuel tank, the fuel within the fuel tank, is what you consume, what you put into it. So carbohydrate nutrition is a very important aspect of sports nutrition and human performance science. So let's get into certain topics related to carbohydrate sports nutrition. One of the topics that I want to talk about is carb loading. So one of the reasons I want to talk about carb loading in this class is because I can bet that most of you, if not all of you, have heard about it, read about it, or even talked about it. It is quite prevalent in the athletic community. It is adopted by many athletes to fuel their muscles prior to competition. However, the thing is, it is quite often misapplied, and this is likely enabled by a very poor understanding of the science underlying carb loading and how it manipulates your muscles' metabolism/physiology. So let's define carb loading by looking at its intention or purpose. And as you can see right here, it is to acutely or immediately and temporarily supercompensate muscle glycogen, to temporarily and immediately increase the size of your glycogen fuel tank that we call muscle. Okay, so this is a temporary and immediate overfueling effect. Can we do this through what we call a carb loading strategy, nutritional strategy? The answer is generally yes, you can. So the next set of questions we want to answer is, how should you carb load and how does it work? In other words, how do you apply carb loading strategies and what is the science underlying those strategies? So I want to refer you to this clip right here. This is a clip from one of my favorite TV shows, The Office, and this is the main character, Michael Scott. And in this scene, he is slamming down a bunch of carbohydrates in the form of fettuccine alfredo right before a 5K. He is doing this with the mindset that this quote-unquote carb loading is going to fill up his fuel tank, his muscle, to maximize the subsequent 5K performance. Is this the way to carb load? Is this evidence-based practice? I think this goes without saying. No, it is not. So let's go ahead and clear up some misperceptions on carb loading, because this, although it is satire, it is not too far off from the perspectives that many people, especially in sports, have on how carb loading works and how carb loading should be applied. So in order for us to understand the proper application and implementation of carb loading, we first need to understand the science underlying carb loading strategy. Let's first take a look at how glycogenesis is regulated. What are the inducers or stimuli for glycogenesis, again, the formation of glycogen, and what are the inhibitors or suppressors of glycogenesis? So, what turns it on and what turns it off? When we understand this, we would have a better understanding of how carb loading interacts with this regulation of glycogenesis. So first, let's focus here. This is an enzyme called glycogen synthase, and you can sort of tell by the terminology what this enzyme does. So synthases are enzymes that catalyze reactions that synthesize something. Synthesize, for example, a large molecule. So glycogen synthase is an enzyme that stimulates the synthesis or formation of glycogen, i.e. glycogenesis. So glycogen synthase catalyzes glycogenesis. So as I said in the previous slide, glycogenesis is basically taking multiple glucose molecules, these are small carbohydrate molecules, bonding them together to form a large glycogen carbohydrate molecule that is stored within cells like the muscle cell. So it's not converting glucose to glycogen, it is taking a bunch of glucose, putting it together to form a large carbohydrate molecule, glycogen. That is what glycogenesis is. I just want to clear that up. So the activity of glycogenesis in a cell like a muscle cell is largely determined by the activity of glycogen synthase, in that when there's increased glycogen synthase activity, there is more glycogenesis happening and vice versa. So this is the target of regulation. So what are the things that regulate glycogen synthase activity and therefore glycogenesis activity? First, what is the suppressor or inhibitor of glycogen synthase and therefore inhibitor of glycogenesis? That is glycogen replenishment. So when the muscle cell is full of glycogen, the tank is full of fuel, this is going to inhibit glycogen synthase activity and therefore decrease glycogenesis. This makes sense because if the tank is full, there is no reason to put more fuel in the tank. So this is essentially saying we do not need more glycogen in the cell. So what is the factor, the regulator, that does the opposite? What factor increases glycogen synthase activity and therefore ramps up glycogenesis within the muscle cell? Well, it will obviously be the opposite of glycogen replenishment, and that is glycogen depletion, or when there's a significant amount of glycogen depleted within the cell. So, for example, after exhaustive exercise. So this would be stimulatory to glycogen synthase activity, which would then increase the rate of glycogenesis, formation of more glycogen. And this, again, makes sense, right? When you utilize your fuels, that automatically is going to trigger the mechanisms, glycogen synthase and glycogenesis, to put more fuel in the tank, more glycogen in the muscle cell. So one important detail that you must remember to understand carb loading from a scientific basis is that the more glycogen that is depleted, the greater the activation of glycogen synthase. This is the target of manipulation in carb loading strategies, meaning the initial part of carb loading before you actually load your cells with carbs is to actually deplete your muscle cells of carbohydrate stores. You have to deplete your muscles of glycogen. The reason you do that is to hyperactivate this enzyme, glycogen synthase. And in so doing, this drastically increases the potential for your muscle cells to form glycogen as long as glucose is available. So glucose will become available after you deplete your muscles of glycogen when you consume dietary carbohydrates. So when you consume dietary carbohydrates, this increases glucose circulating in your body, making it more available for the muscles to form glycogen with. And because glycogenesis potential is so high, because glycogen synthase activity is so hyperactive, you're going to have a whole lot of glycogen formation happening. A whole lot of glycogen formation happening to the extent where you may be able to overfuel your muscle cells with glycogen. In other words, acutely supercompensate glycogen, more glycogen than what your muscle's capacity for glycogen storage is. So with that being said, you now have a general understanding of how evidence-based carb loading strategies would work. As you can see, the best way to maximize muscle glycogen levels, say before competitions, is to initially deplete your muscles of glycogen. Now, there are two ways to do this. First, you utilize or deplete the existing glycogen within the muscle by doing exhaustive exercise. The other way you can do this is by restricting carbohydrates through your diet so that you don't provide substrate for your muscles to form glycogen with. Most carb loading strategies, as shown here, this is called a depletion taper technique, use a combination of both initially within this strategy. So as you can see in day 1, which is 7 days before competition, this is when the carb loading strategy begins. And day 1 is highly focused on depleting your muscle cells of glycogen. So like I said before, you could do that by exhaustive exercise. So in this case, this is just an example, 90 minutes exhaustive run to deplete glycogen stores. And then lower your carbohydrate intake. You don't want to go on like a zero carb diet. That's never really recommended for especially endurance athletes, but you just lower it relative to what you normally would consume. So here I just put moderate carbohydrate diet as an example. So you are exhausting your muscle cells of the glycogen, and you're not providing more carbohydrates or glucose to form glycogen with. So with that being said, all that is happening within your muscle cells is hyperactivity of glycogen synthase. Not a lot of glycogenesis is happening because there's no glucose or very little glucose available to form that glycogen with. So within days 2, 3, and 4, you start to taper down your exercise, hence the taper technique here. You taper down your exercise in terms of duration from like 90 to like 40 minutes a day. This is again an example. And you continue with your relatively lower carbohydrate intake, again, relative to what this athlete would normally consume. So the first 4 days is predicated on depleting the muscle or the muscle cells of glycogen. So from a scientific perspective, you would say that the first 4 days of this carb loading strategy is to hyperactivate glycogen synthase, priming the muscle for extreme glycogenesis when glucose becomes available from your diet. So that's where 5, 6, and 7 come into play. Days 5, 6, and 7 rather. This is when the actual carb loading occurs. This is when you consume a high carbohydrate diet relative to what you would normally do. And here you taper down from 40 to 20 minutes a day. So you're not utilizing the glycogen that is forming within your muscles at a very, very high rate. So you reduce, you taper down your exercise, the utilization of your muscles' glycogen. On day 7, you continue with the high carbohydrate intake, and you don't do really any exercise because you don't want to utilize the glycogen that is forming at a very rapid rate. So what the intention of, or rather the goal of this depletion taper technique, which is a common carb loading strategy, is by day 7 or right before competition, you have overfueled your tank called your muscle cells. You have overfueled it with glycogen because glycogen is the primary fuel source for exercise during competition in this example. So on day 8, just another detail is that you would obviously maintain your carbohydrate intake because this is obviously the day of competition. So you would want some additional carbohydrates introduced through your diet on the day of. But as you can see, conceptually, this is not reflective of what most people think carb loading is. It's not just about consuming tons of carbs. So obviously, if you already had a full tank of glycogen, and then the Thursday night before the football game on Friday, you just consume a crap load of carbs, you are probably not going to overfuel or supercompensate your glycogen stores. Why? Because you have done nothing to prime your muscles to form glycogen at a very high rate. In other words, your muscle cells do not have hyperactive glycogen synthase. So again, conceptually speaking, all effective evidence-based carb loading strategies should begin with some amount of days of glycogen depletion, again, through exhaustive exercise and or through moderation of carbohydrate intake. Okay, so depletion first, then you load it up. So that is the key to effective evidence-based carb loading strategies. Depletion taper technique is just an example, and you can set it up various ways. And this is, again, just an example. From a practical standpoint, as you can see, this is not going to work for a lot of athletes, especially team sport athletes that have frequent competitions throughout, say, the week or the month. So this would only work for certain athletes with less frequent competition. So this is why endurance athletes or track and field athletes adopt these practices, mostly endurance athletes, because they don't have frequent competition. They can afford to do a week's worth of carb loading. But we have to understand that carb loading is simply a technique for athletes to use to make sure that they have a full tank of gas before their competition. But what is most important is their overall nutritional programming, which heavily involves the manipulation of exogenous carbohydrate intake. So for any performance nutrition program to be effective for an athlete or an individual who is training, it needs to be goal-based, and it also needs to be prescription-based, meaning it has to have some objectivity to it. So with that being said, we have identified in the science 3 major variables for nutrition programming. Now, there's obviously other aspects of nutrition other than carbohydrate intake, but since we're on the topic of refueling and maximizing glycogen stores for an athlete, we're going to stick on the topic of carbohydrate nutrition. So the 3 types of sports nutrition variables that have been heavily researched are type, timing, and quantity. So specifically in relation to carbohydrate performance or sports nutrition, for type, we can talk about the different ways to characterize the dietary carbohydrate. So for example, you can characterize the dietary carb as complex versus simple. We can characterize it by its glycemic index, or just simply the source, the food source for that dietary carb. The other one is timing. This is very specific to performance nutrition. So for example, when do you consume those carbohydrates, or the dosage of carbohydrates relative, say, for example, your workout or competition, pre, mid, post, for example. Quantity is referring to how much carbohydrates do you consume, say, before and after workout or throughout the entire day. So let's talk about some of the literature, the scientific literature that covers these 3 factors that outline carbohydrate nutrition programs. So one thing we have to understand first is that carbohydrate nutrition programming, so how we manipulate the time, timing, and quantity, in other words, how do we program these 3 variables into a carbohydrate nutrition strategy, is completely predicated on this, on how to best optimize glycogen resynthesis. In other words, in more lay terms, how do we best optimize muscle refueling? So the research shows that muscle refueling, say on a daily basis and throughout a training program, can be influenced by the type of carbohydrates you consume, the timing of the carbohydrate intake, and the quantity of carbohydrate intake. So let's go ahead and dive into these. So one point of emphasis that we have to understand is that dietary carbohydrate intake will always be a key factor for muscle refueling or muscle glycogen replenishment. And therefore, it's a key factor when it comes down to performance recovery, because in practical sense, when someone is training, they're doing repeated bouts of exercise. So obviously, dietary carbohydrate intake, or the programming of dietary carbohydrate for an athlete, is going to be critical because the athlete is likely depleting muscle glycogen every single day or very frequently throughout the weeks. So to ensure that the athlete has optimal performance not only during training but also during competition, we have to make sure that their carbohydrate intake is optimized as well, for the sake, again, of muscle refueling. Okay, we do not want the athlete to have an empty tank anytime he or she is training or competing. So going back to the 3 variables, time, type, and quantity, let's get a little bit more specific. We want to take a look at total daily intake as part of the quantity variable. We want to look at post-exercise timing of carbohydrate intake, so after you deplete your muscles of glycogen to some extent, and the type of dietary carbohydrates. So again, this is all predicated on maximizing muscle refueling. So as you can see in this 3rd bullet right here, it says total daily intake is the most important factor for performance recovery following exhaustive exercise. This is the same thing as saying Total daily intake is the most important factor for muscle refueling, for muscle glycogenesis, and restoring muscle glycogen following exhaustive exercise. So, glycogen depleting exercise. Now, let's take a look at the scientific literature that uncovers the question of what this total daily intake looks like for the optimization of muscle refueling and therefore performance recovery. So we turn to what we call a position stand. So position stand is written by multiple researchers within this field who authored this paper and make a position on a particular research question. For example, what is the optimal total daily intake to maximize performance recovery in athletes who do exhaustive exercise? So within this position stand, it was stated that based on the literature and the research, 6 to 12 grams per kilogram of body mass per day would be the optimal range for carbohydrate intake, again, for the purposes of maintaining muscle glycogen levels to make sure that muscles refuel properly after training bouts. You can Google this position stand by just typing in some keywords. Type in carbohydrates, position stand, Kirksick et al., 2017. Journal of the International Society of Sports Nutrition, or JISSN. This is a free access journal, so you can pull up this paper and it's going to have a lot of good evidence-based practical information in there for you as it relates to carbohydrate nutrition strategies. So again, 6 to 12 grams per kilogram body mass per day for optimum recovery and maximizing muscle refueling. This is again only pertinent to athletes. This is not relevant for just your everyday individual. Clearly, this is above the RDA for carbohydrate intake. So I believe the RDA is about 130 grams per day. So if you think about a typical 80 kilogram individual, even at the lowest range, I mean, that's what, 480 grams of carbohydrates per day. That's significantly above the RDA. So make sure you communicate, you disseminate this information with context. You can't just say, I learned in school that you should eat 6 to 12 grams of carbohydrates per kilogram of your body mass per day, and say, oh, it's healthy for you. Oh, it's good for you. You have to put it into context. This is for athletes or for highly active training individuals who deplete glycogen frequently throughout the week. And this is, again, only predicated on maximizing performance to ensure recovery of your muscles' glycogen, replenishment of your muscles' glycogen. Okay, so if you're just an everyday individual, sedentary, maybe works out like 1 or 2 times a week, 30 minutes each time, this is not relevant for that person. So always keep that in mind. Make sure you maintain the mindset that everything needs to be disseminated with context. Now, moving forward, let's turn our focus to another important variable in carbohydrate performance nutrition programming, and that is timing. And as I mentioned earlier in the prior slide, I want to focus the details regarding carb timing on specifically post-exercise consumption. And the reason I would like to focus on post-exercise carb intake is because there's a prodigious amount of research that shows the positive impact of post-exercise carb intake on muscle glycogen refueling during recovery periods, in particular short recovery periods such as less than 8 hours, which would obviously be pertinent to individuals like athletes in the meat of their season or off-season training, or someone who is training recreationally with high intensity and frequency towards a particular fitness or performance goal. Now in this slide here, I present results of an examination of multiple studies that investigated the relationship between the amount and rate of carb intake post-exercise on muscle glycogen refueling rates during recovery. If you can turn to this graph right here, the y-axis, as you can see, represents muscle glycogen resynthesis rate, or simply muscle refueling rate. And the x-axis here represents the post-exercise carb intake dosage per hour in the units of grams per kilogram body mass per hour. So first, let's focus on the data points represented by the blue aqua color, as you see right here. Now, this study took the different carb intake dosage rates across multiple studies and plotted each one against the average muscle glycogen refueling rate observed in response to that carb intake. Now, this is again reflecting post-exercise recovery periods. So as an example, if you take this particular point right here, subjects who consume roughly around 0.8 grams per kilogram body mass per hour post-exercise exhibited a refueling rate of about 12 millimoles of glycogen per kilogram of muscle per hour. So when placing a best line fit on this plot of data points represented by the blue aqua color, we can see that there is generally a positive linear relationship between post-exercise carb intake dosage and rate and muscle refueling rate. So just to put this all together in summary, this analysis showed that within a 8-hour recovery window, which is relatively short, post-exercise carb intake greater than 1.2 grams per kilogram body mass per hour appears to be optimal for muscle glycogen refueling. And theoretically, this should translate to optimal performance recovery. So if we take a practical example of an 80 kilogram athlete, this athlete would consume carbs post-exercise at a rate of 96 grams of carbs per hour. Now, this can be in the form of simple carb sources like a sports drink, mixed with carbs from full meals or snacks. A little more on the type of carbs later in this lecture series. So keep in mind, this data represented by the blue aqua color only represents the refueling rate in response to consuming only carbs post-exercise, so with no other nutrients co-ingested. So the next question this analysis addressed was how muscle refueling is impacted by carbs when co-ingested with other nutrients, such as protein/amino acids. Now this data is represented by the purple color. So within each of these multiple studies, subjects consume carbs at varying dosages post-exercise together with some dietary protein. So it was a co-ingestion of carbs and proteins at varying amounts. Now the findings from this analysis were interesting because it showed that one would achieve the same muscle refueling with a lower post-exercise carb intake rate when it is co-ingested with protein. So specifically, you can achieve similar refueling rates with a carb intake of about 0.8 grams per kilogram body mass per hour with protein co-ingestion compared to a carb intake of about 1.2 grams a kilogram body mass per hour without co-ingestion of protein. So it appears there is some positive synergy between protein and carb intake post-exercise as it relates to muscle glycogen refueling. Now, this has been theorized to be largely mediated by more efficient absorption of glucose that comes from the carbs that you just consumed and muscle glucose uptake when free amino acids from the dietary protein that you consumed are also present. So just as a simple summary of this comprehensive analysis on post-exercise carb intake and muscle refueling for individuals like athletes with frequent training bouts or competitions throughout the week, with relatively short recovery windows, so a high need for rapid muscle glycogen replenishment. Here are some takeaways from this analysis. So post-exercise carb intake of 1.2 grams or greater per kilogram body mass per hour appears to optimize muscle glycogen refueling or replenishment during recovery periods. And this should translate to more efficient performance recovery. And this dosage is especially critical, at least within the first 4 hours post-exercise. Secondly, post-exercise protein intake of about 0.2 to 0.4 grams per kilogram body mass per hour co-ingested with carbohydrates may improve muscle glycogen replenishment when post-exercise carb intake is limited to less than 0.8 grams per kilogram body mass per hour. So protein co-ingestion with carbohydrate intake of greater than 1.2 does not appear to enhance muscle glycogen replenishment any further. So what this is ultimately saying is that co-ingesting protein with a lower carb intake of 0.8 grams elicits similar muscle glycogen refueling as higher carb dosages without protein co-ingestion. Now, this information right here presented in the second bullet or the sub-bullet is a bit more practically relevant since for most athletes, post-exercise nutrients consist mainly of protein and carbs. So in most cases, athletes would not need such a high dosage as 1.2 since they are likely consuming protein with their carbs. For example, Gatorade Sports Science Institute does some fantastic research on this, which ultimately shapes the design of their products. So you will see products that have a somewhat decent mixture of carbs and protein for their post-exercise nutrient products. Now, going back to the whole concept of staying within context, this information that we presented here in the last two slides is really not relevant for those who engage mostly in strength training, as typical strength training bouts are not very glycogen depleting types of workouts. So on average, muscles are still about 75 to 90% full of glycogen. Of course, this is just an estimate and would depend on so many different factors. But on average, most individuals engaged in a resistance training program would not need to go to extreme measures in terms of post-exercise and even daily carbohydrate nutrition. So all the data that we've presented so far is really pertinent to an athlete, someone who practices every day, someone who trains every day, someone who competes frequently throughout the week. So this is not to say that carbs are not important for those in resistance training. However, I don't want you guys to get that perspective from all of this. It is still important. You still use muscle glycogen, but you're not utilizing it on average to the same extent as a high performance athlete. So moving on to our next nutrition programming variable, let's take a look at the effects of dietary carb type on muscle refueling, especially during recovery periods. So this information here adds another layer to the timing details I discussed previously. So the question is, what type of carbs post-exercise would optimize muscle glycogen refueling and therefore performance recovery? So the study presented here focused on comparing two types of carbs classified by the glycemic index. Now, to briefly explain what that is, the glycemic index is a general measure, and it's a very general measure, of how fast blood glucose rises in response to consumption of a particular exogenous carbohydrate or a dietary carbohydrate. For example, a dietary carb that is high glycemic is generally one that is digested and broken down into individual glucose molecules at a relatively high rate, and therefore will be absorbed into blood circulation faster, creating something like a glucose spike, if you will. Now, examples of high glycemic carbs include foods with dextrose in it, which is basically another term for glucose, which would obviously bypass digestion since it's already glucose. So this would raise blood glucose levels very, very fast upon consumption. So as for low glycemic index carbs, these are typically larger carb molecules, often called polysaccharides, that generally take longer to digest and to be broken down into smaller constituents, glucose. Therefore, glucose would rise in the blood slower. Now, there is less of a glucose spike, if you will. An example of low glycemic carbs, which are also generally referred to as complex carbs, which is not too accurate of a description, but either way, an example would be carbohydrates found in foods like oatmeal. These large carb molecules are called again polysaccharides, while the high glycemic carbs like dextrose or glucose mentioned earlier is referred to as a monosaccharide. Okay, so let's first go over the experimental design of this study as shown on the left side here. At baseline, subjects came into the lab and underwent a muscle biopsy, and the muscle samples were used to test glycogen levels within the muscle. Now, the researchers ensured that the muscle cells were fully replenished with glycogen before the biopsy, and they did this by instructing subjects not to exercise for a few days and by consuming a high-carb diet. So next, the subjects underwent in the laboratory a glycogen depleting exercise protocol, which is essentially a run till exhaustion protocol. In other words, subjects were asked, aka forced, to run until they hit the wall. Afterwards, subjects were split into two separate treatment groups. One group consumed high glycemic carbs like dextrose, and the other group consumed low glycemic carbs immediately after this run. Total carb amount and timing was controlled between the two groups. Then a series of muscle samples in each group for each subject were collected during the recovery period, and muscle glycogen levels were compared between the two groups. Now, as you can see in this graph on the right, the group who consumed high glycemic index carbs post run to exhaustion had significantly greater muscle glycogen at any given time point during the recovery period. This implies that they had greater muscle glycogen resynthesis rates. In fact, within the 10-hour period post-exercise, the high glycemic group had near full replenishment of muscle glycogen, whereas the low glycemic index carb treatment had only partial glycogen replenishment. As you can see by this data and also other studies that demonstrate similar outcomes, high glycemic carbs had at least in the immediate post-exercise time frame would be optimum for muscle glycogen refueling. So far we know that one, daily carb intake should be roughly 6 to 12 grams per kilogram body mass per day. Two, post-exercise carb intake dosing should be around 1.2 grams per kilogram body mass per day, or 0.8 if consumed with protein. And three, immediately post-exercise, high glycemic carbs would be most advantageous. Again, these are evidence-based recommendations for the purpose of maximizing muscle glycogen refueling in athletes or those who train frequently and intensely and performs muscle glycogen depleting type of training. So with that as a summary thus far regarding carbohydrate nutrition and muscle refueling, let's continue on and explain why higher glycemic index carbs immediately post-exercise resulted in greater muscle refueling than lower glycemic index carbs. So as you can see here, the answer is greater insulin response. There is greater insulin response following the high glycemic index carb treatment because there's a greater rise in blood glucose levels. Remember, insulin secretion is proportionate to the level of glucose rising in the blood. So also remember, insulin is a hormone that not only stimulates glucose uptake, but also glycogenesis in cells such as muscle cells. So together with a rapid rise in blood glucose from the rapidly digesting and glucose liberating carbohydrate that you just consumed, there is much more glucose available in the muscle or for the muscle to form glycogen with, and greater activity of glycogenic enzymes to catalyze glycogenesis, and that is largely mediated by the insulin. Now, the last question we want to address in regards to carbohydrate nutrition and muscle refueling is, does consuming carbs immediately after exercise influence glycogen replenishment in comparison to a delayed consumption? So should you consume carbs as fast as you can post-exercise instead of delaying it for the sake of muscle refueling? That is the general question. So let's take a look at this experiment here. On the y-axis, you have muscle glycogen synthesis, and on the x-axis, you have two time frames post-exercise during which times muscle glycogen levels were tested. The red bar here represents the results for subjects who consume carbs immediately after exercise, while the blue bar represents subjects who did not consume carbs until 2 hours post-exercise. So immediate feeding, delayed feeding post-exercise. So when looking at the data during the 0 to 2 hour time frame here, the group who consumed carbs immediately post-exercise showed significantly greater muscle glycogen synthesis as the group who did not consume any carbs within this time frame. This is obviously an expected outcome because this group had no carbohydrate intake while this group had carbohydrate intake to refuel the tank. So what is interesting is that in the 2 to 4 hour time frame post-exercise, the group who consumed carbs immediately post-exercise, which was 2 hours prior to this testing time frame, had similar glycogen synthesis versus the group who consumed carbs within this time frame at the 2 hour post-exercise mark. So if you add these bars together versus these bars right here, you can clearly see that delaying your first carb intake post-exercise can negatively impact muscle refueling. So the takeaway is that there should be some carb intake immediately post-exercise to maximize glycogen refueling. Now, to layer on top of the info presented in the last slide, we now know that the immediate consumption of high glycemic index carbs post-exercise would be advantageous for refueling as opposed to delayed carbohydrate consumption post-exercise. So next, let's explain the mechanisms underlying this timing effect, explaining why consumption of high glycemic index carbs immediately post-exercise is most advantageous for glycogen refueling in the muscle. Now, this is a multifactorial explanation. So first, consuming high glycemic index carbs will result in a glucose spike in the blood, and this will be followed by an insulin spike in the blood. So, also, we know from exercise physiology that the cardiovascular system is going to adjust blood flow towards active muscles during exercise. And this increased muscle blood flow will also remain elevated post exercise for some time, especially in the immediate post exercise time frame. So, meaning immediately post exercise, muscle blood flow remains elevated. So not only do you have a lot of glucose and insulin circulating in the blood flow is circulating towards the muscles that were active during exercise. These are the muscles that are depleted of glycogen. So, in addition to this, the GLUT4 transport proteins, which are responsible for glucose uptake, are highly active in those exercised muscles because of the insulin signaling and also due to the fact that muscle contractions also stimulate these GLUT4 transporters. So, immediately post exercise, these GLUT4 transporters are very active in those previously exercised muscles. So, altogether, you have glucose and insulin spikes in the blood, you have elevated muscle blood flow, you have a lot of GLUT4 transport activity, and thus you have a prime situation for muscle glucose uptake as well as glycogenesis, glycogen formation. And these effects diminish with time after exercise, so the immediate post-exercise time frame is essentially a window of opportunity to consume your carbs to maximize glycogen refueling. So now this figure here simply provides a general visualization of how improper or inadequate carbohydrate nutrition during training can influence refueling over repeated bouts of exercise and ultimately performance, not only during training, but perhaps a future competition. Now the blue line represents proper carbohydrate nutrition with adequate carb intake, and the red line represents inadequate carb intake. And these little green guys right here represent exhaustive exercise bouts. And on the y-axis, you have muscle glycogen levels, and x-axis is a time spectrum. So as you can see by this figure, inadequate carbohydrate nutrition will have a negative effect on muscle glycogen levels over repeated bouts of exhaustive exercise. So over time, this will leave one with an empty tank after repeated sessions of exhaustive exercise. And this could also be limiting to one's performance during each of the subsequent training bouts. And also imagine if there was a competition or match here for this particular athlete. Not necessarily the optimum state for the athlete for competing, right? Because you have a depleted fuel tank. Now compare this to the blue line here. With proper carb intake, you have adequate glycogen refueling, and thus performance during each subsequent training bout could be ultimately optimized and lead to more efficient gains and optimum status for competition, especially if it was performed, like, say, for example, somewhere around here. Now, to summarize these effects for both adequate and inadequate intake scenarios, you can refer to the text right here. So, adequate or high carbohydrate intake will lead to appropriate insulin responses, which would maximize muscle glucose uptake and also be stimulatory to glycogenesis, which would maximize refueling of the muscle. And of course, this should be conducive to optimizing exercise performance. So thumbs up for that. So the opposite would occur with low or inadequate carbohydrate intake as described here. So thumbs down for that. And you can see here in comparison that with repeated training bouts, muscle glycogen levels become more discrepant between adequate and inadequate carbohydrate nutrition. And so if this is like athlete A versus athlete B, and all other factors that influence performance is controlled for somehow, athlete A will be in a lot better position for competition than athlete B. Okay, and this is all mediated by carbohydrate nutrition. So finally, the last slide of this lecture series. This is a summary of part 2. So first bullet is increased endurance performance or conditioning as a result of aerobic adaptations, which decreases reliance on anaerobic ATP production. Remember, anaerobic metabolism is simply a backup to aerobic metabolism when aerobic metabolism is too insufficient to meet the ATP demand of higher workloads. So there's no such thing as isolated anaerobic training. You just can't do that. that is physiologically impossible. And there's no such thing as increasing one's anaerobic fitness because you are doing some so-called anaerobic training. That makes absolutely zero sense, despite the fact that you will hear or read about it in some type of media or maybe from someone at the gym or someone you know. Second bullet, aerobic power. The two factors of aerobic power are aerobic capacity and anaerobic threshold. I think you guys should know that by now. This is a topic that was emphasized in part 1 as well. Cardiovascular adaptations primarily influence aerobic capacity. Cardiovascular adaptations are the main driver for an increase in VO2 max, which is the measure of one's aerobic capacity, maximum oxygen consumption rate. Adaptations that happen specifically in the trained muscle, so metabolic adaptations, these primarily influence anaerobic threshold. And the metabolic muscle adaptation that is the prime driver of an increase in anaerobic threshold with training is mitochondrial biogenesis. So obviously with an increase in anaerobic threshold, this is improving fatigue resistance, especially at a given workload that was previously hypoxic and post anaerobic threshold pre-training. So being able to do more work without fatigue, that's the whole point. With that being said, by understanding the mechanisms that drive an improvement in one's endurance or conditioning, these conditioning programs should be near your anaerobic threshold. This is so you can induce sufficient stress, the hypoxic stress, to stimulate adaptations to deal with that stress. So you need to give your muscles a reason to change. That is the most fundamental aspect of any type of training program. If you are doing highly pre-anaerobic threshold exercise, so low to moderate steady state exercise, it must be exhaustive, meaning fuel depleting, glycogen depleting. This is to provide enough or sufficient metabolic stress to elicit adaptation. This is referring to that AMPK mechanism, right? So because you're doing low to moderate intensity, you're not in that hypoxic environment. You're not near that anaerobic threshold. You're also not depleting fuel at a very high rate. So in order for it to be exhaustive to stimulate adaptation over time, low to moderate intensity steady state training needs to be prolonged bouts of exercise. Okay, so long duration. We're not just talking about a 30-minute jog. We're talking about hours and hours of continuous exercise per bout. May not be the most practical for your everyday gym goer or your fitness enthusiast.